Morning. I am Adam Dickin, one of the project managers at LATAT. I'd like to welcome you to the final Open Day workshop of the nTRACK Horizon 2020 project. Today, this event will focus on stem cell nanoimaging, and we've got 15 great speakers lined up for you today. And this event will be made up of 5 thematic segments. Firstly, we have the Horizon Europe projects, or Horizon 2020 project, should I say. Then we have nanomedicine, followed up by preclinical models and imaging. Then in the afternoon, cell therapy, and then finally the regulatory aspects. Just a note to all of our attendees today that this event will be recorded and that questions can be submitted using the Q&A panel on the right-hand side of the screen. I will then be raising the questions at the end of the segments to each of our speakers. And without further ado, I'd like to invite our first speaker, Dr. Mohim Mogimi, from Newcastle University, who will be providing our keynote speech for today.
Good morning. So thank you very much for inviting me to this very exciting conference. So I'm trying to give a talk today on general aspects of the pharmaceutical sciences, so in my own perspective in terms of where the pharmaceutical industry is heading to, and also how nanomedicine can really help with current developments, and particularly in terms of the genomics. And how we can actually address some of the problems that we have in the pharmaceutical industry. So let me share my first slide and try to give you a kind of a general perception of what is happening in Europe and in the USA. When you look at in terms of the pharmaceutical spending in both continents, we are spending huge amount of money on healthcare, and you can see, for example, in Europe, we are spending about 17% of the total expenditure on health is actually spent in buying drugs. And again, you can see that in the US is not that much different. And, for example, in 2016, in US, and they have spent in the region of about $3 trillion on healthcare. And again, a massive fraction of that money was spent on pharmaceuticals. But the problem is when you look at some of the highest grossing drugs in the US that has been prescribed to the patients, for every of these drugs that actually works on one single patient, you will find about 3 to 24 other people that the drug doesn't really help. For example, you take this drug, which is for the heartburn, it works on one person, whereas on the 24 other people is totally ineffective. And that actually explains why we're spending so much money on buying the pharmaceuticals, yet the majority of these drugs doesn't work on everybody. And that is the problem, that how do we address these and how do we make the whole process more cost effective? That is where the precision medicine comes into account, which is now is considered a kind of a paradigm shift in the healthcare. And technically, what we are saying is there is a genomic differences among different people, and the population heterogeneity technically arises from the variability in the genes, in the environmental conditions, and also the lifestyle. So that is why we need to understand how these gene variation actually is accounting for all these type of effects, and how we can actually use the drugs in a much more effective way in order to improve not only the healthcare, but also in terms of the saving the costs. So that is why we do see a huge developments in the genomic sequencing. And that is a kind of old slide. And when you look at, for example, around 2016, at that time, they have been able to sequence around 250,000 genes. Now we are in this period, And there are quite a lot of genes have not been sequenced. I don't know the exact figures, but in the next couple of years, it's estimated that in the region of around even 1 billion or 2 billion genes will actually be sequenced. But that is just not the problem, because if you do this sequencing, you also need to understand what are the functions of these genes and how these genes are actually participating in different pathophysiological conditions. But nevertheless, through these type of developments, you can actually see that the market is really growing, not only in terms of the clinical applications of the next generation sequencing, but the market is also expanding in other areas, such as in terms of the research application, in terms of the targeted sequencing segments, and even in terms of the ion semiconductor sequencing segment. So you can see that this market has been really, really progressing globally. And again, in the next few years, that, I mean, the actual market will be worth in the region of 15 billion or even more in terms of in dollars. But what can you really gain from the application of the genomics in terms of the precision medicine? So where we are involved, we're talking about the life sciences. So if we can understand the function of these genes, then that will actually improve the discovery of novel drug targets. It could actually improve recruitment of the patients for clinical trials, so we can actually select the right populations and give the right drugs and see how they actually perform. That also gives a new approach for drug repurposing and also for repositioning, and also can aid the development of companion diagnostics in order to check the biological barriers. Also in terms of monitoring the progression of the disease or the regression of the disease through this type of new drugs coming to the market. But of course, apart from that, the end user is going to be the patients, and that could actually help not only the patients, but also provide new strategies for the providers and also for the payers that I actually outlined in here. So in a sense, if this genomics is successful, and if we know the application of all these genes, then we do have a very, very strong tool in terms of not only finding new drugs, but also helping the patients in a very, very effective way. So this will also give a kind of a good motivation to the pharmaceutical industry, because as it stands, pharmaceutical industry is in trouble, and there are many challenges in facing to develop a new drug. I mean, everybody knows that the finding a new drug molecule is not easy, and on average, from every 10,000 drugs discovered in the lab or on a shelf, only few will get into the clinical trial, and if you're lucky, maybe one will actually get approval and get into the market. And that is actually increasing the cost of the research and the development quite to a large extent. We also have problem in terms of a high attrition rates because of the lack of validation of the novel therapeutic targets. And that is again where the genomic will play a very significant role in helping the industry how to repurpose or reposition the old drugs, or even finding type of new drug molecules. Again, the problem in industry is there are hardly any blockbuster drugs. I'm talking about the small molecules in here. And again, most of these drugs are actually what we had in industry are actually going off patent. And then there's a challenge from the generic and the biosimilars. And again, that is eating up a huge amount of revenue in industry. And apart from that, we have another serious problem, and that is called these after-launch problems. And these are the toxicities and untoward effects that actually appears after the clinical trial. For example, YX was one drug which was produced by Merck many, many years ago, and there are still litigations about this drug which has now been withdrawn from the market. And again, when you're in industry, you have to really market the drugs. There's an upsurge in marketing when there is a new drug, and also there is an increased demand for the lower drug prices by the national health and different kind of providers. So that is why the industry is in trouble. And actually that reliance on the genomics could be a kind of a way forward. Again, to just give you kind of an example of what actually happened in the industry in that period, and you can see that there are different type of companies in here, and you can see that in such a long period, the number of the approved drugs has been actually very limited, and that was to a kind of a great cost. You can see it varies from $3 to $11 billion for any of these companies to actually develop some of those drugs that actually got into the market. And that huge cost is just not only the cost of the development of that particular drug, but also absorbs the cost of the failures of other drugs that could actually not get into the market. So you can see it's a huge amount of the money spent by industry in order to get these drugs into the market. And that is where, again, the genomics could actually help and improve the revenue for these companies. That is not just the problem by itself, because the problem is that as the drugs are getting more difficult to be discovered and also to be developed, most of the small molecules are getting bigger and bigger, and they're becoming even more insoluble in order to formulate. So as a result, industry in some way has pushed towards biopharmaceuticals, has tried to develop kind of protein and nucleic acid type of medicines, and they're actually getting a huge amount of revenue from that side. But again, with those type of medication, you do have the problem of delivery, which again, in many cases doesn't really work. And so the bioavailability of these formulations are not really great. To just give you some example, for instance, drug delivery to the brain has been a massive problem, not only for the small molecules. For larger molecules is almost near impossible. So, and if you look at what industry has done, for example, one recent example has been by Roche, that they have tried to develop a kind of an antisense medicine for the Huntington disease. And in order to deliver these molecules, they have used a kind of an intrathecal route of administration, which is very tedious and is not easy, not applicable to a great number of patients. And during the phase 3 trial, just last March, they actually stopped the trial without giving any reasons. So that is one simple example of a large molecules, which has a huge effect in terms of treatment and management of a kind of a neurological disorder. We also have antibodies. Again, I mean, we all know that there has been huge amount of effort going into development of antibodies for management and treatment of Alzheimer's disease. And their new versions keep coming out. And again, a new version was recently approved by FDA. But again, people have been very skeptical regarding the efficacy of these type of medicines in management of the disease. So the questions are these formulations, are these kind of pharmaceuticals actually getting into the brain to sufficient quantities, or there is something wrong with understanding the principle of the disease, and maybe we are not really targeting the right pathways and right molecules inside the brain. So as a result of these problems, people are now focusing on adenoviruses and trying to improve gene therapy with adenoviruses. And again, there are many hurdles with these type of adenoviruses in terms of the immune reactions and safety, etc., etc. So that is why a whole range of efforts now being done in order to improve the delivery through a kind of a palatable route of administration, such as intravenous route. But again, if you are going to apply these type of routes, you have to be aware of many biological barriers that you have to overcome in order to deliver your, in this case, let's say, macromolecular drug to the target site. And also being able to release these type of the molecules in the right compartment in the cell. So it's not an easy task from a pharmaceutical development point of view. So that is where I don't have to explain anything about the nanomedicine, because you all know more about nanomedicine field. And that is where nanomedicine is trying to help, or the field of drug delivery systems, in order to at least formulate some of these medicines maybe the large molecules like the nucleic acids and nanoprotein-based medicines, in order not only to protect them against premature degradation, but also finding ways in order to cross the biological barrier and deliver them to the target cells. I mean, we have seen some development in this line. For example, the new vaccines such as the Pfizer and the Moderna are actually based on this type of developments. So as you know, nanomedicine, when the definition came out, was defined as the application of the nanotechnology for treatment, detection, monitoring, and prevention of the disease. And in 2005, we corrected that definition because nanotechnology is referring to manipulation of the atomic structure, but that doesn't apply to drug carriers like liposomes and polymeric nanoparticles, but that only applies in terms of the small size, which you are trying to manipulate the biological performance on the basis of the size and surface modification. But nevertheless, these are the many applications of nanomedicine today, not only in terms of the targeted drug delivery, but also in terms of drug synthesis, in terms of development of precision diagnostics, and also for development of biosensors and nanotools in terms of the cell purity, cell harvest, tissue engineering. And again, the majority of these applications are not appearing in eye medicine and eye labs, which you actually use even your cell phone in order to communicate this information or even sense some of these type of changes in the body. We do have a whole selection of nanomedicines in the market, and these are some of the widely used ones. You can see mostly the liposomes, micellar form, or some of the metallic one like the iron oxide. But this selection does not really help the precision medicine. So these are not designed in terms of the genomic science and in terms of using the genomic kind of data to deliver the drugs to the target site and help the patient. So that is the biggest problem now that is facing the nanomedicine, how nanomedicine can really help the genomic science and precision medicine. But nevertheless, what is important in terms of the design of the nanomedicine, whether you are going to just simply apply it for as a kind of a life cycle management of your previous drug, or in order to help the genomics and the precision medicine, you must consider two important facts. The first thing is you need to identify and translate the realistic opportunities which are offered by pathophysiological processes in the body in order to design a nanomedicine. So that is exactly the other way that I think the majority of people are actually doing. They're making a nanoparticle, they're putting a drug in there, then they're injecting it into the patient or into an animal to see whether they can find an application for it. That is not the way forward. You must understand what are those changes in the disease, what are those opportunities at the biological barriers which can be exploited in terms of a precision design and helping the patient in that way. So that is not only the prime condition, but you also have to consider the pharmaceutical manufacturing viability. So that means your nanomedicine has to be absolutely simple. And the structure must be very simple. It has to be reproducible, and it should satisfy all pharmaceutical attributes, not only from the material's purity and materials compatibility with the biological milieu, but also in terms of efficacy of drug loading capacity, being able to release the drug at the appropriate pharmacological rate, and also most importantly in terms of the scaling it up and in production of the cGMP materials. And when you consider these type of important criteria, the majority of the formulations at the academic level or in the academic labs are not really translatable for development of a pharmaceutical product. So that is why you also need to think from an early stage, when you do have those pathophysiological opportunities to design a carrier in the right way, then you have to choose materials that could satisfy the end product. And the other issue is, in my opinion, you also need to realize impact in different ways. Unfortunately, these days, all grant bodies like the Horizon programs or even national grants always ask you, what is the impact of your research? And I think this is nonsense to me, because what you need to actually understand is you need to ignore all these metrics which are actually applied by the political and commercial forces. Because if you go back many, many years ago, what we have today was just basically discovered on serendipity. Basically, people were interested in understanding the basic mechanisms, and they didn't even know what would be the impact of those mechanisms. And when you look at today, we have many of those examples that is actually carrying this field forward. So technically, breakthroughs are often punctuated by the kind of periods of discontinuity before changing the paradigm. So what we need to think about, what is the impact of the work that we're doing in terms of understanding the mechanisms, and the impact in terms of helping the society or the economy will follow anyway at some point, and we don't know when that will be. So that is why it's very important to actually pay detailed attention to basic research or to fundamental research and trying to understand the principles of, let's say, signaling or trafficking. And that is where you can actually design a delivery system that can really deliver its goal. So in a sense, what you want in terms of a drug delivery is you don't want anything smart. What you want is something really simple that you can actually load easily, release easily, you can produce in terms of the mass manufacturing, and is also reproducible. So that is the key message in terms of the development. Just to give you some examples of what we have been doing on those lines for the past 20, 25 years. So in terms of the development of drug delivery systems, we have already always considered a kind of a safe and simple by design approach. And our approach has been integrated from three different areas. First, we have actually applied a kind of a molecular bioscience and a kind of a nanoscience approach in order to understand how these materials or their individual components actually interacting with biological materials, whether at a kind of a single cell level or within a biological fluid, and what are those pathways and enzymatic processes that actually interfere with the function of a component of the material. At the same time, we have actually applied a systems approach to actually select the best materials in terms of a performance in vitro. And then on that line, we are working on a therapeutic axis, and we have been focusing on three specific areas such as cancer, the CNS area, and also in cardiovascular areas such as atherosclerosis, how to design right kind of nanomaterials or nanoformulations to address some of these conditions. And I will give you some examples of what we have been doing and how we think that will actually fill the genomic science in the future. The one area was the CNS, and as you know, a lot of people trying to make nanoparticles to actually get into the brain. And that is not only important for the field of drug delivery, which I don't think is, to some extent, is not going to really address the problems in neurological disorder, but that will only answer the problem in terms of delivering nucleic acid medicines, such as siRNA, mRNA, or the DNA, because at the end of the day, you want to manage for example, an Alzheimer's patient by giving, let's say, 2 doses a year, rather than giving these drugs so frequently every day or every other kind of week. That is not going to work. So you need to develop systems that can address the patients from, let's say, 2 administration of the drug, and you can manage them much, much better. And that can be only achieved with the nucleic acid medicines. And there are a lot of evidence that if you can deliver nucleic acid medicines to the brain, you could technically manage these diseases better. But again, that is not a simple task by itself, because you do need to understand the cell biology of the brain endothelium and exactly how the materials are actually transported. And that is where the antibodies have given a lot of information in terms of the affinity versus the avidity and routing inside the cell, whether you're actually going into the late endosome and lysosomes for degradation, or you can actually push the delivery towards the sorting tubules in order to induce transcytosis. And again, what comes into effect in terms of drug delivery development is how to actually change the deformability of the carrier system and also the size in order to accommodate in the sorting tubules. So by trying to understand these type of processes, we have recently developed a kind of a peptide system. So it's a kind of a phage-derived peptide. And what we have done, we have taken that single peptide and we tried to make a self-assembly into this type of core shell particles or a kind of a nanofiber, if you like. And that is technically a single component arriving from a phage display peptide. And through these different shape modification, we have been able to either target the transferrin receptor or the receptor for advanced glycation end product. And this type of material, when injected intravenously, within 15 to 20 minutes actually resides in the brain and not only crosses the blood-brain barrier, but also ends up in both neurons and microglia, which we have shown with extensive immunohistochemistry. And in terms of the proof of functionality, we have done the pharmacodynamic kind of studies by delivering functional siRNA, such as against beta secretase 1. And you can see the one single intravenous injection, we have been able to lower the level of the base 1 in the neurons in the brain by around 50%. And that is magnificent because we are actually giving a very low dose of siRNA intravenously with this system. That is lower than anything which have even been used for delivery to the liver in this case. But this is the brain that we are actually talking about. So it's a very effective system that we think that could actually help the genomics in the future. We have been working quite a lot in terms of the lymphatic targeting, and that goes back up to around 28 years ago, that we have actually shown that by careful modification of the surface of a nanoparticle in terms of an steric barrier, you can modulate the rate of release or the rate of drainage from subcutaneous injection site, and you can maximize the uptake in the lymph node. For example, if you have a thickness of around 1 to 2 nanometer, you actually can target the lymph nodes, and we're talking around 25 to 30% of your dose residing in the regional lymph node. But if you actually increase that thickness of a polymer coating, then you not only bypass the lymph nodes, you're having very fast drainage, and you're actually ending up in the systemic circulation. And we have applied this type of systems not only for lymph node targeting, but also in terms of the whole lymphatic vessel imaging and also delivering to the drug. In fact, the same principle has been applied recently by these COVID vaccines. I don't think they didn't know about the thickness of the PEG lipid in there, which is around 1.5 to 2 nanometers. But actually what that thickness does is actually helping those vaccine particles or the LNPs to actually drain very efficiently into the lymph node, which comes back to what we described about 20 years ago in terms of lettuce. Sometimes nano is not sufficient enough, so you can actually convert nano to micro, and that is what we have done in terms of the lymphatic targeting again a long time ago. So you inject your nanoparticle, and these are optimized in terms of the conformation of the polymer on the surface, but then you're attaching a targeting moiety at the end, and then you're co-injecting very quickly a type of a pentameric IgM or an hexameric IgM that in the lymphatic system, it causes agglomeration, and that increases the retention in the lymph node by maybe 5 to 10 folds. So by this approach, we have been able to deliver maybe 15 to 20% dose of liposomes that drain very fast from the injection site and retain them in the lymph node for treatment of cancer and other kind of pathological conditions. We have also developed kind of patterns, if you like, kind of between the large and the small microsphere, kind of a hexagonal pattern around with larger particles surrounded by a smaller one, and also cubical type of patterns which can be employed in terms of the tissue engineering. You can grow different type of cells and platforms depending what kind of functionality you have in here, or you can remove some of them later. And we have also designed kind of approaches having a parental large microsphere, and then on the surface of the sphere we have carefully attached this type of smaller one for a purpose of multi-vaccination and immune modulation. And we are still working on that, trying to optimize this type of delivery systems, which could again help precision medicine in terms of multi-vaccination, if you like. In terms of the bone marrow targeting, which in my opinion has been an area which has been overlooked by people in the field of drug delivery, again, going back to roughly 30 years ago, we have designed nanoparticles that could target the bone marrow, and in this case was targeting the bone marrow endothelial cells. And recently we have, that was a kind of a polymeric particle, and recently we have translated these findings in terms of design of lipid particles that very quickly after intravenous injection targets areas of active bone marrow. And then you can see that again on the tail. And again, if you look at on high magnification, you can see that the particles actually residing in active areas of the bone marrow. And again, that is very important in terms of the modulation of stem cell activities in the bone marrow and so on. Again, we have been able to design particles that selectively target spleen, again, in the sense of understanding how microcirculatory pathways actually work in the spleen and through the open circulation and endothelial cell sleeves. So by careful surface engineering, we have optimized the whole range of nanoparticles that within 12 hours, they target spleen to an extent of 60-70% of the dose with minimum retention in the liver. And although these particles are stealth, we have actually shown them that in the spleen, they will be taken up by the macrophages in the red pulp. And through that process, we even identified the receptors that in certain condition can recognize stealth nanoparticles. So in a way, we have simulated the parasite infection, the malaria infection in the red blood cells. These are the inclusion of the parasite, which are filtered around these endothelial cell sleeves and then taken up by the macrophages. So the system we have designed is very similar to what has actually been happening with the malaria parasite. We have also been addressing the safety of the nanomedicine because, as you know, that complement system has been kind of, suspected to play a very important role in infusion reaction, a term which is called CARPA. Again, we have shown that the CARPA is actually wrong, does not really contribute to adverse reaction, and those reactions are arising because of immune cell recognition of the nanoparticles, including the stem cell. And there are some subpopulation of these macrophages that react severely and release pro-inflammatory mediators within minutes of ingesting these type of nanoparticles. And we have overcome those problems, for example, by changing the shapes or even taking the spherical particles, which are problematic, and put them on erythrocytes. And then you can actually simply overcome these kind of problems of infusion reactions without even involving the complement system. So complement system has absolutely no role, in my opinion, in terms of these type of reactions. In terms of the patients, for example, you go back in the literature, you do see, this is a kind of a liver image from a thin colloid, which was done for identification of the tumors in the liver. But then in some patients, they have seen massive accumulation of these particles in the lungs. So that's a kind of a moderate retention, that is a severe retention in the lungs. And eventually it was discovered that this clearance in the lung is due to presence of some monocytes that actually home to the lung during the disease. And when you look at the infusion reactions with modern nanomedicine these days, and when we look at the profiling of the patients, you do see those reactions more frequent in patients that they had a liver disease or a lung disease. And these are the patients that they have a subset of these macrophages in their lungs, which are in direct contact with the blood. Unfortunately, the European Medicines Agency is still outdated, and they think the complement is involved, and they think that this should be tested. And in my opinion, that is a complete waste of time. It has nothing to do with complement. And yet you can use this type of diagnostic nanoparticles to look for patients. If you get the image in the lung, you can disregard them and you can eliminate them for this type of studies or clinical trials or even the medications. So again, you can see by proper understanding of the pathophysiology, you can actually help the genomics and personalization. And finally, in the last two slides, I need to say something about the CAR T cell therapy, which has been a massive development in the field of genomics and precision medicine. And CAR T cells are addressing a whole range of lymphomas, particularly the B cells, B cell tumors, and showing a lot of efficacy. We have also been working on those lines, and we have designed, for example, nanobodies. Nanobodies are a kind of a fragment of the antibodies from the camels and from the llamas. That is the VHH fragment, and we have been able to identify 13 clones of new antibodies against the HER2 antigen. And we have designed a kind of vectors that can transfect T cells and to make a CAR T cell therapy by representing a T cells with at least 13 different type of nanobodies on the surface, i.e. if you have, for example, an antibody against the HER2, and instead of having one, we have a polyclonal version which has 13 different fragments and recognizes different domains of the HER2. And through that, we have been able to actually control that. And now we are trying to translate that in terms of the development of quantum dots, each of them coated with one of those clones. And what we are trying to do is to take the tumor from a biopsies, and technically you can do a high throughput kind of experiments by slicing them and adding a mixture of these quantum dots, each of them covered with one of those nanobodies, to actually see the distribution of HER2 heterogeneity in the tumor as you go deeper into the tumor. And that will allow you to use the right combination of the VHH in order to optimize the CAR T cell therapy, or even using the VHH by itself. So again, that goes back again through the understanding of the genomic and pathophysiological opportunities that can be helping CAR T cell therapy. So I will stop in here technically, and just the last slide showing that how you can even optimize the CAR T cell selection. And again, we are trying to develop this type of microfluidics through microstamping and using conducting polymers. And we have this kind of micro scale type of platforms that you can actually retain the cells because they have the ligands that could interact with the receptors on the T cells. And once you have retained them, then you can train them, and then you can release them by putting an electrical current. So in this case, you only select the right T cells that you want to actually administer into a patient. So that is, I think, that the future, I think it is bright, but it has to be applied in a correct way. And I am sure that in the next 5 to 10 years, we will see massive developments in this field. So I'll stop in here. So thank you very much.
Thank you very much, Dr. Makimi, for really framing this discussion today and helping us understand the scale of the challenges that we're facing and also the hopeful message at the end. With that, we can now enter our first thematic segment, which is Horizon 2020 projects. Today, 3 projects will be being represented. That's StarStem project, iNanoBit, and then finally nTRACK project, of course. And our first presentation of this segment is LATAT's own Mark Massa, the technical coordinator of nTRACK project, who'll be giving us an introduction to the project and an overview.
Okay, thank you very much, Adam, and thank you, Wayne, for your nice presentation, and thank you all for attending this conference about stem cell imaging. So I'm going to go through this European project we've been going through for the last 4.5 years regarding the development of nano-living stem cell visualization and tracking, mainly for the cell therapy and muscle regeneration. So the the project responds to the need to see and to assess how the cells are behaving after they are just injected, they are going into the human being, since there's not a developed system for seeing and assess the viability and the biodistribution, if the migration patterns, and also to know if there is a kind of undesired effect of these stem cells, so they have the potential to develop eventually teratomas or whatever. So this is the starting point for this project. So the idea was, the starting point was to already some knowledge regarding the nanoparticles. So it was a iron core, gold shell nanoparticles covered with the glucose coating in order to facilitate the internalization by the cell. I cannot say the precise process since we don't know it yet. It's doses, whatever, in order to know if have this contrast agent, and also to be able to see and visualize by the regular systems of like a CT, MRI, and PET. But PET was, since it's radioactive, is part of an accessory activities. But so this is the concept behind. So this is the activities, and as you can see now, the idea what was behind is, okay, we have developed the nanoparticles that were just covered with glucose, and they proceed to be internalized into cells. And lately, you will see that one of the key points to demonstrate it's the location, the nanoparticle fate, since it's really relevant to know about the safety and the toxicokinetics of this nanoparticle, since it's not enough to demonstrate its efficacy, but also relevant to know how if there are a kind of adverse effects or some toxicity effects of these nanoparticles once they are into the cells, and these cells are just injected into the mouse or large animal models that we will see later, and then the effects, the regenerative effects of these stem cells are retained, even though they are being loaded by a large number of nanoparticles. In addition to that, one of the activities behind to this European project was, okay, we were just collecting these images, therefore we were able to develop activities regarding radiomics, so AI-based development of algorithms in order to perform and more precisely the single cell development, and also find the migration patterns. So if we go further to the next slide, so the concept behind of nTRACK is, okay, let's go for the nanoparticle synthesis and stem cell labeling. But at this stage, we were starting to thinking in three different ways. First, the escalation, the industrial escalation. The final goal was to be able to be ready for the first in human testing. The second one was, okay, let's go and use the stem cells that are quite advanced, and I will show you in a couple of slides later, that doesn't require more safety profiling, and to demonstrate that the cytokine effect or the paracrine effect of the stem cells are retained despite nanoparticles are intracellular located. And the third one was, okay, how the regulatory framework, how the regulatory authorities consider this new way of labeling cells as a drug or as a contrast agent, and to provide guidance from the very beginning of the project, where we go into which are specific studies to be performed in order to reach this initial goal to be ready for the first in human testing. Then we go for this characterization and functional analysis of these stem cells, loaded and unloaded, or labeled and unlabeled cells, and then set up a number of animal studies to reach the proof of concept in vivo in smaller rats, mice, and also with large animals in a musculoskeletal lesion model in sheep. So, and in addition to that, to perform some images analysis by CT, MRI, and PET, and then all these data to take advantage for the development of these radiomic activities, radiomic analysis, to be complemented as a part of this nano labeling of these stem cells. So the first, is what type of nanoparticles? As I said, it was the iron core, gold shell nanoparticles that will enable different kind of image modalities. And then there's a glucose coating to increase and to facilitate the cellular uptake of these nanoparticles. And then as a kind of Lego, just to add the radionucleotide or other things. And here's another key point that it was a huge effort from the consortium of this transition from the lab scale, that you can perform a number of strategies, a number of different modifications of these nanoparticles to a more regulated and more strict condition that also will be talked a little bit later during this conference, the scaling up. So the scaling up thinking into different perspective to generate different batches and to be compared these different batches and analyze these batches to be ready to be starting a clinical trial, but also to be considered that the regulatory bodies will be asking to perform this kind of activities or this kind of physicochemical analysis and different kind of quality assessment requirements. Then, as I already advanced, that the Plugistem is another Israeli company that was involved in this consortium, was just providing these stem cells, the placental derived stem cells that can be allogenic. They have already demonstrated that there's no immunogenicity behind. Actually, they are running several clinical trials. That means we took advantage of these features in order to avoid to repeat or to perform some safety studies just for the stem cells. That means we're just adding these nanoparticles and comparing and demonstrating that these stem cells remain stable, remain with the same functionality. We can escape from, we can just get rid of the safety profiling of the stem cells per se. And then the imaging setup, the proof of concept in vivo, because at the end of the day, what we wanted to demonstrate that we were just able to track, to enlight the cells by these nanoparticles in different models that could be relevant to this human disease, this muscle regeneration in different settings. So these are the techniques that we have been using, and different models in small and large animals. But also, in addition to that, we have to demonstrate for if in one side we were just thinking about the efficacy profiling, efficacy proof of concept, we also have to consider the non-GLP and NGLP preclinical nano safety. So the activities performed so far were just directed to demonstrate and looking for the worst scenario for those conditions in order to understand the potential toxicities that maybe can be found, and also to identify the best conditions for this therapy. That's why we perform several in vitro studies to identify the intercellular fate and if there is any kind of exocytosis of the nanoparticles, several pre-toxicity and toxicokinetic studies in rodents, and then performing that actually is ongoing, the GLP preclinical safety regulatory studies as well the safety assessment of the production, cell labeling, etc. For this. So then we have the full package, kind of a preclinical development, as it was a drug, since there were no previous ground regarding this kind of product we were developing. So I'm referring again to the regulatory context. So one of the activities that has been key for this project, it's been the regulatory strategy roadmap. It's been guiding, in a sense, to go for to reach the final goal, to be ready for the first in-human testing, and to get in touch with different consultations with the authorities, not only the EMA, but also another notified body, to say, okay, so we have this product, this is our plan, and this is our intentional use. How do you see if the assays are enough, or you require more studies to be approved to go forward into the testing? So let's go back a little bit about the safety profile of these nanoparticles. So we have prepared a battery of assays. As it was a regular drug, a new chemical entity, one new biological entity. So we performed the cytotoxicity, as you can see, the immune cells, the HP1, and also the PLX, the placental-derived stem cells. We also tested the immunotoxicity, the inflammation, and genotoxicity in a number of cell types. And so far, no relevant effects has been found. That means that the nanoparticles could have a kind of neutral effect once they are internalized into these stem cells. In addition to that, we go for in vivo with a number of endpoints to see, as I said, as it was a new chemical entity. So different clinical signs, the hematology, biodistribution, and the cross pathology, etc. And again, and we were quite happy about that. So no relevant, no observable effect has been detected. That means that, okay, you know that once you injected the nanoparticles, that some of them used to be accumulated in the liver, and we have found it, but not in high levels. The nanoparticles used to be accumulated into the spleen, but we already have found it. We also have found this, but it didn't cause any relevant indication of pathology or degree of toxicity. And I want to go back to the regulatory roadmap after the safety, since one of the, I used to talk as a kind of regulatory tour. We've been just jumping around saying, okay, we have this kind of technology, we want to develop this kind of nano labeling. And okay, as a development of stem cell therapies in general, right? But which are the potential uses? So for distinguishing responding, non-responding, or for monitoring the efficacy, all of them. But according to the experts, so we have to set a clear application, a clear definition, because different uses implies different regulatory roadmaps, and we could not go for different strategies. So finally, we selected the primary purpose of the nano labeling that will just drive us into specific studies and assays, but also specific communications with authorities, was just to monitoring cell therapy with labeled stem cells to be used during development and in non-clinical and clinical for new cell therapies. We aimed to be a kind of a technology to be ready to be used as a kind of a transversal or enabling technology for different kind of stem cells in order to respond to several questions. Where the cells go after the injections, do they live, do they die, do they remain? To have a fine-tuning for the doses is a critical point for cell therapy in a clinical setting, since, you know, the cost of productions, the price of the therapy can vary a lot if you're injecting a number of millions or another. And to have a clear picture about the areas that are being treated, since these cells could be visualized in different imaging technologies. So we quite recently, at the beginning of the project, we had a meeting with the EMA, with the Innovation Task Force meeting, the proposal to have a kind of a guidance to say, okay, you are the experts, we want to do that, this is our plan, and get some feedback, received some feedback, but it was quite unclear, it wasn't a clear positioning from the EMA, so we keep on going, and quite recently, at the beginning of 2021, we also have another scientific advice meeting, at that time with a notified body, and hopefully we got both the Paul Ehrlich Institute and BfArM that were combining for medical device and also as a new molecular entity, as a new drug, because during this time we did not receive a clear statement about the definition of what we were developing, since it was just defined as a kind of a borderline product. And by more details we provided by the in this afternoon, in this conference, so that we go more in detail about this point in terms of the definition, the borderline concept, and how we can just go through these authorities to get the approval. At the end of the day, is what we want. And next is the imaging studies, just to show you a small example how these images perform with SPECT and CT radio labeled. We can see this, that the cells and the nanoparticles remain on the injection site for a number of days. You can see on day 6 it remains, and also day 8 starts to fall signal, but it can be still be seen slightly on day 10. So somehow between a number of assays studies performed, this is an example. Okay, the signal remains, and also the cells. As a complementary studies, we set up a muscle injury model in sheep that, as far as we know, was quite unprecedented. So the starting point was the muscle injury model performed a few years ago in rat, was translated into a sheep model. Since the sheep model, the structure of the muscle sheep on the muscle of the sheep, it's really similar to the human ones. And also we would use human level imaging systems. So that means we ease the translation into human setting in terms of the protocols and the key features of these systems, and shortly will be submitted for publications. And here you can see also the similarity how this injury in rat, the gastrocnemius muscle, the area that's injured, is performed by a kind of trocar, just extracting a relevant piece of muscle. And here we can see against the injury area that the setup of this model was already performed in a quite nice way. And now I will be performing this imaging study with large animals to see to demonstrate the proof of concept in vivo in a large animal, and then to be added all this information for this scientific dossier to be submitted to the authorities if it's possible. And this is all we have to go through. This project started 4.5 years ago. It was including the pandemics that I think to all of us have been high impact, not only at work levels, but on any or on all levels. And this is it from my side. Thank you very much.
For that great introduction to the nTRACK project. Obviously, it's a lot of information of 4 years of project into only 25 minutes of presentation. I'd just like to remind attendees today that the Q&A panel to the right should be accessible. So if you have any questions for our presenters today, please just drop your questions there. Our second speaker for the H2020 project section is Dr. Mark Leahy from the NUI Galway. He's the Chair of Applied Physics and the lead of Tissue Optics and Micro Circulation Imaging Group. Today he'll be telling us more about the STAR-STEM project. Thank you for bearing with us. A small change in our programme whilst we get Dr. Lee connected properly. We'll introduce the next talk and then Dr. Lee can present afterwards. So I'd like to introduce Dr. Helena Alsisk, the Head of Innovation Strategy at CNT. She's also the Innovation and Innovation Manager of the nTRACK project, and today she'll be giving us some insight into the current trends in regenerative medicine.
So good morning, everyone, and thank you, Adam, for the nice introduction. So today I'm going to tell you a few words about Cambridge Nanomaterials Technology, who is the exploitation partner of the nTRACK project, and then I am going to tell you a few things about the current trends and innovation management in regenerative medicine. So Cambridge Nanomaterials Technology is an innovation management and technology strategy development consultancy company based in Cambridge. Our company helps other companies, universities, and government institutions to develop their nanomaterials-related technologies, their products, R&D strategies and partnerships, as well as to work on their IP and market strategies. CNT is also specialized in nanomaterials, in additive manufacturing, and gives advice on these technologies. As you will see later on, we are also very much involved in collaborative R&D projects. Since 3 years ago, we have also opened a sister company based in Brussels called CNT Innovation, and both of these companies are fully operational and eligible to participate in Horizon Europe projects and Horizon 2020. So these are the services that we are offering to our clients. We offer scientific and technology advice and strategy. We prepare market research and collaboration and investment strategy. We help partners in intellectual property development and strategy. We help in R&D innovation management. We perform consultancy and training services, and in European and UK projects, we are involved in the part of innovation, exploitation, and dissemination management. So at the moment, we have 10 Horizon 2020 projects running. You can see them here in the orange box. The projects that you see above are our previous projects. We have just recently started a new Innovate UK project, and we are signing a grant agreement at the moment for one of Horizon Europe projects. So all our projects are related to nanotechnologies, application in different fields. So we are involved in nanomedicine projects, but also in renewable energies and energy, circular economy, 3D printing, recycling, up to networking projects and characterization projects. The role that we perform in this project are listed here, and we usually take the lead of the exploitation and dissemination work package. So we take care of innovation management, and during the project we identify the key exploitable results, and based on them we prepare the exploitation and dissemination strategy of the project, and then the corresponding exploitation and dissemination plan. We develop this exploitation plan further, so we develop business model, and we follow that by business plan preparation. To support these activities, we perform different training for our partners, so we can train in IP protection, but also in preparation of exploitation and business plans. During the projects, we prepare also surveys for the consortium partner, but also for the stakeholders outside of the project, with the aim to determine and analyze what is going on outside of the project, and also to know the barriers and obstacles for commercialization. We give also an advice on development of products and market penetration. We prepare patent landscape reports and analysis, literature review. We are involved in IPR management and monitoring, as well as in preparation of customized patentability reports to support the IP protection measures in the project. We support the partners in finding future funding and engagement with investors. We organize also different workshops and open days. And from the dissemination side, we can prepare the website, virtual expo, logo, leaflet, brochures, and all material for the project. We can also support the setup and management of the advisory board, and we facilitate the establishment of relations with EU clusters and related projects. So one of the things that we have developed recently is the virtual expo, with the aim to support the commercialization of nanomaterials application. So this is a virtual expo platform covering different thematics. So at the moment we are covering energy, biomedicine, environment, circular economy, additive manufacturing, characterization, construction, data management, aerospace, automotive, sport, and network building. So we have prepared some expositions for the projects that we are working on, and we have added them to the corresponding thematics that I have shown. So when you enter this virtual expo site, you will be able to select different thematics. So as you see on the first floor, we have our biomedical expo, with regenerative medicine expo, with an expo for lab on chip microfluidic devices. Then on the second floor, we have some circular economy and carbon capturing. And then on the third floor, we are covering characterization and energy. So once you enter the RegMed Expo, you will land on this side where you will see more details about the nTRACK project. The aim of the preparation of this virtual expo was to support the exploitation of the project results, to increase the visibility of the project, enable the promotion of project partners, and allow engagement with the project partners during the project, but also after the end of the project. The aim is also to support the organization of workshops and open day during the project. So once you enter the RegMed Expo, you will be able to find different menus by clicking on this orange dot. So for example, you can get to the project website, you can enter the exhibition hall with all partners, you will be able to fill in a contact form, see a project video, and then when we support the organization of open days, you will be able to register and see and download the material related to the open day. So here is how the exhibition hall for the nTRACK partners looks like. So again, you will be able to enter the specific exhibition booths for each partner. So here is just an example of the exhibition booth of BioNTech, one of the partners of the consortium, who will present a bit later. So these are the features that you can access through this exposition booth. So you will be able to enter the page with their presentations and with their publications, and to download these publications in PDF. Then to fill in a contact form, enter the About Us and read more about BioNTech, see their products and services, then of course see some promotional videos, and by clicking here you can visit their project website, their company website. So here are just four examples of a few other partners of the nTRACK project, and all of them will be presenting later. Cambridge Nanomaterials Technology is also leading two private consortia, which are meant to be technology tracking activities for our private members. One is dedicated to nanocarbon enhanced materials, and the other one on advanced materials for additive manufacturing. So you can see that the members of these consortia are usually big industrial end user companies. So we have members like Airbus, Bosch, Henkel, Whirlpool, Johnson Matthey, Nokia, Rolls-Royce, Schneider Electric, etc. So this is just a few other activities of our company. So we organize also physical meetings. This is just an example of the meeting that we organized in Brussels for the Medlock project. So you can see we can organize the conference and also the exhibition for the project partners, where they will have the live booths. Unfortunately, we had to switch in the last 2 years also to virtual conferences, but still on both we are very proud to have a quite large industrial participation. Here are just a few examples of some other activities that we are performing. So we can perform training workshops for projects and also for private clients. And this is when we have, this is a picture of one of our projects where we have helped the government agency to prepare the roadmap for graphene application. So now I'm moving to the overview of current trends in innovation management in regenerative medicine, and I will concentrate on the technologies that have been developed in the nTRACK project, so on stem cell producers, stem cell tracking technologies, imaging, and nanoparticles for biomedical use. So during this 4.5 years of the project, we have used different tracking tools to determine these trends and to help our partners during the process of technology development and protection of their results. So at the beginning of the project, we have prepared a technology monitoring report to track the technologies outside of the consortia. And we have updated this report every 6 months. So we are currently working on the 9th update of this report. We have also used an exploitation survey to collect more information about this, and also the preparation of business plan. So here you can see the methodologies that we have used to determine this trend. So we have used the patent search and analysis, publications analysis, press releases analysis, key players identification, competitors analysis, related initiatives and project mapping, market analysis, and investors overview and analysis. So I will have one slide for each of these results later on. And as I said, we are covering the thematics of stem cell producers, stem cell tracking methodologies, imaging and machine learning methods, and nanoparticles for nanoimaging. So here are some conclusions of these activities that we have performed in the 4.5 years of the duration of the project. We have identified all the relevant patents for our project partners, and we have presented them the patents, and we are working also on some patentability reports on when they are needed. So here you can see some results, and we can say that patents related to the technologies that we were tracking have increased very much in the last 4 years. We have seen that the most increase in patenting activities was in the thematics of imaging methods, where the number of patents has almost doubled in the last 4 years. Here you can see a pie chart of the countries where the patents have been filed. So as we have expected, the most patents are patented in China, followed by the US and the world patents. So as we are preparing these patent reports for all our other projects, we have seen that usually China is having around 80% of all patents. So it was a curiosity that for this technologies, actually the US patenting activities and the world patenting activities, which are mostly coming from the US, are quite high. As I said, we are following also the publishing trends, and the conclusions are almost the same like for the patenting activities. So again, we see a lot of activities coming from China, and again the publishing is very intense in the areas of imaging and machine learning. To give our partners an overview what is going on the market, we have also followed the press releases, and the aim of this was to identify the state and trends of the stem cell market. The general conclusion of this activity is that this market is very much oscillating. It is very rapidly changing. And we have seen in this 4.5 years that a lot of stem cell companies have been found. Many are going to bankruptcy, they are changing names, many of them are merging, and many of them are being bought by large multinational companies. We have also seen that large investments are being made in regenerative medicine companies almost daily. So really each time we check to see how this activity is going, there are a lot of press releases on large investments. To help our partners in development of their technologies, we have used also different methodologies to determine the industrial key players and to analyze them for them. So we have identified the key players by using the patent search, literature overview, into looking into clinical trials and into the press releases. So again, we have determined and analyzed for the partners the key players for stem cell production, for imaging, and for nanoparticles. To give a deeper insight what the competitors are doing, we have again for these three thematic stem cell production, imaging companies, and nanoparticles producers, analyzed the key players and presented them to the partners. So these are just a few examples of tools that we have used to determine this. So we have looked into the growth of number of employees, the employees distribution, and the latest headhunt. We have presented the partners also the products and technologies that the competitors are working on, and also some pipelines of their products. So this is just a small part of the competitor analysis we have performed in this project. To help also in identification of what is going on outside of the consortia, we have also mapped relevant initiatives and also the related projects. We have analyzed them, and you can see here just a few projects that have been analyzed for the partners in detail. So today we will have the presentation of the two sister projects, but we have presented this information about all the projects to the partners, so we can actually map the key players and identify possible competitors, but also possible collaborators for the future. We have also performed the market analysis, again, for the sectors of stem cell production, for medical imaging, and for nanomaterials production. These reports, we have collected the information that are actually in public domain, and we have compiled them, analyzed and compared them in the report we prepared for our partners. And we have also included a live link so the partners can go and purchase this commercial reports if they're interested to see more details. And as the last activity, we have prepared a report on investors overview. We have concentrated mostly on corporate venture capitalists. We have presented them also in the report. So these are just a few examples of the venture capitalists that we have identified. We have always presented some kind of indications in which type of industry or products this investors have been investing, so the partners can find actually the right investors for them. So going to the conclusions of this 4 years desktop research, as I said, the number of patents and scientific articles is constantly increasing. The most dynamic field of research and patenting is being performed for imaging technologies. The large number of patents and scientific articles, as I said, is coming from the Asian Pacific region, followed by the US. We have seen that the stem cell market is very dynamic, rapidly changing. We have seen that many stem cell companies are being founded, merged, changing names, disappeared, or being brought by multinational companies. We have seen large investments being made almost daily in companies working with regenerative medicine. We have seen a huge demand in imaging and machine learning techniques. And we have seen also that well-established stem cell and imaging companies are constantly growing. So thank you very much for your attention, and please feel free to visit our project partners at the RegMed Expo site.
Thank you, Jelena, for that interesting presentation. I think it's quite shocking, just the lack of European patents filed, and it really reminds us why projects such as nTRACK really trying to improve our industrial excellence here in Europe. So now we can go to the first Q&A section, where we have our first question for Mark. So the first question is, after the meetings with regulatory authorities, which is your conclusion on the product definition?
Okay, it's hard to say. I mean, the previous background we were just focused was the one company said Macforce, who was commercializing and developing a ferrofluid system for direct injection into the brain for the thermal therapy. And this was classified according to the German authorities as a medical device. Our experience is that the current situation is quite unclear, since, as I said, it's a borderline product, so it can be considered according to the more formal definitions as a medical device or as a drug. So what my impression is that the regulatory authorities needs or requires some insights from the research side in order to provide tools and the sources to generate a kind of guidelines, because the nanomedicine in some instances, Moin showed a few examples, but it was more in the formulation side rather than a therapy, and it's really difficult. So we are kind of a twilight zone, if I can say that, to know where we stand. So our decision was to go into the worst scenario and to be ready for the requirement for these authorities. In the meantime, and quite recently will be published a paper regarding this kind of tour together with two more partners of the consortia, in order to provide a kind of a guidance, provide some information to the scientific community and to the regulatory authorities to define a kind of a clear framework to allow the progression of this kind of technology. So sorry, I cannot ask to be clear, but my answer is we are on it. So we are just progressing, and it's really difficult. It's really case by case. Sorry about that.
That's great. Thank you very much. And we also have a question for Jelena, which is, would the presented information be available outside the consortium? Oh, Jelena, we seem to be having some trouble with your microphone.
Okay, now?
Yes, great, we can hear you. Thank you.
Yes, so as soon as the project is finished by the end of the month, we will be able to compile this information and to offer them also outside of the consortia. Also, we can customize the information and go deeper into some of the thematics that I have shown, and we are able to offer this also commercially. Also, the virtual expo is going to be more open when the project is finished, and we are also able to accept and prepare virtual expos on all the thematics that I have shown before.
Great, thank you. I've just received good word that we've managed to reconnect Dr. Martin Leahy, so we can once again have his presentation, giving us an introduction into the StarStem project.
Okay, hopefully you're all hearing me now. If Mark nods, I know that we're okay. Yeah, I see him smiling, so hopefully that's good. Yeah, so sorry about that. We didn't actually find out what's wrong, but I think you're hearing me, so that's good. So I'm going to talk a little about the StarStem project, and much of what I will say feels like you've heard it before, because we were funded under the same scheme as Mark's project and others, and the main theme was to use nanomaterials to track stem cells in large animals and humans, and so that's what we intended to do. We have one small, or actually for us, maybe a significant change to that, in that we took the risk and decided to introduce also extracellular vesicles in our proposal. And as you know, with EU projects, that's a bit of a risk, because these are tenders. They tell you they want somebody to work on tracking stem cells, and usually if you give them something that they didn't ask for, they don't look very well on it. Nonetheless, we survived, and I think it could be an important additional thing to have in the program, right? So it adds something different to what others are doing. The other different thing you will notice is that we use optoacoustic imaging in addition to MRI. And in fact, our mainstay is optoacoustic imaging. This means that we can follow the nanoparticles using the photoacoustic effect, which is when the light is absorbed strongly by gold nanoparticles, and if the light pulse is very short, i.e. nanoseconds, then there will be a massive vibration, and then we can listen for that vibration using an ultrasound system. So after the vibration, everything else is an ultrasound system. And so our target application in this case was in osteoarthritis in the knee, and the application of MSCs, or mesenchymal stem cells, to solve that problem. And indeed, MSC-derived extracellular vesicles. So these are little parts of the MSCs that still have the information and material, we believe, that is important for the healing function in osteoarthritis. And yeah, part of the project is to figure out what that is doing, okay, after injection into the joint. So not systemic injection, but injection into the affected area. For the physicists here, I know for myself, it's always useful to have some concept of the size of things, although I notice many biologists don't seem to feel the need to put scales on anything. For me, it's very important because I like to know what size of things I'm dealing with. So when we talk about EVs or exosomes, exosomes are part of the EVs, the ones less than 100 nanometers. And they're about the same size as a virus. You know, we all know much more about viruses now than the last time I spoke to the nTRACK meeting about about this project. And of course, then we have microvesicles, and cells are, of course, much bigger than the EVs. And there are other advantages in using EVs. There are, we believe, less of the complications that you have with stem cells. So potentially stem cells could turn into tumor cells, or they could turn into something else that is undesirable, or do things that are undesirable. And they're just so much more complicated that the regulatory pathway becomes very, very complicated for cells. Nonetheless, people, including Mark, has talked about some success in moving forward and engaging with the EMA and others, as indeed we have tried to do too. So the project has involved our own base here in Galway, on the west of Ireland. I suppose we're the most westerly university in Europe. And we have a management company in Dublin who's helping us to manage the project. University of Cambridge is doing the MRI work and the sheep, so the large animal. And again, this is the same animal, if not the same model as Mark used. I think he was talking about muscle. We're talking about the actual knee and the cartilage in that knee. And it turns out that the sheep knee is very closely related to the human knee and is a good model for that disease. We work with the University of Geneva, and they are developing the cells and the materials that the cells reside in, the media, which turns out to be quite important. Technical University of Munich and Ithera, which is a spin-off from there, and also Ithera Scientific, are all involved in the optoacoustic system development and in scaling up our nanoparticles that we invented at NUI Galway. We have included ICFO at Barcelona, which is a wonderful team working on nanoparticles, and the idea to be able to produce liters of this stuff. So if people are looking for quantities of nanoparticles, maybe I can, of these particular nanoparticles, gold nanostars, which we believe are the brightest and most heavily absorbing nanoparticles. And the reason we want them to be the brightest and the most heavily absorbing, of course, is the more signal, the more energy you absorb, the greater the acoustic signal that you can then pick up with your ultrasound system. So in optimizing our scheme, we are trying to get the maximum amount of energy deposited in the nanoparticle from a pulse of light. So we're using a specific wavelength of light that can go as deep as any wavelength of light can go in the tissue. It's using the 1064 wavelength, which is actually also quite easy to get a laser that provides a good pulse, repeatable pulse at that wavelength. And there's low water absorption, low scattering, and so deep penetration. Helps us to see deeper and to affect therapy actually deeper as well if we go down that route. So why osteoarthritis? Well, it affects 10% of the global population, will be more than 20% by 2050, partly because we're all getting older. And of course, that will be even more of a problem in Europe as we age faster, I suppose, than much of the rest of the world. Not that we're aging faster, I suppose, we're just not replacing or building up our young population as fast. So optoacoustic imaging is non-invasive and it has a limited depth. This is one of the big challenges with using that. It's spectroscopic, so we can pick out very specific materials within the body and identify where they are. So we can see, for example, the difference between oxygenated and deoxygenated hemoglobin. We can, and in this case, we can see specifically the gold nanostar, which we have tuned to absorb exactly at the 1060 nanometer wavelength that we wish to hit it with in order to vibrate the nanoparticles. So Why gold nanoparticles? Well, because they absorb maybe 10 times more than any typical dye that you could use, and the nanostars are a bit better than nanorods. Okay, so everything is optimized to improve the sensitivity of these materials. So the competition was fluorophores and dyes and radio labels, other kinds of nanoparticles. So we are trying to improve the energy deposition and, of course, the uptake of the nanoparticles. They're not going to be seen in the cells, and the cells are not going to be seen unless the cells have taken up sufficient numbers of the nanoparticles. So you need not only to ensure that you have the best possible size of nanoparticle and shape for energy deposition, but you need to make sure that the charges and other important parameters are correct so that they will be taken up by the cells and yet not be toxic. And charge has, as many of you will probably know, a big impact on toxicity. So we're trying to maximize the sensitivity at depth. So we're building this nanostar that we're going to put inside the cells or attach onto extracellular vesicles. The vesicles are about the same size as the nanostar, so we can't put them inside the vesicles, but we can attach them there too. We want to inject this material inside the cells into the knee joint to achieve this healing process, and we want to know where they go and what they do inside the body so that we're ready for validation by a clinical trial. Not that we're going to do a clinical trial in this project. 4 years is way too short to get to that point, but we want to be ready for that with the documentation. Okay, and of course the end game here is that we want these wonderful cell therapies to cure these intractable diseases, make lame people walk, make blind people see. That's the promise of the stem cell therapy, but of course the regulatory path has to be secured if we are going to achieve that. Okay, so here's an overview of the project. Looks very remarkably similar to Mark's poster, I have to say. So we have our nanostar, we have the nanostar tagged MSCs, and we get them into cells, we study that, we look at them in tissue, then we look at them in mice. And indeed, and of course, with the mice, we can see in the whole body where they go and what they do. With optoacoustics, we're well able to see the full size of a mouse. So we can see if they go in the liver, the spleen, wherever they go. But a better model of arthritis, or at least an extra model of arthritis, which will be necessary in order, before you can go to humans, you want to look at least 2 mammals, will be the sheep knee. And in fact, the knee is not here or down here, it's way up there, because that's the model, that's actually where the knee is. This is more like the, well, this thing here is the wrist. The knee. Farmer would probably tell you that's the knee of a sheep, but in terms of the model, the actual knee joint is the equivalent of the human knee joint is up there, right? You can imagine them being upright. So the idea then is that we can see the optoacoustically the concentration of these nanostars, and thereby the concentration of the cells or the EVs inside the knee, and by adding spions, these iron oxide particles, we also wanted to be able to see them in MRI. That actually part turned out to be much more challenging than we expected. We thought that was the more straightforward part of the project, I suppose, but challenging nonetheless. Here's a diagram I like to show to show the challenge we have in imaging of anything inside the human body or animal tissue. And it is that there's a relationship, this empirical relationship between the sampling depth, how deep you can see, and the resolution that you get. And it actually transcends all these very, very different modalities. So, you know, in the hospitals, we see ultrasound and MRI and X-ray CT and so on, and they are able to see very deep the whole body, but the resolution you get is about 1 millimeter, right? And you can get slightly better with ultrasound, but not much. If you go to higher frequency ultrasound, for example, but your depth will become very rapidly limited, which is why they typically don't use high frequency ultrasound in the clinic. They use it in discovery in small animals. Photoacoustic imaging or optoacoustic imaging is somewhere in here. With optical coherence tomography, which we also use in this project, we can see more or less subcellular resolution, and we can see about 1 millimeter into the tissue. So that's useful because you're down at least at a depth where you have blood vessels and normal biology going on, and it's real 3D biology. And of course, you all know about confocal microscopy, and even super resolution microscopy, Stefan Hell tells me, falls on this line where you can see as deep as you want, but the resolution will never be better than 200 times smaller than the depth that you want to look. So if you look, you know, a centimeter below the surface, you will get 50 micron resolution. If you look 1 millimeter below the surface, you get 5 micron resolution. If you use super resolution microscopy, you know, you're typically looking tens of microns below the surface. And so you get 200 times smaller than that as your resolution. And up here, you're looking tens of centimeters below the surface. So you get maybe 500 microns or 0.5 millimeter. Or if you go deeper, you get 1 millimeter resolution, right? And this is simply, I suppose, crudely, because we have to integrate more signal as we go deeper into the tissue. Okay, so if we want to do clinical MRI, we see deeper, we get this lower resolution. If we use photoacoustics, we can get 50 micron resolution at centimeter depth. And then after that, we start to use OCT, and going deeper than that, of course, we're not seeing inside the body. So we're not going to talk too much about that. Our nanostar looks like this. It has very sharp tips, which gives the enhancement of the absorption we require. So there's a big energy deposition at 1064 nanometers. The shape of this gets us all the way up to 1064, which is part of the challenge. Really easy to do nanoparticles that absorb at 500, 600, 700 nanometers, but these longer wavelengths are harder. Obviously, you will all, if you've worked in this game, realize that not everything that you actually make looks exactly like this 5-legged star here. You even have things that look more like gold nanospheres within that, so the purity is not 100%. The idea is that we would be able to do kind of liter level sizes of the, and we've achieved this large scale production of nanostars. So how do we make them? Well, we have a very small gold nanoparticle onto which we put some silver particles, and then gold grows on top of that, yielding these spikes. So the crystalline growth yields these spikes, and we optimize the method so that we get these longer wavelengths. You all know that one way of getting longer wavelengths is to use bigger nanoparticles, but there's a limit to how far you can go with that. It's been proven over thousands of years. You can stretch the nanoparticles to rods, and then you can get longer wavelengths. These don't seem to be successful in vivo. I think that's partly because if you have two rods that line up next to each other, then instead of having an aspect ratio of 8, they now have an aspect ratio of 4, and so on, and then maybe less than that again. So your wavelength is very dependent on how these things agglomerate inside the tissue. You want to have a large spike at the wavelength of choice. So around about 1,000 nanometers here, we're trying to maximize this spike and minimize this one over here. Actually, this one over here is telling us really the concentration. This one here is really telling us how well tuned it is to the specific wavelength that we're interested in. So we have the two wavelengths actually, because we have this core, which is essentially still a sphere, and we have these spikes, which is a bit like having three nanorods, right? You can imagine 3 nanorods kind of stuck together somehow. I don't know how I do that with my fingers, but you get the idea. One very important part, which is done by our collaborators in Geneva, is producing the materials to put the nanos, the cells, the stem cells into, so that they have a good life, good properties, good survival, good action, and so on. And in fact, in this case, we've been able to use human and sheep MSCs only. We thought we might have to use mouse MSCs as well, which would add another complication to things. But because in the end we went with a nude mouse, they are capable of taking sheep or human MSCs anyway, so we didn't have to go with the mouse MSCs in that case. Yeah, and we incubated the cells then with either the spion conjugated stars, or just spions, or just nanostars, gold nanostars. And here is a typical image of my wrist, and so you can see this section of my wrist here, and if we look at the cross section, we can see where the blood vessels are. We can see where the melanin is, and various things. And of course, this is a top-down look at it. These are blood vessels, which you can kind of see really with your eye, but you get better quantitative information from it using this optoacoustic system. So here is our optoacoustic system. We've used a couple of different types. One is a handheld one, and another one is like a micro MRI sort of thing, scheme. So in that case, we have pulses of light hitting the blood vessels or the melanin, as you saw in the picture earlier, and they're giving a signal back, and the whole thing, image formation, is done exactly in the same way as is ultrasound. And indeed, it will look like an ultrasound system when it's wheeled around in the hospital. So here is an MRI that we've taken of one of our sheep knees. And when we zoom in, we can see the bone and the fibrous clot and the bone shelf and the cartilage, the parts we're interested inside the sheep knee. With optoacoustic imaging, we can see a bit more detail, and as well as the anatomical outlines, we can actually see the concentration. I don't know if you can see that, but that says SO2 there, or concentration, oxygen saturation, the saturation of the hemoglobin, what percentage of the hemoglobin is carrying oxygen. And for reference here, you see this is actually an MRI image of the finger joint, first joint of the middle finger. You can see that the optoacoustic images are a bit more detailed and clearer, as well as having this spectroscopic information about total blood volume, SO2, hemoglobin, oxyhemoglobin, and so on, and water, in fact, over here. So because it's spectroscopic, you can use any wavelength you want to interrogate the tissue. We're not looking at the nanostars now, we're looking for other chromophores, you can get more information. Okay, and this is the second joint of the middle finger. So similar information, just a bigger joint, bigger part of the finger. And that's a close-up view of optoacoustic versus MRI, which gives you a sense of the kind of detail that you get with the optoacoustic image. And of course, this finger joint is not much smaller than a mouse that we use in our studies. Okay, so the nanostars, when you dry them out and you try and image them in electron microscopy, you see that they start to agglomerate. And of course, you're not really sure whether they've agglomerated because you've done this process to image them, or they're agglomerated anyway. But you can get that information really from spectroscopic techniques, which because the spectrum that is absorbed is really closely related to the size of the particles. So if they've stuck together, they will absorb at different wavelengths. So we have had to do our cell labeling and titration. So only 20% of the MSCs contain internalized membrane-bound nanoparticles. We had to improve all of these things. This is our mouse model that we used, and because we used that mouse model, we did not have to use mouse MSCs. I've got to say a couple of words quickly about OCT. It's the most rapid growing imaging methodology in the history of medicine. It sees subcellular resolution about 1 millimeter deep within the tissue, and has been used for many things, including here we show that we can get nanoscale information. So even though the resolution is on the microscale, we get nanoscale information about the structures within it using a technique called nanosensitive optical coherence tomography, which is something we developed ourselves. And here is shown just helping us to see the difference between a diseased tissue and a non-diseased tissue. And this is in 100 kids in a hospital in Illinois. And they're coming in off the street. They've got their kids' ears, which are not specially prepared or anything like that. But we can see, you can see here a few nanometers shift in the structural size between diseased tissue and non-diseased tissue. It's a nice application of that. I don't really, I think, have time to go into the details of that, but we can show these maps showing where the large structures are and where the smaller structures are within the tissues. So to wrap up then, I just repeat what I said earlier. Our target was to use nanostars to inject them into the knee to cure osteoarthritis. But in order to do that, we need to show where they go and what they do, as well as in the knee, in the whole body. So this is where the biodistribution comes in. And so get the data we need for clinical trial and learn more about the disease and the imaging modality and the MSCs and EVs at the same time. These are the people who worked on the project. So thanks to all of them, and I'm happy to answer any questions you may have.
Thank you, Dr. Leahy. I think we can all safely say that was well worth the wait, and I'm glad that we could have you connected today. Now we're moving on to our first break for today, where we'll be returning at 11:30 Brussels time. And I'd just like to encourage all attendees today that the RegMed Expo can be seen at the top of your screen, should be in the navigation bar, and I'd well recommend you check that out. See you soon. Sorry about that. So welcome back, everyone. So we can now continue with our second thematic segment, which is nanomedicine. All attendees today should be aware that any unanswered questions will reach our speakers and we can reply via the mail. With that said, I'd like to now invite our first speaker of this second segment, Dr. Emre Tureli, who is the CSO of My Biotech and an expert in drug delivery systems and nanoparticle development.
Thank you for the introduction. Good morning, everyone. Today, I will talk about the GMP production of nanoparticles, especially about the gold nanoparticles, which we produced during the nTRACK project. So what we wanted to have was gold nanoparticles that are pegylated and that are coated with glucosamine to facilitate the uptake of these particles by the cells. So these particles can be used to track the fate of the cell in the human body. The first thing what we have done was just to establish a method based on the literature, based on our partners' experience, how we can prepare these gold nanoparticles, mostly with batch synthesis methods, where we just tested if we can get the particles, where we understand the method, how we can get the particles, how we can do the PEG coating, how we can do the glucose amine coating, and what are the critical steps, what are the steps that are required extra attention in terms of GMP compatibility. Because when I think about a technology transfer concept, especially for the GMP production of the nanoparticles, I need to make sure that every method that I'm using is within the GMP regulations, and we need a high efficient method. We cannot lose a lot of particles on the way where we are synthesizing the particles. I need to ensure that I can validate the method, and more importantly, I can control the method and method parameters, so I can be sure that the quality of the product at the end has all the required specifications or is within the required specifications. So we have determined a couple of steps during the production of the particles where we would need to change in order to get the production method more GMP compliant, in order to be able to control the parameters of the production efficiently, and in order to be able to show that we have always the same particles at the end of the production. One of the things that are mostly used in production of nanoparticles, in case of gold nanoparticles, but also in case of other nanoparticles, is the mixing process, which is a very important process and which is a very batch dependent process. That means that if you are doing a mixing in a small container, in a small batch on a lab size, the mixing is always efficient, so you get the desired particle size or desired particle characteristics, what you want. But once you start increasing the batch size, at some point you will see that once you further increase the volume, the particle size changes, the particle properties changes. So in this case, it is advisable to use a continuous mixing method, so that you are sure, no matter which batch size you are producing, that the mixing is always efficient, which affects the particle size, which affects the particle properties. This was one of the changes that we have done in the beginning. Then there was also 2 steps involving washings of the particles with the centrifugation, which obviously works. But especially in case of centrifugation, since you first sediment the particles, they do agglomerate on the bottom of the vial or falcon or your container what you are using. We disperse them in a bigger scale is also a challenge, so that we have changed this as well. We use then cross-flow filtration, this is a technique quite different than a dead-end filtration, where you can wash the particles, where you can wash the free salts, free APIs, you can wash out the excipients, free excipients, you can wash out the organic solvents if it is used in the process, keeping the particle size stable and particle properties stable. And the last step which we changed in the production protocol was how we did the coating. Again, it involves a mixing process, so that we changed into a continuous mixing under controlled conditions. So when we set up the production process, you can see here, we first start with the gold core manufacturing, that is from the gold salt. And this is our intermediate one. As I said, the first one was done with the continuous mixing process. Then we purified them with CFF. This is our intermediate 2, which is followed by a PEG coating, again purification, buffer exchange, glucosamine coating, again purification with CFF, and at the end concentration with CFF. If we didn't establish the CFF, we would be centrifugating the particles all around in every step, which would at the end most probably lead to loss of particle properties or the decrease in the concentration of the particles during the process. As I said, it definitely works at lab scale or small scale, but if you want to increase the scale a bit, you would need to come up with different methods to ensure that you can either do continuous manufacturing, or you can do purification with systems like CFF, where you keep the particles stable, keep the particle concentration stable, but only change the external medium. One of the challenges in this project was the high gold concentration that was required at the end of the production process, so that we decided to set up a design of experiments to determine the critical process parameters, which might lead to decreased gold concentration at the end of the process, to ensure that we reach the high gold concentration at the end of the process. And we, of course, set some specifications for critical process parameters. Before I go into that, just a couple of slides. What are these control strategies, how we can use quality by design in the production of nanoparticles according to GMP regulations. So the idea is, once you start with designing your process or testing your process, you have two different things that can affect your process. The first one is of course the ingredients, the excipients, the API, the solvents that you use for your process. And the second thing is how you do the formulation, how you run your process, how you design your process. And most importantly, you need to understand every step of your process, which means that you need to know what are the effects of the parameters that you use during your process on the end quality of your particles. How you can understand your process? You can understand your process with a risk assessment to determine, okay, these are my process steps. I need to go through 5, 6, 7 different steps, and at which step or at which part of which step I have the highest risk to decrease, in our case, decrease the gold concentration or lose the gold concentration. And based on that, you can put together your critical quality attributes, critical process parameters, critical material attributes. Critical quality attributes is more regarding to your end product. So this is the quality of or attributes of end product you want to reach. Critical process parameters are the process steps that are critical, in our case, to be able to reach the high gold concentration. And critical material attributes are the attributes of your starting materials, in case they might have an effect on the product or production according to your risk assessment. Then you would need to establish, of course, some acceptance ranges for critical material attributes and critical process parameters. What I mean for acceptance ranges is, for example, you found out that the process works better at 2 bars, it is obvious that you won't be able to keep the process at 2.00 bars at all the times. So you would need to come up with a limit from 1.95 to 2.05 bars where you know your process is still stable. And during your process, you would need to think about some control strategies in process controls to find out or to monitor your process if everything is working accordingly. How we do it is basically, there's only one way to do it, that is the quality by design. And this quality by design is really needed for the GMP process development and the control. This helps you to understand your process, which means in the first place, the most important part is a proactive approach to pharmaceutical development. So it doesn't wait until something happens and you try to find out what was not okay or where is the problem. You think what could go wrong, and you try to determine the steps where something could go wrong and try to control them on one hand. But on the other hand, after these steps, you try to analyze your product in that intermediate stage with in-process controls to be sure that everything is still okay with your product and with your process. That is something that you don't do at the end of the process development. That is something that you do in the beginning of process development, and it begins with predefined objectives. So you need to clearly state or write what is that you want to reach. You need to have a list of process control parameters which you can control really during your process. And of course, it needs to be based on a risk management, on a risk analysis, where you can scientifically show that what are the risks and how do I cope with them. So by starting by quality by design, you start first with the quality target profile. This is critical quality attributes of your product, which might include, for example, in case of nanoparticles, the particle size, surface characteristics, concentration, physical state, assay, impurities, and so on. And you would also need to determine the critical material attributes and critical process parameters based on evaluation of your process. And then you do a risk assessment based on these critical quality attributes. And then you do a design of experiments, and at the end you make a continuous improvement based on the results of the design of experiments. I will just now show you an example how we have done for the regarding the risk analysis for the gold concentration in the sample. As I said, the idea was to keep the gold concentration in the sample at a high level. That was required so that we can use these gold nanoparticles for the tracking of the cells. So we listed here on the left side the different processes. This is gold core manufacturing, purification with CFF PEG coating, purification with CFF buffer exchange, glucosamine coating, purification with CFF, and concentration with CFF. And then did a risk assessment based on the process step, if it is a low, medium, or high risk that we could lose, or we could have a gold nanoparticles, the concentration of the gold nanoparticles decreasing. And we found out that at the end, the highest risk is at the end of the process when we are concentrating the gold nanoparticles, that we might see a decrease in the gold concentration. So based on this risk analysis, the CFF process is identified as the relevant risk for gold concentration. We did an experimental planning based on design of experiments. And then, of course, then determine the gold concentration to set the specifications. We have used a box banking design where we have listed different flow rates that we can use for the cross-flow filtration, different pressures we use for the cross-flow filtration, and different times we use for the cross-flow filtration, and always recorded the gold concentration at the end of these experiments. Of course, before you start with something like that, you would need to have an idea about, okay, which are the flow rates that I can use during the cross-flow filtration, or which pressures I can use during the cross-flow filtration, so that at the end the data makes sense. You cannot go and start with 50 milliliters per minute when you know, okay, I won't be able to do any cross-flow filtration with 50 milliliters per minute. So you need to select your limits which you want to try, or your parameters which you want to try based on either your previous experience or the initial testings of the methods, to have at the end a design space which makes sense, and most importantly, a design space which you can use. The gold concentration required was 30 milligrams per milliliter, and we have given as percent. So if it is 100%, it is the gold concentration is 30 milligrams per milliliter. We have also did a statistical evaluation of the model, which we found out that the model is significant. The lack of fit is not significant, which is required, so that it's not a coincidence that we see some effects of different parameters. We have also investigated the design space. This is the 3D investigation of the design space. In order to be able to make it more comprehensive, you can do it based on only two factors, or on the one factor, sorry. For example, here is the gold concentration in percent. So this is a mistake. It shouldn't be milligrams per milliliter, but percent. And this is the flow rate. And as you can see, if you go down with the flow rate, you have a decrease in gold concentration, but if you go too high with the flow rate, you also have a decrease in the gold concentration, so that you have here a quite stable area which you can use for your experiments. The same we did for the pressure. If you increase the pressure, the particles tend to agglomerate more, where you lose the gold concentration, so that you want to keep the pressure as low as possible. So that we ended up determining optimal flow rate 140 is 160 milliliters per minute, with a pressure of 1 to 1.4 bars. So how does this risk analysis looks now? So we had different CFF processes that we apply for the manufacturing of gold nanoparticles, which were either medium in risk analysis or high in risk analysis. And we took some measures based on the design of experiments that we have done, and show that if we stay within these limits, the resulting risk is low, that we lose any gold concentration during the process. Okay, that was actually just a part. Of course, that was not the only risk analysis that we have done during the setup of the process. But I just wanted to give you an example how you do a risk analysis, how you do a design of experiments in establishing most complicated GMP production methods for the gold nanoparticles, which require more process knowledge and understanding than the basically normal or the manufacturing methods that we have used to just press tablets, capsules, or fill some vials. Just to in this case, for example, give you an example how we determine the factors to keep the gold concentration as high as possible, and what measures we have taken, and how we could then decrease the associated risk. Thank you very much for your attention. And that was the end of my presentation.
Thank you, Dr. Torelli. As a chemical engineer, I found that presentation really enjoyable, and don't worry, I'll prioritise audience questions over my own. Our next presentation today is Dr. Simon Schwartz from Weiderbronn Research Institute, where he leads the drug delivery and targeting group at CI BBM Nanomedicine.
Well, thank you very much. I'm going to talk to you about cancer stem cells, about how to target this specific type of cells that you will find in the tumors, and also a little bit on exosomes and how those extracellular vesicles actually act as have a role in what is known as intercellular communication crosstalk with these cancer stem cells and the rest of the tumor cells. So the first thing I would like to tell you is I think that you should know is that cancer stem cells are a subpopulation of cells within a tumor, or a given tumor, that have a very specific kind of features that distinguish these cells from the rest of the tumor cells. So they are rare. There are just very few of them. Normally, actually, if you check tumor cell lines, they have to tend to be always a very specific proportion, a very specific fraction of those cells, and that depends a little bit of the kind of the tumor cell line that you're working with. But at least this is something that you see quite often in tumor cell lines when you work in vitro. Not that clear that this is also happening in vivo for sure. They have this cell renewal ability and this capacity to be very much resistant to most treatments. So they are the ones responsible of actually making sure that the tumor will survive to chemotherapy. And they have this ability also of surviving to non-addition. And this is very relevant because this is the reason why actually these cells will be able to detach from the tumors, will be able to go into the bloodstream, will be able to survive and then to reach these cell organs and produce the metastases. So these are the cells that we want to target when we treat tumors with drug delivery systems. No? So it is obvious that from the practical point of view, when you work with cancer stem cells, it's quite difficult to distinguish them from the rest of tumor cells because apparently you will not find very specific distinctions. So a few years ago we designed a system. This is a vector that we designed and we tested in tumor cell lines. So in a way that you will introduce the possibility of expressing a fluorochrome. This is TD tomato, what you have in here. So under the promoter of LDH1A1. So this is a gene, LDH1, which is highly active in cancer stem cells. So the idea was, if we take a promoter that is active in cancer stem cells and we attach a fluorochrome, then these cells will become red. And this is a very simple way to put it, but and we will distinguish cancer stem cells from the rest of tumor cells. And this is what you see here in this figure. So in these pictures, so the red cells are cancer stem cells, the rest are tumor cells. And these allow us to work and distinguish one from the other one, and then to see if our delivery systems actually target the cells that we want to. And also if by doing so, we can change and improve, of course, the response to chemotherapy. Actually, the cells are the one that if you separate them and you take them and you grow them in non-attachment conditions, they will be able to produce these tumor spheres, as you see here, this red one, and also to reproduce the tumor. So if you take cancer stem cells and you let them grow, they will always reproduce the tumor. So it's kind of, they will replicate that perfectly well, no? And actually, they are the one responsible, as I was saying, not just of tumor growing, but also of metastases. So these are cells which are extremely relevant, and fortunately, they have a very specific expression of genes which you will find in these stem cell type of cells, like some of them like CD44, LDH1 that I was mentioning, also CXCR1, for instance. So there are a bunch of them that can be actually used to target those cells. So let's say that classical chemotherapy is trying to kill tumor cells, sometimes with more or less success. But it's quite often, it's quite common that at the end, even if it seems that the tumor actually responds very well to the chemotherapy, some of these cells will remain. So some of these red cells, some of these cancer stem cells will remain, and they will eventually give rise to a new tumor. You will have recurrence, you will have a tumor regrow, or eventually will have metastases if drugs have not reached them at the right site. So in theory, anything that will target those cells and will eradicate those cells from the tumor will be sufficient to bring the tumor to remission. As I was saying, fortunately, some of these cells have very specific cell surface receptors like CD44B6. This is a specific one that you will find in colorectal cancer stem cells, for instance. And that allows us to define and to design drug delivery systems with specific drugs against cancer stem cells, and also targeted actively towards against these receptors, normally using either aptamers or nanobodies or antibody fragments, etc. So you name it. But unfortunately, this is not a perfect solution, and that's because even if you take out cancer stem cells from most tumors, and this is something that you will see very often in vitro when working with cancer stem cells and tumor cell lines, there's this ability in most of them. This doesn't happen always, but in most tumors, they have this ability. So tumor cells, which are not cancer stem cells, they change, and they start a very specific phenotype reversal dynamics that bring these tumor cells to cancer stem cell status. So in other way to put it, so these non-cancer stem cells, they eventually, if cancer stem cells are missing, they will detect that, and they will eventually become cancer stem cells. So the tumor will regrow. So this means that we have to not just target cancer stem cells, but also this reversal phenotype dynamic. And this seems to be very much dependent on what's called the epithelial mesenchymal transition pathway. So there's a bunch of genes which includes like snail's log, twists, AKT2, for instance. Those are genes that when cancer stem cells are missing, they are activated. They activate this epithelial mesenchymal transition pathway, and because of this, these tumor cells will differentiate again to a more cancer stem cell phenotype. So, and those cells will be the ones responsible of again reestablishing the tumor. No? So therefore, this is a very important pathway that we have to keep in mind. So, and one of the interesting things of this is that this suggests that somehow this equilibrium of cancer and non-cancer stem cells is translating to you is that there are some mechanisms that involve for sure intercell communication that somehow are responsible of keeping this equilibrium. And it is supposed, and this is what has been recently established and published, that exosomes and extracellular vesicles, which include exosomes, but we will not get into those details. But let's say that extracellular vesicles and exosomes are capable of intervening in this intercell crosstalk communication. So, as you know, exosomes come from early endosomes and multivesicular endosomes. So they are small vesicles that are formed, including part of the cytoplasm of the cells, and therefore they contain a lot of things. So they contain microRNAs, they contain proteins, enzymes, etc., and those are secreted to the exterior part of to the extracellular space of the cell. So those exosomes, because they have these cargo loads, these payloads, I mean, they are capable of transmitting information to other cells when they are uptaken by other cells, and they are. So, so this means that if we go to the models that we design, like this is an example in which we have cancer cells, cancer stem cells in red and non-cancer stem cells in green, these crosstalk and these let's say, transformation and translation from differentiated states to non-differentiated states or dedifferentiated state. This is regulated in part at least by the content that these exosomes have. So in other ways, we could actually isolate cancer stem cells and extract cancer stem cells exosomes, and we can do the same with non-cancer stem cells. And they have different effects, indeed, because they have different contents. Actually, if you check these cells or the exosomes, you will see that those coming from cancer stem cells, they have a lot of course, TD tomato, which is the floricum that we're expressing, but also aldehyde genes, NANOC, OC4, these kind of genes that you would expect to find in cancer stem cells. And they actually are capable of internalizing in other cells, as you see here. Those are the exosomes, and those here in red, for instance, are the ones going into other cells. So this is the mechanism they will use to transmit this information. And this has consequences. It has consequences because it means that these cells will, through the use of these exosomes and this information, they will use them as in fact, as a drug delivery systems to make sure that something happened in other cells. And these other cells might be at the local level, like immune cells, like fibroblasts. You know that one of the things that the tumor does is that they activate the fibroblasts at the local level at the site of tumor implantation, but also on the distance when they are conditioning niches in distal organs, for instance. So what you have is that these exosomes are conditioning these cells, are transforming these cells, so they activate the fibroblasts, they also activate endothelial cells for neoangiogenesis, they also activate immune cells, etc. So they block often the immunological response, etc. So they have a very strong consequences in that sense. And this is an example of this. So if you take fibroblasts and you activate those fibroblasts, one of the things that you will see, and this is a 3D organoid made with fibroblasts. It was a collaboration that we did with IBEC, with the Catalan Institute of Bioengineering here in Catalonia. You will see that these fibroblasts have this tendency to produce these threads, which are essential for local invasion. I mean, they actually act as if they were railways in a way that the other cells, and this means mainly tumor and cancer stem cells, will follow. If you add to these cultures exosomes from cancer stem cells, you will see that the activation is much stronger. So the number of threads that you will produce from activated fibroblasts, they are much more abundant. So there's an hyperactivation of this activity, and this happens in fibroblasts that comes from breast or from lungs, for instance. So it means that they really have an impact on how fibroblasts perceive the presence of these cells, these exosomes, and therefore they will help on the invasion. And this is what you see at the local invasion. So if what you see here is a combination of activated fibroblasts with these tumor models expressing the TD tomato. What you see is that when you add exosomes coming from cancer stem cells and the fibroblast gets strongly activated, then they will help the rest of the cells, and this is what you see here in greens and reds, to spread. Okay, so the red cells, which you can actually see often at the very tip of these threads, are cancer stem cells. So this is a continuous behavior that somehow cancer stem cells are helping to regulate. I'm not saying that this is the only mechanism, but at least this has an impact. So therefore, what happens if you go to in vivo? If you go to in vivo, something similar happens. So if you take these exosomes from non-cancer and cancer stem cells and you inject them IV in the brain cell of a mice model and see what happens, the first thing that it's going to happen is that because you use this intravenous injection, they will immediately go to the lungs, and that's why we check them into the lungs what happened, because they will condition the lungs to a future uptake of cancer stem cells or tumor cells. No? So by conditioning these animals with cancer stem cells and with exosomes from non-cancer stem cells, you can see differences. So if you do it with cancer stem cells, you will see that these animals have much more tumors, much bigger metastases. So the lung is kind of becoming more prone to admit these cells and to actually help the tumor grow. So you are conditioning this niche. And this is something that it might be happening at the distance from the primary tumor. You have to keep in mind that when we talk about treatments, primary tumor often is not such a big deal in the sense that unless they actually affect very vital structures that might be in the vicinity, I mean, they can be easily removed by a surgical procedure. So that's not what we have to be focused on. We have to focus on what happens when these tumors spread, especially when these tumors perform metastases in the distance. This is one of the examples of what you will see when you pre-treat these animals with exosomes from cancer stem cells. It will be a very huge activation, as you see here in the very left, of fibroblasts in these lungs, and these will help the tumors to grow. And also, if you check on what happened, this is an in vitro model, what happened on neovessels and at the level of endothelial cells, you will see that also these exosomes are increasing very much the capacity of these endothelial cells to perform neoangiogenesis. And all of this will certainly help tumor to grow. And one of the signals that you will see also is an increase in the production by the fibroblast that gets activated of interleukin 6 and interleukin 8. And here happens something interesting, and is that these increases in interleukin profiles, they are much, much bigger if what you use are exosomes from non-tumor cells. And this makes sense. It makes sense because it means that when you are treating these fibroblasts with exosomes coming from non-cancer stem cells, the message, or at least this is how we interpret this data. So the message they receive is that cancer stem cells are missing somehow. So this increase in interleukin 8 is extremely big. And we know that if you treat tumor cells with interleukin 8 or with this extracellular medium from these cultures, they will induce the production and an increasing production of cancer stem cells. So this is again a mechanism to restore the levels of cancer stem cells in these tumors. Okay, so one of the good things about this and the use of exosomes is that, therefore, if they are being used by cells as, in fact, natural drug delivery systems, we can actually perhaps do the same thing. This is an example of how we do these things. No? So we actually transform cells, and it depends on the cell source, but it is something you can do quite easily. So you can transform cells to make sure that they express, let's say, a protein, for instance, a recombinant protein, in a way that they will overexpress this protein and they will load the exosomes in the multivesicular endosomes of these proteins, and then they will expel these, they will secrete these extracellular vesicles to the medium, so you can actually fish them or purify them, and you will have nice drug delivery systems with a load of loaded with, in this case, with this recombinant protein. So you can actually use them as therapy. And this is an example of precisely this. So we did, we transformed cells. This is, if I recall, this was made with HEC293 and with also with CHO cells. So we actually transformed them to make them express a protein, which is GLA. GLA codes for alpha glycosidase. This is an enzyme which is defective in a rare disease that's called Fabry disease. So it's a lysosomal protein in a way that in this disease, GLA is defective in the lysosomal compartment. So it's not capable of metabolizing what's called GB3, which is global triacyl ceramide, and it accumulates. So there's a substrate that gets accumulated because the lysosomal compartment is not working properly. So what we did was to express this GLA in extracellular vesicles to purify them and to treat knockout models of Fabry disease. And what you see here is that in most organs like liver, spleen, kidney, and also in plasma, there was very strong reductions on the accumulation of this substrate, this GB3. Remarkably, we saw these reductions in the brain, and this is very important because these reductions in the brain means that somehow this treatment is also capable of reducing the amount of substrate inside the parenchyma. And one possibility of this happening, and we think that's the correct option, is that these extracellular vesicles with the GLA are capable of crossing the brain-blood barrier, and this is very good. Actually, if you compare the effects of this with the current marketed treatment, which is algacidase alpha, this in black here, you will see a very strong difference. So in brain, alpha-galactosidase, algacidase alpha is not capable of reducing the substrate, whereas these extracellular vesicles with the GLA are. So this has clearly a very strong impact, and this opens the door of using this kind of systems as drug delivery. The same thing happens if you change the protein and you do the same with, in this case, this was an example with angiogenin, and you also see that you can actually increase the percentage of neovascularization when you deliver extracellular vesicles with angiogenin in some in vitro models. I think I'm going to stop here. Well, let me add just very fast a couple of questions. One of them that I want also to show you was whether active targeting might have also a role in this in improving the targeting in this case, as we were talking of cancer stem cells. And these are some examples of how using some peptide moieties that are actually director moieties like RGD in this case on liposomes, they're capable of increasing the internalization of liposomes like 3 times more if in endothelial cells we use RGD than if you don't. And sometimes these director moieties can even modify a little bit the biodistribution of the nanoparticle. And this is another example of using polyurethane nanoparticles with RGD, in which you see that biodistribution makes them a little bit different. So you have less accumulation in liver and spleen, a little more in lung. So at the end, this active targeting it works in the sense that it might increase the internalization, but at the same time, it might also change a little bit the biodistribution. So you have to take care of this, especially in vivo, when you go in vivo. These are some examples of how adding some other targeting systems like cetuximab, in this case, the internalization of in this case was polymerase micelles with gelatin was much higher in cells, and this happens in breast, or in this case, when using PLGA-PEG nanoparticles loaded with paclitaxel and targeted with cetuximab, we saw an increase in the efficacy in cancer stem cells precisely. So it means that that's possible. Right? Let me talk to you just to finish about this other example. This is targeting CD44, particularly a variant isoform of this receptor, which is the B6, by using a specific antibody fragment against the CD44 B6. This is a work that we did with Bruno Sarmento in Portugal, and we designed these polymeric micelles with niclosamide because it's a very good drug that has anti-cancer stem cell properties with this antibody fragment against B6. And what you see is that it's not just that they internalize much more in the cancer stem cells subpopulation, but also they have much more efficacy in reducing the levels of cancer stem cells, especially when you work with colonoscopy. So when you work with these 3D models in which they are in non-attachment. So it means that you have the possibility to increase, at least in vitro, this efficacy. And this is also to show you, and I think it's my very last slide, this is also to show you that in vivo, by using these systems, we were also capable of reducing the level of circulating tumor cells from these tumors. And it was a very nice reduction of the level of circulating tumor cells that we will see in the bloodstream, meaning that effectively you can actually perform and you can indeed influence very much how these cells will detach and go into the bloodstream and perform metastasis, etc. So these are certainly options that we have to keep in mind for the future. And that's about it. Just to thank you all of our funders and of course collaborators, and particularly to Patimobi in Porto and to the Instituto Nacional de Ingeniería Biomédica, INEP, and many others. Thank you.
Thank you very much, Dr. Schwartz. Now, our last presentation for the nanomedicine segment is Dr. Josep Samitia, the Director of IBEC and a Professor at the University of Barcelona. Their expertise is in nanotechnology research applied to biomedicine. Seem to have some minor difficulties with Dr. Samitia at the moment. Oh, Dr. Samitia, can you hear me?
Okay, I am here.
Yes, we can hear you. That's great.
Okay, sir. I think that I need to change, I don't know if you look correctly, my screen. Sorry, one moment. You look correctly my screen?
Not just yet. Are you able to get it into full screen?
Okay.
Yeah, towards the bottom on the right-hand side, there should be full screen mode on PowerPoint.
Yeah.
It's in presenter mode now, so if you go to the bottom, there's the three dots.
Three dots, yes. Yeah.
And then you should be able to hide the presenter view. Okay. Oh, we'll be back in one moment. Oh, we'll be back in one moment just while we resolve the issues with the screen. Thank you.
Okay. It is okay? Okay. So good morning, and sorry for this delay to start my talk due to technical issues. I would like to share with you the work concerning the use of nanoparticling in the substrates in order to regulate the architecture of intercellular communication during mesenchymal cell condensation. So this is a work that has been performed in the Institute of Bioengineering of Catalonia. And this is a research line that is concerned about the intercellular communication. So in pluricellular organisms, intercellular communication guarantee a continuous and efficient flow of biological information between cells and their surrounding, and is essential for the adaptation to a community environment to sustain homeostasis, cell specialization, and correct function. Here in the figure, there are a nice example of how fibroblast growth factor propagation during the lateral line formation in zebrafish, in which cells evolve to form these luminal structures that allow for a coordinated and enhanced response. Initially, a freely diffusive ligand model was proposed for fibroblast growth factor activity. But in this paper from Durdo et al. In Nature, published in 2014, they demonstrated that fibroblast growth factor become especially concentrated in microluminal structures that assemble at the center of these organs and especially constrain its signaling activity. Luminal signaling provide a potentially general mechanism to locally restrict, coordinate, and enhance cell communications within tissues. In the case that we are working in the last years, the mesenchymal cell condensation, this is a prevalent morphogenetic transition in which mesenchymal stem cells cater to form intimate cell-to-cell contact. This is the basis of steatogenesis, leading to both cartilage and bone formation. So in the figure, the mesenchymal stem cells usually concentrate to form these prechondrocytes, and these structures are the basis for early chondrogenesis and until to form the generation of new cartilage and the differentiation of these mesenchymal stem cells. So to form that, and during this process of mesenchymal cell condensation is a concurrent to the formation of an extensive gap junction communication network. And this is an essential element and this essential process of architecture in order that to promote the communication between the cells inside the 3D tissue. As we can see in the example here, where the expression of the gap junction protein alpha gene is followed in the development limb of mice. So during all this time, you have worked in the analysis of this communication in the case of chondrogenesis to form cartilage tissue. And in this case, connexons are formed by homo or heterohexameric arrays of connexin proteins. Connexin hemichannels or connexons accumulate and dock with opposite connexon from adjacent cells to form dense gap junction plaques, which are continually regenerated by the addition of connection subunits at the edge and internalization from the center of the plaques. Such continuous renewal ensure the maintenance of the established intercellular communication network. So here in the figure, it is possible to observe these connexons and the plaques of the gap junctions. So this is the process finally to produce cartilage tissue. And cartilage tissue is a complex natural tissue that shows multi-level organization in which nearly cylindrical cells are aligned and connected side by side and end-to-end along the proximal distal axis of the limb. Cartilage is a non-vascularized tissue in which the communication between cells occur both by diffusion and by direct cell communication through gap junctions. The proper distribution of gap junction is of capital importance in cartilage architecture to obtain a well function of the tissue. Cellular interconnectivity through gap junction is established during this process of embryonic patterning that is still maintained at different levels in healthy adult cartilage. Moreover, osteoarthritic patients showed a loss of the usual ordered distribution of connexins from gap junctions in the cartilage damaged zones with higher levels of connexin 43 and connexin 45 are expressed in the damaged proliferative zones. So this process that appears in the first steps of differentiation to form cartilage from the migration of the cells to form these the aggregates, and finally to produce the differentiation, is a complex cellular process that requires a key time and size scales. The formation of precartilage condensations illustrate the complex nature of cellular process and the need for control of the cell microenvironment over multiple time and length scale. So that has been the work, and basically the work of one of my PhD students, Ignacio Casanellas, that at this moment he are finishing and to submit the thesis in the next weeks. And that work has been co-directed with Dr. Ana Lagunas from my lab. That is one of my senior researchers. And the main objective of the thesis of Ignacio has been to study the influence of cell adhesion in the establishment of gap junction intercellular communications during this mesenchymal cell condensation, and try to tailor this tissue communication architecture, modifying the patterning of the substrates. In order to perform that, you have the rationale has been that mesenchymal cell condensation is regulated by cell adhesion. Therefore, you use dendrimers, or in this case nanoparticles, dendrimers-based nanopatterns of the cell adhesive peptide arginine glycine aspartic acid, RGD, of fibronectin, to conduct a systematic study of the influence of local surface adhesiveness of the establishment of the gap junction during mesenchymal condensation. Weakly charged solid substrates can be nanopatterning in liquid-like order with large and well-defined spacing by absorbing polyaminoamine PAMAM dendrimers. This approach allowed us to obtain reproducible nanopatterns on large areas that are compatible with the standard cell culture protocols. So in the picture, we observe this RGD in the fibronectin proteins, and how we link the RGD to the dendrimers using this liquid process, the deposition, and after we can observe the distribution of these nanoparticles over the structure of different substrate, and in this case polylactic acid. So these are different examples or different maps about the distribution of these nanoparticles over the substrates, analyzed by atomic force microscopy. So in the pictures, in the upper pictures, they are the direct image, topographic image of the nanoparticles over the substrates, obtained in different concentration of conditions in the deposition of liquid. And in the lowest part of the figure, we observe the different contour maps of the densities, analyzing what is the minimum interparticle distance between the deposited dendrimers. So we observe a non-uniform distribution of these particles in the substrates, and that produce clusters, in some case of nanoparticles, so clusters of particles with distance, interparticle distance lowest than 70 nanometers. So 70 nanometers is a little the minimum distance necessary to allow the interactions between the integrins to form adhesion contacts. So here we show three different distributions of nanoparticles over the substrates, labeled S90, S45, and S18, and that represent that in the surface, the distribution in the surface of the different substrates, with 90% of the surface covered by clusters with less than 70 nanometers of interparticle distance, or only 45 of the surface with less than 70 nanometers of interparticle distance, or in this case, the lowest probability to obtain clusters, that it is the substrate S18, with only 18% of the surface covered by clusters with distance, interparticle distance with less 70 nanometers. So these are technological processes that allow us to obtain different distributions, different levels of clustering, or the coverage of the surface with different levels of clusters, showing this parameter of 70 nanometers as the key distance to obtain the interactions between the integrins to form adhesion of the cells. So in the next picture, we show a video about the first 40 hours when you introduce the mesenchymal stem cells over these nanopattern substrates, and you analyze the formation of this aggregation of this clustering. So the red circles indicate where are located some of these clusters, this aggregation of the mesenchymal cells, and these are the first process of these aggregates to promote after the differentiation and to start the development of cartilage tissue. So this process, we have analyzed also the process of evolution of the movement of the cells of this type of substrates, and in this case, during my talk, we will focus about the architecture that it is produced inside of each of these clusters. So we could culture the human mesenchymal stem cells on these nanopatterns under chondrogenic induction. So as control, you use pristine polylactic acid, that it is labeled E0, and fibronectin coated polylactic acid, that is labeled SFN, as controls of the system. And you analyze the evolution of different the formation of these aggregates, depending in substrates with different type of aggregates of these nanopattern dendrimers with the RGD. And we analyze what is the area of the condensates, and we obtain a maximum of the area with the case of S90, and also we analyze what are the density of cells in each of these type of systems. So we analyze that as the cell density increase in the condensation sites, intimate N-cadherin mediated cell-cell contact are established. The surface tension of the forming tissue has been found to be proportional to the cadherin expression levels. Therefore, to evaluate the condensate's surface tension, we examine N-cadherin gene expression, CDH2, and you found that the RNA relative expression of CDH2 decreased with the local surface adhesiveness of the substrate. Nevertheless, more stable condensate are obtained in S90 nanopatterns, analyzing the projected area of the cell condensate, and they retain the 3D structure until 14 days in culture, while the cell condensate in the rest of the substrates collapsed. So clearly, these structures or this nano pattern in the cases of S90 drive in a good way the process of condensation and maintain the conditions for the stability of these condensations. Also, we have analyzed the gap junction intercellular communications. For that, we analyzed the protein production and the structural organization, in this case of connexin 43. And for that because it's the most widely expressed and studied gap junction protein. And also it has been recently found that this protein modulates cartilage structure through its C-terminal cytosolic domain. So gap junction largely depend on the size of the gap junction plates, which it is determined by the number of arrayed connections. We measured the extent of the gap junction plaques through the immunofluorescence of connexin in the cell condensation after 6 and 9 days of chondrogenic induction. By skeletonizing connexin 43 immunostatic image, we obtained a 3D representation of the intercellular communication network from the skeletonized image. We analyzed the average branch length and the number of endpoint boxes. Shorter branch and fewer endpoint boxes indicate a more intricately shaped connexin 43 network. Gap junction interconnection increased with increasing local surface adhesiveness, with a peak at the S90 substrates. So in the next picture, we show this structure of the production of connexin 43 and the end point boxes that allow us to analyze what are the intercommunication inside of the system. And these are the examples of the values that we have analyzed. So this is because more protein expression does not necessarily imply a higher degree of international communication, as they need to be connected. In order to quantify the level of gap junctional intercellular connectivity, we analyzed the connexin 43 three-dimensional network of the condensates and quantify the amount of endpoint boxes. So, and we obtain in this case also this maximum for the S90 extractors compared with the controls and also compared with the other type of systems. So clearly the non-uniform clustering of the substrates is the best structure that facilitate the production of the connexin, but also the good architecture to increase the maximum the connectivity inside of the aggregates. So in order to analyze the functionality of this gap junction network, we observe the speed of biotinylated or fluorescent tracers following injection in a single cell. And this is the most common method to demonstrate the presence of the gap junction. So we conducted a neurobiotin tracer uptake assay in cell condensates. Neurobiotin is a channel permeable dye that can penetrate from the exposed connexin 43 connections and diffuse in bio across the gap junction network. So for that also we observe clearly that in the case of the substrate of S90 is where is produced the maximum diffusion of the neurobiotin inside of these aggregates. So analyzing all of that and to be sure that this process is mainly obtained for this process of or is managed or driven by this the integrin adhesion and the cadherin connection between the cells, we have produced some disturbance of the interaction with the substrates through the integrin blocking by the addition of RGD functionalized endocrine solution in order to decrease the level of production of connexin 43 in S90 cell condensates. and that is observed in that, but not in S18. And this indicates that integrin-mediated cell adhesion is responsible for the transduction and regulation of substrate information that affect the gap junction innovation networks. So also analyze the cortical actin assembly that can be observed in early mesenchymal condensate, the mechanism by which substrate information diffuse within the cell condensate to establish the gap junction network may involve the actin cortex. And the cell cortex is formed by an actomyosin network, which is located below the cell membrane. So in this case, when we perform a treatment of 6 days condensates with the myosin 2 inhibitor, in this case, that decrease the percentage of area immunostained by connexin 43 in the S90 condensate, showing that clearly this cortical actin organization is related with the architecture of the mesenchymal condensation process. So finally, that indicate that integrins are engaged in the process of architecture to form these aggregates and the network of these aggregates, and the distribution of this clustering of nanopattern of RGD that change the adhesion of the substrate in the first layers of the aggregation of the process of the mesenchymal stem condensation, drive the process of interconnection between the cells and what will be the level of organization of the different gap junctions inside of the aggregates. So in order to assure all of these process, we have performed different experiments to modify and to change aggregates from one type of substrate to another after 3 days or after 6 days. And in this case, we can observe that this process is reversible. So in this first phase of aggregation of the stem cells aggregates, the process of architecture process to produce and to differentiate the cells is a reversible process, and we can modify when we change from a good substrate S90 to a bad substrate at S18 and vice versa. So as conclusions, we observe that a bigger and more stable condensate that's obtained in highly adherent nanopatterns substrate, that in this case S90, optical balance between the integrin expression and surface tension regulate the mechanical stability of the cell condensates on the nanopatterns. The S90 nanopatterns show better gap junction network, and the establishment of this gap junction network during mesenchymal condensation is an adhesion gate mechanism in which S90 nanopattern configuration act as an on switch to trigger the cell response. Finally, the substrate information propagate in the condensate via cortex interactions. So finally, this is the basic part of the work, but the more interesting finally is that we have performed an in vivo trial using pelylate plus chitosan scaffold and introducing these dendrimers and these distributions of the clusters of the RGD dendrimers. And you observe that in this case, osteochondral defect that was produced in sheep knees, and after six months, you observe a strong recovery when you introduce this scaffold with these distributions of the dendrimers with RGD. So thank you very much, and my knowledge of my team, especially to Dr. Ana Lagunas, and the next, I hope, Dr. Ignacio Casanellas, that we will submit the PhD thesis very quickly. And thank you very much for your attention.
Thank you so much, Dr. Sametia. Now we're going into the Q&A segment, and our first question today is for Dr. Torelli. And that question is, are there any particular challenges associated with upscaling nanoparticle formulations in comparison to more conventional formulations?
In case of nanoparticle formulation, the most pronounced challenge is the scale-up due to the nanoparticles being prepared in the lab scale in a smaller volume, where you can freely choose your method of mixing or your method of purification. And when you want to transfer this to a GMP scale in a bigger scale, you would need to optimize the mixing process actually almost independent of the particle type you are using. It always requires some kind of a mixing, and you would need to also come up with a solution how you can do the purification of your nanoparticle formulations during the production. These are mostly the most two critical steps in such a scale up, which you would need to invest a lot of time when producing the nanoparticles for clinical trials, for example.
Great, thank you very much. And our next question is for Dr. Schwartz, which is, do you think that actively targeting drug delivery systems will improve their intratumoral accumulation in vivo?
Hello. Well, it depends. So it's true that if you, in normal terms, and I'm talking about what's seen in average when you work with in vivo models. So it's difficult to think that by the fact of adding in a drug delivery systems an active targeting moiety, you will substantially change what's the in vivo biodistribution. That normally doesn't happen. So it's true that most drug delivery systems, when they are injected IV in animal models, they biodistribute in a kind of similar way. So there is a strong uptake by the reticuloendothelial system, and in liver and spleen. There's also some accumulation, sometimes higher than others in lung, also in kidney. So at the end, this is more or less what you will see. So the fact that you add an active targeting is per se not going to change very much this biodistribution. But it's true that one thing is whether you might change this biodistribution. The other thing is what happens when you reach your target tissue. So, and this is important in cancer. So we've seen that by adding targeting moieties, you might help increase the internalization of these nanoparticles once they reach the target, the right target. So you will not change the biodistribution, but once in there, they will, make easier the internalization of these systems inside the cells. And this is something that you can actually see when you work with cancer stem cells. So by targeting them, you increase the level of uptake of these cells, and therefore you might have more efficacy in the mid and long term. So more or less, that's about it. Thank you.
That's great. Thank you very much. With that, it brings the nanomedicine segment to a close, and we will be returning today at 2 p.m. So that's in 1 hour 10, where we'll be talking about the preclinical models and imaging. So have a lovely lunch, and we'll see you again soon. Good afternoon and welcome back. So beginning today, we'll have our third thematic segment, which is on preclinical models and imaging. Our first presenter today, our first speaker today, should I say, is Dr. George Loudos, the co-founder and CEO of nTRACK. He has a focus on molecular imaging of nanoparticles and biomolecules, and today he'll be showing us about the benefits of preclinical real-time imaging.
Hello everybody, thank you very much for this kind invitation. For me, nTRACK is a very critical project because when it started, our company was at the very early stages and now we are much more mature. So I really feel very happy to be part of this project. I think it has assisted a lot our growth. So I'm going to speak about the preclinical real-time imaging, a tool that we have used extensively in nTRACK project. And I will try to give a little bit the concept of multimodal imaging and how this can help in ethical drug research. So BioNTech is a CRO company who is performing several imaging studies, but we are also a manufacturer of medical imaging systems for animal studies. So we have a double phase. And as you can see, we have our laboratories inside the research campus. We have our animal facility. We have our radiochemistry laboratory. We have our in vivo imaging laboratory and now cell studies. So we can carry out a full study, either as a CRO company or as part of a European project. And all this experience has made us think a lot about how it is best to use animals. We all know that small animal imaging is an important step in preclinical research. First of all, because imaging is in vivo and not in vitro. Secondly, because it's a non-destructive technique, which means that we can use the same animal and perform a series of studies over time on this animal. Also, each animal serves as its own control, which means that we can have multiple time points from the same animal and not a statistical time point from multiple animals. In many cases, we can image the entire animal simultaneously, which is also very important because we can get information from different parts of the animal body. And of course, using small animals is always the gap. We bridge the gap between the cell studies and the human studies. And we should remember always that many drugs are used for human imaging as well. When we speak about small animal imaging, we have many potential targets. So first of all, we can image small molecules, we can image peptides, we can image, we can image antibodies, we can image aptamers, nanoparticles, stem cells. And to do that, we have to use a variety of different scientific disciplines. When we speak about imaging, and if you're familiar with imaging from the clinical practice, we have also a variety of modalities. We have the anatomical images like CT, ultrasounds, MRI. We have also nuclear medicine images like PET and SPECT. And we have also optical imaging, a modality which is mainly used in preclinical research. So having this full portfolio of imaging modalities, many times it's a question to understand and to decide which one we're going to use. So when we speak about preclinical imaging, first of all, we need to understand the biology of the problem that we want to study. We have to see, for example, what are the potential targets on a tumor, on a specific disease. And then when we know what are these targets, we have to select some compounds that they can go to these targets. They can provide an imaging signal that we can detect. And this one will let us select the proper imaging modality. Then we have to perform something which is called labeling, and this means to try and attach on a specific compound, the peptide, the antibody, the stem cell, something that will give us an imaging signal. And that something can be radioactivity, it can be a fluorescent dye, it can be a contrast agent for X-rays, for MRI. So this process is called labeling. We usually have to test everything initially in cells, and then we have to work on small animals, and finally work on larger animals. And in a way, this is also the nTRACK story. We work with cells, we work with mice, we plan to work with larger animals. And finally, we want to translate this to the clinics. So if you see all these expertise needed, we understand that preclinical imaging is not something that one group can easily perform. We need the collaboration of more than one experts. And the most challenging thing is to find a way that these experts can understand each other. So it's really fascinating for me field, and with a huge potential in preclinical research. It's a long discussion to try and describe all the imaging modalities. So I'm trying to use a couple of slides to let you know what are the two things which are more important for us. So on the one hand, we have spatial resolution, which means how small are the structures that we can see. And then we have the sensitivity, which means how weak signals we can detect. We have, for example, imaging methodologies like X-ray and CT that they can image very small structures from a few micrometers up to the human level. We have microscopic techniques that they can detect the cell level, but they cannot, for example, detect thick tissues because the light cannot easily escape from the tissue. We have nuclear medicine techniques that they can go from sub-millimeter resolution up to the human level. And now when it comes to the sensitivity, we can see that the optical imaging and the nuclear medicine techniques are really sensitive, which means that we can have a very weak signal and still get images, while MRI and X-ray images need a much higher concentration of either cells or contrast agent. For example, in nTRACK, this is why we decided to use nuclear imaging so that we can radiolabel some gold nanoparticles and see them at very low concentrations, while the same gold nanoparticles can be tracked with X-ray imaging, but only when they are in higher concentration. So by using the combination of these two modalities, we are sure that we can detect them even when they are at very low levels. These are some typical preclinical studies. As you can see, we can get fantastic images when it comes to tumor imaging, bones, cardiology, nanoparticle imaging, lung imaging. And although these fantastic images are nice for preclinical research, there is always one bottleneck. And the one bottleneck is that the cost of those imaging systems is many times huge for many laboratories. Another thing that we need to take into account is that at the very beginning of testing new compounds, we don't necessarily want to have these fantastic images, because here we already have decided that our tracer is targeting a tumor. We have decided what is the best time point to do this imaging. So we think that there could be some more ways that we can test in vivo many compounds and try to minimize as much as possible the time from initial drug discovery till having one new drug. And this is always a challenge in preclinical research, not only how to make this time shorter, but also how can we image or test several compounds in an efficient way so that we are sure that we have more candidates for the clinical trials, or at least we have the optimal candidates for the clinical trials. And again, imaging plays a nice role in this chain. So up to now, to test these different compounds, we can do either biodistribution studies. Some groups are using imaging in clinical cameras. We have, of course, the nice preclinical scanners that I've shown before. And in our case, we tried to propose something in between as a company, using our experience from preclinical imaging. Using also the fact that optical imaging is a well-established technique, we decided to extend the optical imaging towards radioisotope imaging. So we developed a series of systems that they can detect the radioactivity in 2 dimensions in real time. And we wanted to introduce this aspect as one new tool which stands between ex vivo biodistributions and advanced 3D imaging. And in this way, we would like to answer as many as possible questions that are always present at the very early stages of developing new compounds. And the first question when we develop a new compound, of course, is to see if it reaches the target or not, if we have tumor targeting, for example. Then there are some more questions like how long this compound can remain on the target, if it's concentrating on other organ tissues over time. We have to decide what is the best injection route, if the compounds are stable post-injection, what happens at the first minutes post-injection, how long these compounds stay in the blood circulation. And all these are very interesting questions, especially for nanoparticles, because most of them, there are new compounds that we don't know yet their distribution. Some additional questions have to do with what is the best injected concentration, how shall we prepare the animals, what is the concentration of the target, and if we would like to do more studies, what is the best time point to take some mice for biodistributions, and if we have access to 3D imaging, when is the best time to do this nice tomographic imaging. So, as many groups are working with biodistributions and few groups are working with nice 3D imaging systems, we decided that we could have something in between, some a technology that can provide multiple time points on the same animal and let us decide what are the latest steps when testing a new compound. So some typical examples include oncology. For example, here we have one compound that targets a tumor, but by doing this real-time imaging, we can see the distribution in the bladder, in the tumor, in the intestines over time, and we can identify when it is a good time point to do the nice 3D imaging. We can do the same with PET tracers like FTG, which is mainly intended to target tumor cells. So we can do again a nice dynamic study, even with frames of 30 seconds. We can draw time activity curves. And by having this information, we can understand how our tracer works, and we can decide what is the best time points to select a few mice for biodistribution, and in the end, reduce the number of mice we need. The same has been done for lung studies, and lungs are also an interesting case because when we give a compound through the trachea, we mimic inhalation, and then we would like to have a tool that shows us in real time how the compound is washed out through the lungs. So to also speed a little bit, one interesting thing is that many times when we do ex vivo biodistributions and we have no ability to do images, we have bad injections or our compounds are not stable and they form aggregates. This means that if we blindly run a study with 20 or 30 mice, it's likely that no compound will ever target a tumor or what we want to target if we have a bad injection or if we have aggregates. And this means that the total study will give us no results. So again, imaging in a simple way is a tool that helps us design the rest of the study. And so why this means a refinement? First of all, imaging is a non-invasive procedure. The animals are kept warm, they are anesthetized, so we can ensure minimum pain and stress for the animal. But the most important thing is that when we do real-time imaging, we have the ability to do very short studies. For example, in the upper level, we have a study of 3 minutes compared to a tomographic study of 20 minutes, which means that all the animals here were stressed for only a few minutes compared to the 3D imaging. So this again improves a lot the refinement of the animals. But obviously, the strongest advantage of real-time imaging, of imaging in general, is the animal reduction. To get these nice biodistribution curves, we have to sacrifice a different number of animals at 1 hour, 2, 4, 6, up to 24 hours post-administration. And to get this statistical variation on the curves on the right side, we have to sacrifice more than 5 animals per time point so that we are sure that the statistical variation is low. However, when we do real-time imaging, we can get the same information from a few animals over time, so we can have all those time points from the same animal. And by using a small number of animals, we can get the same statistical information at a much lower cost. To give you a small example, assuming that we have 3 study groups, for example, we have one new compound, a control group, and a standard compound that we want to test. In this case, we have 3 study groups. There we have 5 time points per group, 6 animals per group, and all this would lead to 90 animals. However, if we decide to use imaging, we need only 18 animals to do the same study. And even if we decide to sacrifice 1 animal per time point, and this is something that we do sometimes to confirm the imaging data, again, we need about 23 to 28 mice instead of 90. This example is very small. Usually in typical studies, we don't have 3 study groups. We have more than 5 groups. We should have more than 10 animals per group. We have more than 5 time points. So you can imagine what is the advantage that we have in terms of mice, and not only mice, because when it comes to the cost, we should not think on the mice. The cost of a mouse can be €2 if it's a normal mouse, but it can go up to €1,000 if we have a specific mouse model. The cost of hosting mice can be a couple of euros per day per mouse, but assuming that many studies last for more than 3 months, you can see how easily the numbers increase. And then if we use specific cell lines to create tumors, we have again to prepare a big mouse population. If we want to use radioisotopes, we have to think many times that the cost of specific radiopharmaceuticals can be several hundreds of euros. So by multiplying this by 500 or 50 mice makes the number increase very fast. And of course, when we do biodistributions, we have to analyze all these mice post-mortem. And of course, this is again a big burden. And something that many times we don't take into account is that we decide the study with 100 mice versus a study with 20 mice. Many times this big study needs a different planning. We have to split the groups. We may have to execute the studies at different time points. So again, the results may not be very robust. So in total, imaging can really simplify our life. And I would like to add one more point regarding replacement, because of course, it's not easy to replace a mouse with something else. However, when it comes to imaging, many times we want to optimize the imaging protocols. So we have designed one mouse phantom. We did it accidentally to serve our own internal needs. And that helped us to optimize protocols for specific organs imaging, because we can just use tracers, fill specific organs with these tracers. It can be just simple isotopes, so we don't have to buy expensive pharmaceuticals. We can play with different ratios in organs, tumors, to see, for example, if we can image a very weak spot when there's a high concentration in the bladder. We can also test clinical cameras with mice in case we would like to see if they can help us. And in all those cases, we realized that there was a way to mimic animal studies without using mice. And it was a surprise for us that this phantom was a need, because many groups worldwide have asked this phantom from us. So it looks like there are also ways to replace animals, especially when we start working with different new modalities. And many times we can have something in between a phantom and the mouse. So this is our mouse phantom, and reaching the conclusions, I think that in vivo imaging, apart from providing these nice, fascinating images, gives a real value to preclinical research. First of all, we can use imaging to acquire a full data set that gives us the same information as in the case of biodistributions. We can start with two-dimensional real-time imaging to get an evidence of our experiment. But if we move to 3D real-time imaging, then information is quantitative, is comparable to the biodistribution studies by using only a fraction of the number of animals. This helps us to save a lot of experimental time and costs, as I tried to describe before, because there are many hidden costs when we do animal studies. And also, if a group introduces imaging in its workflow, the experience shows that the daily workflow changes a lot. We start working in a completely different way, and we feel more and more eager to get images before start sacrificing mice. So this is really helpful and, of course, ethical. So from our side, we really believe that preclinical imaging should be introduced as an official method for biokinetic studies, because it has a lot of advantages over ex vivo biodistribution. And we believe that imaging can make a huge impact in preclinical research. And I think that this is something which is now already accepted, not only from the scientific community, but also from many companies who invest more and more in imaging. So I would like to thank you for your attention. This is the BioNTech team, and we are very happy to contact us anytime for questions or for potential collaboration. Thank you.
Thank you very much, Dr. Loudos. I found the dynamic studies particularly fascinating. It's some impressive capabilities from BioNTech. Our next speaker today is Dr. Mario Morota from LATAT, where he's the head of regenerative medicine labs. Today, he'll be telling us about the application of large in vivo models for regenerative medicine.
Hello, good afternoon to everyone, and thank you for joining the workshop today. Today I will talk about the small and large animal models that we use it for studying muscle injuries in the nTRACK project, in the nTRACK approach. I don't know if it's working, the presentation. Can you see my presentation? Okay. That's okay, that's working. So what we do always is try to develop animal models which imitate the human pathologies. So we try to translate to the animal models the problems, the pathologies that we can see in the human clinics. So in the animals, we can study, we can evaluate the evolution of the diseases, evolution of the pathology, and also to evaluate the new therapeutic strategies for the treatment of the injuries, in this case, muscle injuries, and then translate all this information to the human clinics for future therapeutic approaches. Our strategy is normally going, in the preclinical studies, is going from the small animal models. When possible, we start with small animal models, and then we move to the large animal models, which are closer to the human situation. That was the nTRACK approach to first testing the medical approach in nTRACK in small animal models, then move to large animal models and leave the project at the door, just ready to go to the first in human clinical trials. So in the case of nTRACK, we use both animal models, a small one and the large one, the rat and the sheep animal models for studying muscle injuries. And we developed the new large animal model in the sheep, which is a new model that we have generated within the project, within nTRACK, based on the information that we had from the previously developed small animal model in rats. First of all, let me introduce the small animal model in the rat. It's a surgical model of muscle injury, which is an excellent model because it can closely mimics the human muscle injuries that we can say in the athletes, in human patients. This model is based on the use of a medical device, commonly used in the hospitals and clinical settings for taking biopsies. This is an automatic biopsy gun with a needle that, when placing the needle transversal to the muscle and then shooting the gun, the needle goes into the muscle and take out a biopsy, producing a muscle fiber rupture. This transversal biopsy, this transversal injury, taking out the muscle section, imitates the muscle fiber ruptures that we are produced in humans when running, when doing sports, when working, or in an accident. So the stretching of the muscles, breaking the muscle fibers, can be imitated by the transversal biopsy in the muscle, as we produce in this model. What happens in muscle? You can see here a transversal section of the gastrocnemius muscle of the rat. This is the area of the metagastrocnemius muscle in the animals. The muscle that we always injure in the animal models. We know very good the anatomy of the muscles in the hind limb in the animals. And this is the area of the injury that is induced in the muscle. The needle crosses the muscle from side to side, taking out a biopsy and producing the muscle fiber rupture in the muscle. And using this model, we can evaluate the whole process of muscle regeneration in the animals, which is very important to be able to evaluate all the different stages. For example, the generation after the injury, the inflammation period, the muscle regeneration to stimulate the muscle healing, and also the fibrosis or the remodeling at the end of the process. You can see here in green, for example, a specific marker for fibrosis that we use a lot in our studies, which is collagen 1. We can see the strong production of collagen 1 during the muscle regeneration process. And at the end, we can see the scarring that remains in the site of the injury after the muscle healing process. We can also evaluate, as you can see here in the bottom line, a marker for muscle regeneration, which is a very important marker here in red color, which is a marker that label the new regenerating muscle fibers that appear in the site of the injury to regenerate the muscle injury, the muscle section. This marker appears in the muscle fibers at the beginning, at the early stages of the muscle healing process, and at the end disappears because it's replaced by the adult isoforms of the myosin heavy chain, which is a very good marker to evaluate the muscle healing process. It's like a traffic light in the middle of the muscle healing process to evaluate what is happening in the muscle healing process. And we can also evaluate the animal models by clinical imaging. You can see here, for example, in the rat, the study of the evolution of the disease using 7 Tesla's magnetic resonance imaging, a high definition MRI, in the 3 different views, the coronal, sagittal, and axial views. And you can see here in the right leg of the animal, the edema, and how the edema is progressively disappearing during the muscle healing process. It's reabsorbing by the muscle after the injury, during the following days after the injury. The most important thing is that we have a rat model, an animal model that closely mimics the human injuries that we can see in professional athletes. For example, you can see here 2 different images, MRI images from 2 athletes from Football Club Barcelona, a grade 1 and a grade 2 types muscle injury in these patients. And you can see how our rat model closely imitates this type of injuries. So we have an excellent animal model reproducing the human pathologies, human muscle injuries. So using this model, we evaluated different treatments. You have here some examples of treatment that we tried with this animal model in rats. The first one was platelet-rich plasma, PRP, in combination with exercise, and the second one was a cell therapy-based treatment. The first of the treatments were the results of the study were published in 2017 in American Journal of Sports Medicine, which are one of the best journals in sports medicine in worldwide. And we demonstrated that the post-injury exercise and platelet-rich plasma therapy improve the skeletal muscle healing, both treatments. But importantly, these treatments are not synergistic when combined. And this is very important because our results in the preclinical model in rats, using the animal model in the rats, corroborate the data obtained in humans in randomized control trials. These authors demonstrated that a single PRP injection is not having any added beneficial effect over exercise post-injury that were doing these athletes, demonstrating that the combination of PRP and exercise is not providing a synergistic effect. Similar data that we obtained in our model. So demonstrating that we have an excellent model to evaluate also the treatments in the animals and in humans. The other treatment was the cell therapy-based approach that, as you can see here, we also published in American Journal of Sports Medicine the last year. And we demonstrated that the muscle precursor cells enhance the functional muscle recovery. And in that case, is showing a synergistic effect in combination with exercise, which is a very important thing. And in contrast with the PRP approach. Let me go deep into this study because it has a very nice results and has also the cell therapy in a similar approach that was planned to be performed in the nTRACK study. Just to show you the study design in this work, we perform a pre-injury training period in the animals to train the animals for 2 weeks, doing a progressive pattern of exercising, increasing daily the duration and speed of the running treadmill in the animals during 14 days. At the end of the training, we perform the surgical injury in the animals as described using the biopsy needle. And then we administered the different treatments. The first one was the ultrasound guide intramuscular injection, 36 hours post-injury, always following all the process under ultrasound control, always monitoring the injection, as you will see. The other treatment was a combination of the cell treatment in combination with the post-injury daily exercise, with a pattern of exercise also for 14 days, also increasing every day the speed and duration of the exercise during 2 weeks. At the end of the study, we evaluated the muscle force analysis in the animals, and we collected the samples to evaluate the histological outcomes in this group of animals. Let me show you how we establish the muscle precursor cells in primary culture. We can take muscle biopsies from gastrocnemic muscle from the rat, and then using an enzymatic digestion protocol, we can collect the muscle precursor cells and put it in cell culture. And we can also expand these cells in primary culture to get a large number of cells for the cell treatments, for the cell therapy approaches. Before using the cells for the cell treatments, for the cell therapy, we always corroborate the myogenic properties of these cells in order to provide the ability to fuse and form polynucleated muscle fibers in culture, to demonstrate the myogenic properties of these cells. So as you can see here, we can demonstrate that these cells has a strong potential to fuse and form polynucleated muscle fibers, but not only form muscle fibers, but also are doing functional muscle fibers. As you can see here, the cells are spontaneously contracting in cell culture. So these cells can generate functional muscle fibers in vitro, demonstrated that they have a strong myogenic potential to be used in the in vivo approaches. Then we injected the cells into the animals after the injury, always followed by ultrasound control. As you can see here in this video sequence, you can see here in the top of image the echogenic, the white echogenic part of the indicating the muscle injury in the gastrocnemius muscle in the rat, and how we can reach the muscle injury with a needle to inject the cells exactly in the site of the injury. As you can see here, we can go under ultrasound control and release the cells exactly in the site of the injury. The results of this study were that both group of treatments, both cell and cell exercise treated animals, showed a total muscle force recovery 14 days after the injury, which is very important because these animals had a functional muscle. From the functional point of view, the muscle was totally recovered. This important data was also corroborated by the histological outcomes. We measure the size, the caliber, the growth of the muscle fibers, the regenerative muscle fibers in the site of the injury in these animals. And you can see here how both group of treatments has a more than twofold of increase in the size of the muscle fibers in the animals in the regenerating area. And we can also demonstrate that the cell treatment and the exercise had a synergistic effect. The cell treatment has a strong potential to recover, to promote the muscle healing, but the cells and exercise had a synergistic effect. It's even more effective. We also evaluated the fibrosis in these animals, measuring the levels of collagen 1, and we saw a strong decrease in the intramuscular fibrosis in these animals, which is also very important in order to prevent the scarring after the injury and to prevent the re-injury in these patients. Another injury in the site of the muscle injury. We also evaluated the levels of the developmental myosin heavy chain, the marker of muscle regeneration, and as you can see here, we saw a strong decrease and a faster replacement of the developmental myosin heavy chain isoform by the adult isoform of the myosin, which indicates that the treatments induce bigger and more mature muscle regenerating fibers, indicating an accelerated muscle healing. Our treatments are accelerating the muscle healing process in these animals. So as a summary of the muscle treatments in the rat model, we can say that all treatments provided a beneficial effect, cell therapy, exercise, and PRP. But muscle precursor cells demonstrated the strongest treatment efficacy, especially when combined with exercise. Cell therapy and exercise synergistically collaborate, synergistically enhance the muscle regeneration. So this data, this preclinical data in the rat, can suggest potential therapeutic strategies to be used in humans in the future treatments. With all the information in the small animal model, then we went to nTRACK to generate a large animal model in the sheep. As you know, nTRACK aims to develop a safe, scalable, and highly sensitive multimodal nanoparticles for stem cell tracking. The nanoparticles are intended to load, to label the cells, and these cells are going to be injected in the animals for cell tracking, to evaluate the biodistribution, localize the cells, and also evaluate the participation of the cells in the muscle regeneration process in the animals. So, in the context of nTRACK and based on the previous data of the small animal model in rats, we generated a new, for the first time, a new animal model in the sheep in order to be used in the nTRACK project. And this is how we generate the muscle injury in the sheep model, which is very similar to that performed in the rat model, just a scale up to the size of the ship using also an automatic biopsy gun and automatic needle, which is bigger in size. The needle caliber is 4 millimeters, which is bigger to increase the size to the size of the ship. And then the procedure is very similar to in the rat. We always localize under ultrasound control the muscle that we are going to injure, to provoke the injury. We place the needle in the transversal position to the muscle, and then when shooting the gun, the needle goes into the muscle, taking out a muscle biopsy and generating the muscle fiber rupture. As you can see here, we can evaluate the site of the injury and also the size of the injury generated in the animals after the surgical procedure. In the right panel, you can see the defect, the separation of the muscle fibers generated by the muscle biopsy. So we're generating the muscle fiber rupture in the similar way that produced in humans by stitching the muscle. So highly reproducing the muscle injuries that we detect in muscle patients, in the human patients. And you can see here what happens in muscle. This is the histological view of the muscle injury in the sheep. The needle goes into the muscle 2.2 centimeters, generating the injury, which is very similar to that produced in the rat. The only difference is because in the rat, because of the size, the needle crosses the muscle from side to side and crosses all the muscle. In the sheep, the needle goes into the muscle only 2.2 centimeters. But the injury is exactly is very, very, very similar and with exactly the same procedure. So once defined the injury in the sheep model, we evaluated the longitudinal evolution of the disease of the skeletal muscle injury in the sheep, defining 4 different groups of analysis at 4 different points in time. Then we evaluated histological outcomes, the histological evaluation in the animals at 3 days post-injury, at 7 days post-injury, and 14 days post-injury. And actually, in the last group of animals, we evaluated the histological evaluation at the end of the testing process, 20 days after the injury. But we also evaluated the clinical imaging analysis, the clinical imaging data of the muscle injury in these animals, to have both histological data and clinical imaging data by MRI. As you can see here, this is the histological and immunohistochemical analysis, histological data of the muscle injury model in the sheep. We evaluated not only the evolution of the muscle injury by histological approaches, but also by immunohistochemistry, evaluate specific markers for angiogenesis, for fibrosis, and for muscle regeneration. And as you can see here, we demonstrated a strong neovascularization of the injured area with the marker alpha smooth muscle actin, which labels the new vessels going to the muscle injury area, indicating a strong angiogenesis in the site of the injury, probably because the muscle injury needs some nutrients, oxygen to regenerate. So we can see a lot of vessels invading the injured area. We also evaluated the fibrosis, the production of collagen 1 and the position in the muscle. We detected a strong production and deposition of the collagen 1 in the sheep model. And we also determined a large number of neuroregenerative muscle fibers in the sheep model. And this is very important because we can see a lot of muscle fibers at the end of the process, at 28 days after the injury, which is very important because when compared with a small animal model in the rat, we can see that in the sheep, in the large animal model, muscle regeneration process takes more time than in the rat model. As you can see here, the rat model, 26 days after the injury, is practically recovered the muscle injury, but the sheep, in the sheep, we can still detect a lot of muscle regenerating fibers, indicating that the muscle process is delaying, is taking more time than in the rat, which is very important because it's more similar to the timing that we always see in human patients, in the muscle injury in humans, that normally takes around 6 or 8 weeks after the injury. We also evaluated the muscle injury evolution in the SHIM model by MRI clinical imaging, 3 Teslas, high definition clinical imaging. As you can see here, we can evaluate the edema that persists during the different days, during the whole period of analysis in the SHIP, also in the 3 views, in the coronal, sagittal, and axial views. And this is again important because when compared with the rat model, with the small model, we can also see that intramuscular edema is still clearly visible in the SHIP 28 days, 1 month after the injury, whereas in the rat it's practically disappeared. Also corroborating that is very similar, is more similar that the SHIM model than the information that we have from the human patient, from the human injuries, human muscle injuries. Summary of the animal models in nTRACK, we can see that we have available both small and large animal models of muscle injuries, which closely imitate the human pathology. The large animal model more closely reproduces the human pathology than the rat model of the muscle injuries. And animal models permit the evaluation of the cell tracking, cell therapy, and the pharmacological approaches of skeletal muscle injuries. So the animal models permit to study, to perform, to evaluate potential therapeutic strategies in preclinical studies prior to initiate the first in human clinical trials, prior to go to the human clinical trials, which is very important to have all this information before to go to humans. Just to finish, let me just give a brief overview of 2 other projects in which we are using large animal models. For example, in SBR, in Smart Bone Regeneration project, we are using the large animal model, the SHIM model, to perform the preclinical testing of the implantation of scaffolds in the sheep, in which we have generated critical size bone defects in the femur to promote bone regeneration. So in these animals, we are going to produce large bone defects, generating a large defect in the femur of the sheep, and then implanting a smart scaffold to promote bone healing and also to evaluate what is happening inside the bone, to evaluate, to monitor the bone regeneration in these animals. We're also working on the Triangle project, which is directed to perform the 3D bioprinted personalized scaffolds for tissue regeneration of the ankle joint. In Triangle, we are going to use a similar approach that in nTRACK, using first the rat model for the previous testing of the bioimplants in the small animal models, and then with the optimal bioimplants, we'll go to the SHIM model. And performing the preclinical testing of 3D bioimplants in animal models of Achilles tendon injuries and also astragalus articular cartilage bone defects in the ankle joint to promote tissue healing, generating 3D bioimplants specific, personalized for every patient, every individual, and also for every tissue. 3D bioimplants for tendon repair and other implants for cartilage repair in the animal models. Just let me thank you to my team, the Regenerative Medicine Laboratory at LEITAT, all the people that has participated in all this work during these years, and of course all the team that has been working the nTRACK consortium during these years. Thank you very much for your attention.
Thank you very much, Dr. Morota. Our final speaker of this segment is Professor Lang Tran from the Institute of Occupational Medicine, where they are the principal scientist in computational toxicology. And today they'll be talking to us about the origins of Alzheimer's and its potential treatments.
ladies and gentlemen. I'm going to speak to you about our research at the Institute of Occupational Medicine.
Full screen presentation mode, please. I believe that's okay. Yeah, thank you.
Okay, fantastic. So I'm from the Institute of Occupational Medicine. I collaborate with LATAT on many occasions through the framework programs, and we work really on nano safety, the safety of nanomaterial, industrial nanomaterials. However, in parallel, because we are Institute of Occupational Medicine, where traumatic brain injury is very much on our table every day. So we also work on neuroinflammation and neurodegeneration. I was invited to give a talk to the nTRACK workshop. And to be honest, I was wondering on how to pitch my presentation. However, the last presentation has assured me that maybe this is the right audience and this might be of interest to all of you, is because as you will find out, I hope you find this talk very interesting. Okay, so everybody know what Alzheimer's disease is. It's a neurodegenerative disease named after Dr. Alois Alzheimer. It was first presented in 1906 at a medical meeting, perhaps very similar to our workshop today, where a case study of a 51-year-old woman suffering from a rare brain disorder was discussed. And of course, they present brain autopsy. And in this autopsy, you can see that there are plaques, there are lesions, and there are protein tangles, and they are characteristics of this disease. And as everybody knows, currently there's no known cure for Alzheimer's disease. But however, by presenting what we know so far, and especially to the community of people in these audiences, maybe I hope to excite you and urge you to contribute to what we know so far, and hopefully we can find a proper cure for this disease. Now, in all this type of disease, it's good just to flash out the cost of it. You know, from when I compiled this talk, there's about 46.8 million people in the world suffering from dementia. And the estimate cost of it by the 2010 statistics was about 818 billion US dollar. Okay, and this is bound to raise into trillions of US dollar by the year 2030. And most of this actually goes into the social care because there's no cure, and the patient can live on for years before they die. So, without further ado, currently, there are two kinds of hypotheses about Alzheimer, about the beginning of the course of Alzheimer. And there's actually several hypotheses, but I'm choosing the two that are very prominent at the moment. And they are the vascular hypothesis and the amyloid cascade hypothesis. And for the rest of the presentation, I will discuss how those two are actually complementary to each other and how it helped to move us forward. The vascular hypothesis, as you know, believes that Alzheimer begins in the blood system, and especially in the cerebral vasculature of the brain. And the amyloid cascade hypothesis, in short, believe that everything begins with this plaque that was observed, and the plaque would then initiate a cascade of events leading to neurodegeneration. So before we go any further about the mechanism of Alzheimer's disease, I just want to remind everybody, I assume that we all know, to the left here, you have what is typical of a large blood vessel. So you have the blood in here, you have an endothelial layers, and underneath there, you have a lot of smooth muscle cells. So this actually brings the talk to the similar level to the last talk about injury and so on. So we have a lot of smooth muscle cell in here in the large blood vessel. However, as you move from the large blood vessel to the capillary, to the small blood vessel of the brain, what you find is that the smooth muscle density is reduced. Okay, so by the time you reach the small vessel of the brain, as you see, what you see in here are the blood inside, and then an endothelium layer, okay? And then right behind it, you get straight into mission critical area, okay? So behind the endothelium in the blood vessel, let me repeat, you have a basement membrane and a lot of smooth muscle cell. By the time you get to this small blood vessel, the capillary of the brain, then what you see here is, again, the endothelium with a thin basement membrane. And behind that, you're into mission critical area where you have all the astrocytes, the neuron, the microglia, and so on. So the smooth muscle cell disappear, but it doesn't mean that the smooth muscle cell function are no longer required. Okay, so this is a very important thing. If we go back to the big vessel again, if there's any injury in the endothelium, then it would stimulate the smooth muscle cell to proliferate, and they begin to make collagen. I think our last speaker also spoke about this function to some extent. Okay, and but when you move down here to the small vessel of the brain, then the pericyte has taken over the function of vasodilation and constriction that are attributed to the smooth muscle cell in the larger vessel. However, the proliferation and the generation of collagen, which is typical of smooth muscle cells, is now passed over to another cell, and that is your astrocyte. Okay, so this is something that we have to bear in mind. In the blood vessel, if there's an injury, it stimulates the smooth muscle cell to proliferate and make collagen. In a small vessel at the brain, if there's any injury that cut across the blood-brain barrier, then the astrocyte will put on the smooth muscle cells hat and also begin to proliferate. However, it will generate another protein, which is not collagen, but it does everything that collagen does. Okay, just remind ourselves again that, you know, collagen is actually made eventually generated in the form of fibers that you see in here. And it begins as precursor collagen known as pro-collagen, okay? And the pro-collagen cleaves and into oligomer that would then join together to make a fibrous entanglement, as you see in here. And when the astrocyte begin to generate a material for repair, they use a material called called amyloid. Okay, and the amyloid also begin as a precursor, and it is cleaved by beta and gamma secretase. It is then joined together into protofibril and into amyloid fibril. So you see, if there is a damage that cut across the blood-brain barrier, then it is the amyloid, it is the astrocytes that step into a smooth muscle cell function, and it generates something very similar to collagen. As you can see, this is amyloid, this is collagen, and it does the same thing. It just tries to heal the wounds, so to say. So what I want to point out in here is that in the case of an injury, the astrocyte generate a type of material like collagen. Currently, in the Alzheimer research, people do not appreciate this parallel between collagen and amyloid, and they tend to say that the amyloid is misfolded. No, it's not misfolded. The amyloid has been mobilized and folded just like a collagen to do something very similar to what the collagen does, which is tissue repair in here. Okay, and the amyloid, of course, is generated as a response to injury of the blood-brain barrier at a cerebral capillary level. Okay, and so far we know that amyloid can be produced by platelet. It's produced by astrocytes, as I point out, and in the ultimate case, it will be produced by neuron. Okay, there's an additional function which we do not see in collagen, but we see in astrocyte, is that it has been reported that this protein is actually antibacterial. So that's quite interesting. And again, I want to emphasize that the astrocyte is a multitasking cell. Okay, it takes on the smooth muscle cell function and produces amyloid fiber from the amyloid precursor. So very similar to what the collagen's been produced in the case of injury of the large blood vessel. And in neuron in particular, the precursor amyloid is actually a protein used for synaptic plasticity. Now, here's some of the nice pictures that we had from the plaque. And I like all of you to concentrate on the fact that they're very small. They're about 5 to 20 micron, okay? And they can be very condensed plaque, or they can be diffuse plaque, okay? It shows you that some of the plaque look like there's tissue contraction and the wound is being healed. And there are diffuse plaque in which the wound doesn't heal properly. And so this is something that can be used to explain the origin and the progression of this disease. Now, as you can see, if you think of the plaque as an injury, then there are several ways the injury can take place. Now, if the injury is only on the blood side, in other words, it only affects the endothelium and it doesn't cut across the blood-brain barrier into the brain, okay? So if you imagine the first type, the lightest type of injury is you have an endothelium dysfunction, you have a damage to the endothelium alone. Then the endothelium begin to activate the receptors such as ICAM, VCAM, the RAGE receptor, which is a very signifying receptor of acute inflammation of the endothelium, and it generates cytokines that would then attract the immune cell to the area of injury. So on the right in here, you can see the receptor activated, and on the left in here, you will see the release of NOx, and it leads to the reduction of NO, and this is a landmark of endothelial dysfunction. And the other thing that happened as a consequence to the injury to the endothelium is that you have angiogenesis as manifest by the upregulation of HIF1 alpha, which lead to VEGF. And of course, the endothelium cell will also release the protease MMP2 and MMP9. This is to regulate the basement membrane, because the effect of the injury on the blood side in here and the cytokine release will also activate the astrocyte, and they begin to produce some amyloid that make the basement membrane thicken. So the MMP2 and MMP9 is there to try to modulate, to keep the basement membrane smooth and not over construction of the basement membrane, which is fibrotic. Okay, but one thing we also find very interesting is that while we know that as a consequence of the injury, there's release of thrombin and interact with the plasma fibrinogen, etc. And there is of very important that we see the rise in the microparticles level in the blood. And this microparticle has been observed in many cases, like following lung inflammation due to air pollution, for example. And nobody knows the nature of these microparticles in the blood until recently. With COVID-19, people actually characterize these microparticles, and they discover that they are made of clotting material. They are micron size aggregation of thrombin and fibrin and fibrinogen, and they are now relabeled as microclot, actually. But they are basically the consequence of the damage, as you see. And this microparticle flow down the bloodstream, and they interact with the endothelium cell downstream, and it has the ability to activate this endothelium downstream. And most interestingly is that, of course, when you have an injury to the endothelium, is that it attracts platelet, as you heard from the last presentation, and the platelet will come to the site of injury, and it begins to release amyloid. And this amyloid are known as amyloid 140, okay? And this lesion are known as cerebral amyloid angiopathy in neuroscience. But basically, it's not nothing more than a fibrotic scar of the amyloid kind. And to stress the second point is that the platelet aggregation and the release of amyloid is not unique to the cerebral vasculature of the brain. You can see plenty of this amyloid in the cardiovascular lesion as well, okay? And just to end this slide, the injury that can occur on the endothelium, it can be caused by hypoperfusion and reperfusion. In other words, there is a sudden lack of oxygen, and then the oxygen starts again. And that is enough to cause an injury at the small blood vessel of the brain. So in this slide, I'm talking about an injury that only touches upon the blood side. Now, of course, the injury could just happen on the blood side, or it can damage across the blood-brain barrier, okay? And when you have a damage across the blood-brain barrier, then this is known as a microbleed, or as in the picture I showed you earlier on, is the micron-size microbleed. Now, I'm sure all of you have heard of the term blood-brain barrier leakage. Well, basically, this is it. This is actually an injury. Blood-brain barrier leakage is an injury. Sometimes you choose a slightly different way to describe it, but it dilutes the fact that it is an injury. And an injury obviously requires the body to activate a repair process, okay? When the leakage sounds harmless and people do not pay so much attention, but it is a micron-sized injury. And when there is a micron-site injury, here on the brain side, the prostaglandin release will lead to the degeneration of the parasite and the decoupling of the neurovascular unit. The prostaglandin also degenerate the oligodendrocyte, and the oligodendrocyte are the cells that supply the myelin to the neuron's axon. So at this stage, it looks as if the neuron is deprived of myelin product, and therefore it looks at this stage very similar to multiple sclerosis. And everybody who diagnosed the case of brain injury at this stage, we might not know whether it will go further into Alzheimer or it is actually stopping at the multiple sclerosis stage. Now let's move further. Is that once you have a bleeding in here, then there's C5a that come into the neurovascular unit, and with it you have all the cell, most importantly the neutrophil, the PMN cell, but also the monocyte from the blood that come in and mature into macrophages, and you also have the local resident macrophage such as the microglia. They too become activated and they move from TREM1 to TREM2, which means the activation of this cell, and they go about phagocytosing all the debris of pericyte and oligodendrocyte and so on. And of course, as I mentioned about the MMP9, this cell also release MMP9, and that in a large amount, as in a chronic inflammatory stage, it might help to destabilize the plaque because it actually degrades the amyloid protein. Okay, and as I say about the PMN cells, they are, as you know, a marker of acute inflammation, and they are back of oxidant, and they also release glutamate. So as a consequence of damage that cut through the blood-brain barrier, you can see an inflammatory response in the neurovascular unit. And the consequence of it is that there's a lot of cytokines and glutamate being released, okay? And in response to cytokine and glutamate, the astrocyte will put on a smooth muscle hat and it begins to produce material, okay? Material that goes to interact with the basement membrane proteoglycan system, and it tries to close the bleeding, okay? So this is a normal injury repair kind of responses. If you can get over the fact that the amyloid is not misfolded, but it is a peculiar contribution by the astrocyte due to the lack of smooth muscle cell in the small blood vessel of the brain, then it makes perfect sense. It was contributing material to stop the bleeding. Okay. Now, the interesting thing is that the astrocyte is a multifunctional cell. Apart from occasionally doing the job of a smooth muscle cell, it also generates lipid for the entire neurovascular family. And its also very important job is to protect the neuron from over glutamate exposure, because glutamate is a neurotransmitter, and overexposure of neuron to glutamate can lead to excitotoxicity. But if the astrocyte are doing a proper job, if they proliferate and they fix the wound, and at the same time they hoover up all the excess glutamate, then the neuron will never be drafted into this sorry affair, so to say. And the problem is, as in cardiovascular disease, is that in some cases, the astrocyte become compromised in its function, okay? The astrocyte can become compromised because of aging, for example. That is why I was very interested in the last talk about injury and so on. I was wondering whether you can actually look at the issue of cell senescence and the failure to repair as a way to forward as well. And here, as you know, Alzheimer affects old people more than anyone else. So what you have here is a case in which the astrocyte become compromised. It become compromised because of all the oxidants that are released when the PMN affected. And it's also compromised because with senescence, the astrocyte no longer proliferate as it should be. And the consequence of all of it is that the neuron become exposed to glutamate more than it should be. And when the neuron become exposed to glutamate, then what really happens is the neuron begin to make amyloid too. Okay, but to point out a point of detail, the astrocyte made amyloid. It is a type of amyloid 140, just like the platelet. And when the neuron begin to generate amyloid, it is the amyloid 142. Okay, so that is the point that we have to bear in mind. So when the astrocyte is no longer functioning properly, it fails to produce a kind of repair which stop the bleeding and reduce the inflammation and return everything back to normal. When that fails, then this plaque is known as an unstable plaque. Okay, when the plaque repair work wonderfully, then we have a stable plaque. When the plaque is not repaired properly, we have an unstable plaque. Okay, so we are now reaching an unstable plaque situation in which, because the repair is not done correctly, the inflammation will persist, and the astrocyte, unlike the smooth muscle cell, also had to protect the neuron, which it doesn't do properly. The neuron begin to enter the equation, and constant activation of the neuron eventually lead to apoptosis, inflammation, and neurodegeneration. So that is basically the story of the second half of Alzheimer. We begin with an injury, then I explain how it leads to neurodegeneration. And then in the second part of this talk, I will say what caused the injury. Okay, so now that we move into the degeneration of neuron, as I say, this microbleed, it all begin with this microbleed that cannot be repaired properly, and it leads to an overexposure of the neuron to glutamate when the astrocyte is no longer there to do a proper job to hoover up the excess glutamate. Then what really happened in the neuron is that it begins to erect the receptor, the NMDA receptor, and following that, you have the phosphorylation of the ARP and the production of amyloid 142 oligomer that then lead to a fiber formation. It's also worth pointing out is that because of the distance of the neuron from the site of the injury, so only a fraction of this material get to the microbleed. The other material, the amyloid materials, aggregate near to the neuron, and they form what is known as the neuritic plaque. But in fact, the materials that are generated by the neuron, some of it will go to contribute to the fixing of the microbleed, but other just gather together and become an aggregation of amyloid near to the neuron. This is known as a neuritic plaque. Now, as a tragic consequence of the activation of the NMDA receptor, calcium begin to flood into the cell. Okay, and this is a very important. Is that the glutamate is actually just a Trojan horse. The glutamate exposure activate the NMDA receptor, but it is the calcium influx that eventually drive the neuron into apoptosis or inflammation. Okay, now as the calcium begin to flood into the neuron much higher than control level, then the cell sense it as an injury, an injury to the plasma membrane. Okay, and like all injury, it leads to repair. So this is a very interesting story. Is that you start with what was the tissue injury and the tissue fibrosis, and eventually what you see in here is a fibrosis at the cellular level. You have a plasma injury, the cell sense it as a plasma injury, and it set itself to repair. And the way the neuron try to close this gap in the plasma membrane is by moving the cytoskeleton protein from the good part, in this case along the axon, try to move it to the synapses level where the calcium influx takes place. So it does that by disassembly one of the cytoskeleton protein known as the fiber actin cytoskeleton is dismantled and then it's shifted up to the synapses where it reassembles to close the leakage. Okay. Now, one of the very interesting things that happened is that this F-actin cytoskeleton, they are part of the cytoskeleton network, you understand. And the actin are actually tied up to the microtubule protein of the cytoskeleton. They're all tied together. And the tau, which you have heard in the neurofibrillary tangles that we talked about earlier, the tau protein are nothing more than little screws that tie the actin to the microtubule. So in order for the actin cytoskeleton to be disassembled successfully, the tau had to be removed. Okay, so this is done through the phosphorylation of tau. Okay, once the tau is phosphorylated, this actin cytoskeleton protein will be able to disassemble nicely and it shift up to the synaptic. Okay, now if that repair, that stop the calcium influx, then just like all the other cases, life get back to normal. However, this is a thankless task because no matter how you try to close the calcium influx channel, the glutamate exposure would then stimulate the neuron to open up again through the NDA receptor. So this now runs into a circle. It's just run into an infernal circle. And this phosphorylated tau, they begin to stick together into a neurofibrillary tangle. And there's been a discussion that maybe these neurofibrillary tangles, they are nothing more than just aggregation. The reason is why they need to be stuck together is because as in oligomer form, the tau can be toxic by accidental interaction with the organelle of the cell. So that's why they are better off being stuck together. Now, at an early stage, these residual protein are moved into the lysosome, okay, for degradation. However, as things become chronic, eventually the lysosome system will also become dysfunctional. And you have a mitochondria dysfunction due to overload of calcium influx, or you have lysosome dysfunction because there's too much protein that it can no longer degrade properly. And all of this lead to eventually to inflammation and neurodegeneration. And one last thing I want to say about what caused the neuron to undergo inflammation and neurodegeneration is that, as you can see, when you make the amyloid materials, the oligomer version of this amyloid can be taken up by the cell due to endocytosis, and they go into the lysosome as well. And of course, that is the reason why if you have too much oligomer, it might cause lysosomal dysfunction. This was the reason why people say that the amyloid oligomer can be toxic, but it's just a matter of dose, okay? And you can, in practice, the oligomer are rather transient because they are just a stepway before the formation of the fiber. But if you are unfortunate enough to be born with an APOE epsilon 4 allelic lipid, the APOE epsilon, they're not attached to the lipid particle, and they can exist independently, and they have very high affinity to interact with the oligomer. So if the APOE epsilon 4 interact with the oligomer, stop it from moving into a fiber formation stage. In that case, you have a lot of these oligomer APOE epsilon complex, and again, they would be endocytosed and lead to the lysosomal dysfunction. So you see, this is how it really happened. And in here, I just explained to you the second mystery after the misfolding the amyloid is why do we see the scaffold, the tau scaffold protein actually forming the neurofibrillary tangle. Now that actually would summarize the story from the injury to neurodegeneration. Now, I just want to point out is that, looking now on the first part of it, we ask ourselves, what actually caused this micron-sized injury? Well, there is ischemic injury. But when we talk about ischemic injury, you have to bear in mind that there's a whole range of severity, okay, of injury to the blood vessel. Very often when you talk about ischemic injury, you think of stroke, you think of major damage to the BBB, clot formation, vascular dementia, and that kind of thing. But of course, there are also microbleed, micron-sized microbleed. And these are actually silent. Probably you and me have it without actually thinking twice about it. Okay, and then below this micron-sized injury, you just have, slight irritation or complication of the endothelium and the blood and the basement membrane that can just lead to thickening of the basement membrane, for example. So in Alzheimer, we should be looking at this range of injuries, this small micron-sized damage. And this is when, we can, I believe this talk is relevant to our workshop in here, because we do need a kind of MRI machine that can pick up this micron-sized injury. And we need to have nanoparticles, nano biomaterial that we can deliver to where the plaques are. More of it later. So as I said quickly, you have all these ischemic injuries that vary by degrees of severity, okay? And, this injury is not restricted only to the cerebral vasculature. You can have injury to the cardiovasculature. This is very well known. It can injure the lining, it can injure the small vessel of your gut. This is intestinal ischemia, okay? And there, amazingly you see amyloid plaque in the guts. And of course, everybody knows it can cause macular degeneration. And there you can also see this amyloid plaque in the eyes, okay? And of course, it damaged the small vessel of your hearing system as well, and it can lead to deafness. Okay, so now I'm moving more or less towards the end of my talk in here, and we ask ourselves what causes this amyloid injury. Well, I work in traumatic brain injury, so hours after the initial trauma to the brain, you can see the damage to the central nervous system. and you can see the plaque there, okay? But there are other factors that contribute to it. In short, there are the factors that cause high blood pressure, such as a high salt diet, or for some reason the LPS has been released from your gut into the blood. They can damage the blood vessel. Air pollution is known to contribute to dementia, and air pollution actually is a form of respiratory disease. Also, respiratory disease like sleep apnea and so on, they do contribute to the damage to the blood vessel. Type 2 diabetes, well known to contribute to endothelium dysfunction, and so is the cardiovascular disease because it alters the blood, the circulation. And of importance, there's a gender issue in here, is that in women, when they reach their menopause, then there's a rise in the resin angiotensin that would then again lead to hypertension. So all this kind of thing can lead to hypertension. And this hypertension, they're not steady. They can cause the kind of hypoperfusion, reperfusion, and it leads to the ischemic injuries that you observe. The most interesting thing is this factor here, is the aging. As you grow old, okay, you have your autonomic system become imbalanced, and it leads to a sympathetic overdrive, okay, and a parasympathetic underdrive. And this sympathetic overdrive leads to hypertension, and the hypertension can lead to ischemic injury. More interestingly, is that as you grow old, as I say, the parasympathetic underdrive suppress your vagus nerve function, and that contributes to a phenomenon called inflammation. Okay, and inflammation means that the restoration of homeostasis and the reduction of inflammation become very, very difficult. And as I say, ischemic injury is lead to damage to the capillary and the plaque formation, and it can occur initially in the gut, okay, and then it move on to the eyes and the ears, and eventually it reach your brain. And this is the journey of neurodegeneration. It begins outside the brain, but eventually it gets there. And your body has given you plenty of warning. As you can see very early on, the elderly will be subjected to gastrointestinal disorder. And then, as you know very well, as you grow old, you begin to lose your sight and you become deaf. And most importantly, is that as it reaches your brain, it first affects the olfactory region. So you lose your sense of smell. And then it then went on into the entorhinal, the amygdala, and the hippocampal area. This is where cognition is. So you can see, and without actually telling the whole story in here, you notice that this function in here is gastrointestinal. Okay, it is something that, funny enough, you observe that in COVID-19 as well. So, okay, so I move very quickly now. So basically, what we need is we need tools. We need tools so that we can assess. So we need all the biomarker of the early condition, as I described, and then a biomarker of endothelial dysfunction and so on, and then into neuroinflammation and degeneration. And together with treatment, we need therapy. So the earliest thing is basically a program for vascular health, okay? And then this is something that maybe I think is very relevant to this workshop, is the use of nanoparticle to identify the plaque and to deliver a drug to stabilize it, because it is the unstable plaques that actually lead to neurodegeneration. This is, you can read this later on, but currently there's only 1, 2, 3, and perhaps 4 treatments for Alzheimer, and none of them are actually what I say very good. At the best, they can only delay the inevitable. And so the challenge for us here is to detect the disease early and act on it so you can actually reverse it. Because once it gets to the stage where the neuron become degenerate, then there's no reversal, because the neuron are end stage cells. So maybe we can talk further about whether there is a way to stimulate stem cell to generate neuron or whatever. But you have to reduce the inflammation for all this to work, and that is the most important thing. So in conclusion, I said that AD is actually a process that leads to neurodegeneration. And it is actually a brain injury, but it is a very low-level chronic ischemic brain injury, okay? And contrary to what people still believe, the amyloid is not misfolded. It's folded like a collagen for doing things very similar to collagen, okay? Then there are three types of amyloid plaque. You have the plaque that exists on the blood side only, the plaque that across the blood-brain barrier. There you have a subtype, the stable and unstable plaque. Okay, and what I show you is tau and F-actin tangles. They're nothing more than the consequence of the neural degeneration. And currently, there's a very poor treatment of Alzheimer, and so we need early diagnostic, and if we act quickly enough, if we act through public health and so on, we can identify this condition early enough. It is, and I do believe it is possible to prevent and reverse this disease. And with that, thank you everybody. Thank you for inviting me, and I will be taking questions.
Thank you very much, Professor Tran. Unfortunately, we're running a touch behind schedule, so we're only able to ask one question during this Q&A session. Our first question, well, first and only, is to Dr. Morota, which is, when is it important to use large animal models for regenerative medicinal studies?
Thank you, Adam. Large animal models are important in order to approximate as much as possible to the human situation. So we can see, for example, in the nTRACK project, we have a nice small animal model of muscle injuries, but the large animal model in sheep is even closer to the human situation, imitating better the human pathology. So the sheep model, the large animal model, imitate more and better than the small animal model, the muscle injuries in humans. So another option that there was not small animal models available. For example, in large bone defects in other project, in SBR project, we are doing femoral bone defects. So and it's not possible to do in small animal models. So we have to go to the large animal models to generate the defect and put the implant. So large animal models is always needed to get closer to the human situation.
That's great. Thank you very much.
Thank you very much.
And with that, that concludes our third segment today, and we can move into the fourth, which is cell therapy. Our speaker today is Dr. Ish Shalom, a project manager at Pluristem Therapeutics.
Hi everyone. Hopefully you can hear me very well. My name is Eliran Nishalom. I'm from the research department at Pluristem Israel, and my talk today will be about our placenta-derived cell therapy and how we take the story of our cells, how we characterize them and suit them to the right indication, clinical indication. Okay, so at Pluristem, and for those of you who know us very well, if there is such a person in the room, we are a cell-based technology company that's located in Israel. We are produce and expand placenta-based cells in our facility. Our platform are suitable for many opportunities. We develop a cost-effective platform that allows us to expand variety of cells, and we are more than happy to share the story of Pluristem with you today. The flow of the presentation will basically include the following. We will talk a little bit about the cell-based technology platform, but mainly I want to focus today about the cell mechanism of action and how all the data that integrate in a company take us to take the right decision when it comes to integrate and go to the clinical stage and development. So our cell-based technology platform in general is include a state-of-the-art 3D bioreactor cell expansion system. It's very cost-effective. It's patent in variety and very large amount of patent. It automized, it's scalable, and GMP certificate. We have a variety of tests that we are doing in-house in order to keep the batch-to-batch consistency and our comparability of batch-to-batch is very, very high. Our product is off the shelf, meaning that is while we decide to take it to the clinic, we just need to expand the cells and take them to the market. And as for the nTRACK process, in the last summer or so, we did one of our biggest shipment of clinical grade vial for you guys. So we were very happy to do that because we think that the nTRACK goal is very important, and you will understand why. So when it comes for the mechanism of action of our cells, I want to talk to you a little bit about the source of the cells. So we are basically take the placenta from electivity surgical, Caesarean surgical, sorry. Right now we are having two off-the-shelf product, the PLX pad and the PLX R19. Both of them are from the same origin of tissue, but they are expanded in different culture medium, what gave them different properties. The R18 is a product that I will not talk about today so much, is pro-hematopoiesis indication. A clinical indication, and I want to focus about the PLX pad today because this is a cell that we sent to you guys for the nTRACK consortium, and this product represents regenerative effect, is reduce inflammation, stimulate growth blood vessels, and stimulate repair of tissue damage. When it comes for characterization of our cells as part of the mechanism of action, we already show that these placenta cells, as we call MECs, like properties, and you will understand in a minute why we call them this way. This is adherent cells that express all the known MECs marker like CD105, CD73, and so on, and they are not show in vitro differentiation capacity. That's why we call them MECs like. And they also show very less and very pure engraftment properties, as you can see in this figure. And biodistribution for both our product show a very limited engraftment of the cells. You can see that up to a very short time after the injection, we almost cannot see the cell in the animal after injection. So as far as from cell distribution and cell imaging, in vivo imaging, we are the best we were able to show until now. It's when we inject them IM or IV, we all see that we see almost a total or complete elimination of our cells in the mice. And as far as we know, there could be some infiltration of immune system, immune cell into the injection site that eliminate the cells. Now, we think this, we think so because when we are take biopsies from the injection site, we see that there is some major infiltration of immune cells to the site of injection. But this is as far as we can do as far as for cell imaging and cell monitoring. So this is why we think that the nTRACK conservation goal is very important, and it will be crucial to us to understand better what happened with our cells after we inject them to the patient and to the animal that we are using for our preclinical experiment. So if we continue to talk about the mechanism of action of our cells, usually under ischemia condition, but not just ischemia, we are usually dealing with inflammation and loss of muscle tissue volume. And when we inject our cells into the body, our cells are stimulated and react to the surrounding environment. Our cells are secreted variety of soluble factor that could be pro-angiogenesis like VEGF. They secrete also TNF and other modulation factors that could be contribute to their activity, and also other factors that could be supporting regenerative effect. So all these secretion factor all together, and not just one of them, are crucial to their activity during the treatment after injection. And today I want to focus mainly on their immunomodulation capacity in order to make you understand better of cell, because I think this topic is less discussed in all the previous presentation today. So when we are talking about immunomodulation of our PLX pad cells, we're talking about few major properties. Our cells are contain and secrete all kind of cytokine or other factors from their surrounding environment, like pro or anti-inflammatory cytokine. They secrete EVs, extracellular vesicles, and also known to induce and activate the IDO pathway that's responsible for the tryptophan metabolism. All these three very important pathway is influence of variety of immune cells and causing to some kind of dysfunction, what eventually caused to a major immunomodulation phenotype. So I want to take you for a very short journey in the next slide, and to show you some research data that were generated in our company. As far as for the pro-inflammatory cytokine, when we take our cells and co-culture them with PBMCs, after activation with CD3 antibody, we see that there is a significant decrease of pro-inflammatory cytokine like TNF-alpha, interferon gamma, IL-10A, and IL-17, and more cytokine that extremely important to elevate the inflammatory response. So our PLX pad inhibit this phenotype, and this is one of the main mechanisms that cause to decrease of the inflammatory response. When we looked about anti-inflammatory response, we see the opposite pattern. We see that cytokine like IL-5, IL-10, PD-L1, and other cytokine are elevated. So to continue with our previous slide, we see that the elevation of anti-inflammatory cytokine and decrease of pro-inflammatory cytokine are ending at the same results. As far as for the EVs, for the extracellular vesicles, we are and our collaborative show that when we are take the same PBMCs and activate them, this time with PHA, we see that our EVs inhibit stimulation of T cells, as far as very similar to the result that we find when we co-culture our PLX with the same cells. The result of this phenotype is probably because, as you can see in the left image, in the right image, sorry, because T cells and monocyte cells are able to uptake the extracellular vesicles and localize them to the cells. Another very interesting pathway that is very important to our cells is IDO pathway. As you can see, when we are stimulate our cells with the interferon gamma, which is used as an inducible factor for the IDO pathway, we see a major production of the keratin metabolite that create after tryptophan metabolism. And this production is almost totally stopped after we do a IDO knockout with CRISPR technology. So this is just to show you the CRISPR technology and our capabilities in the research department and how we take advantage of the technology in better understanding our cell activities. In the right panel, you can see that IDO knockout is restore the inhibition of the, or stop the inhibition of T cell proliferation, as you can see in the right bar. What happens when we look on the total PBMCs cells? So when it comes for PBMCs that contain NK T cells, monocyte and T cells, we can see that when we co-culture them with the PLX, we see inhibition in the T cell proliferation. You can see also that elevation in the secretion of IL-10 that considered to be immunomodulator factor. Another very important modulator of the immune response is the Treg. You see that when we co-culture our PLX with PBMCs or activate PBMCs, we see that elevation in this subpopulation of T cells. When we again knock out different or specific gene in this regard, we see that knockout, for example, for interferon gamma receptor enhance expansion of Treg, and the opposite phenotype when we knock out the TNF alpha, TNF beta, which is not surprising because TNF beta is one of the major factors that cause to elevation of Treg. So this is another paper that is very interesting. Our cells are also increase the expression of M2 macrophage like CD209 and decrease other marker of M1 macrophage, which is considered to be more pro-inflammatory. As far as from the macrophage, the same in phenotype is present here. CCR7 is decreased, and other marker is increased. Both of HLA, ABC, and DR is decreased. What make I think that polarized macrophage is blocked by the PLX pad product. As far as for the dendritic cells, the same phenotype is repeated here. All the above marker that you show in the slide is decrease, but causes think that PLX pad is reduced maturation of dendritic cells. As far as from our second product, which I will not focus today, is the R19. I just in very short explanation, this is a product that we are established for another clinical indication. We are take this product for hematopoietic clinical indication, like under genetic or DNA disorder or acute radiation syndrome. We see some bone marrow failure, major bone marrow failure, that cause to a low red blood cells count in all the three mentioned cells subpopulation. And when we eject the cells again stimulated by the surrounding environment, and they are produce several soluble factors like cytokine growth factor that cause all the cell population to recover and yeah. So taking all this result together, how we can take this from the research batch to the clinical stage, and how we can take advantage of the knowledge that we have on these two products and take it to the clinic. So if we go back for a second for ourselves, for the placenta, we have a full chain supply that allow us to expand the cell from the placenta up to the injection site in the clinic. We are usually inject the cell intramuscular, and this is our clinical ongoing trial that we have currently. We have several clinical trials for each product. For the PLX pad, we have 3 ongoing clinical trials. One trial is for muscular regeneration following hip fraction, acute respiratory syndrome associated with the COVID-19, which is very related to the immunomodulation mechanism of action that I just showed you, and also a chronic graft versus host disease. As far as from the PLX R19, we have 2 ongoing clinical trials that focus on hematological deficiencies like acute radiation syndrome and hematopoietic recovery following hematopoietic cell transplantation. So I will now show you some of the results of the ongoing clinical trial. As far as from the hip fraction surgery, this is a major clinical indications that many people suffer from. 6 million total number of hip fraction worldwide. And the PLX is generated to stimulate this clinical indication, and not to stimulate the clinical indication, sorry, but to stimulate the muscular regeneration in order to help this patient that suffer from this clinical indication. Well, in the previous clinical trial that was test and enroll injury following total hip replacement, we enrolled 20 different 20 subjects. The PLX demonstrated a significant increase in muscle volume and strength compared to the placebo. The first study, this is the first study that actually show efficacy of cell therapy in this skeletal muscle injury. And as you can see in the panel in this slide, and now we are still under the ongoing phase 3 study of PLX-PAD. And we expect that in the upcoming months or so, we are able to open the file of the subject and to see the intermediate result. We are very excited about it, and we see what will happen there. This IPGEN study is under, of course, the Horizon 2020 program. As far as the ARDS associated with the COVID-19 program, this clinical trial were stopped at the last December. We noticed efficacy trend from the study for a patient treated with 300 million of PLX cells. In the United States and in the European study, we saw increased survival rate by 40% and a little bit less in the European study, and also saw very significant increase in the ventilation-free days of some of the patients that were enrolled in this clinical trial. Another interesting finding that we noticed is blood CRP decreasement in this patient that were suffering from this infection, and this is again show our immunomodulation mode of action of our cells, as I showed you before. As far as for our second product for the PLS-R19, ARS was a clinical trial that already ended, actually. It was under the animal rule of the FDA. We saw that after treatment of our R18 product, we saw a major survival rate compared to the placebo, as you can see in the slide, and we see a significant recovery of this blood lineage for the platelets, red blood cells, and the white blood cells. So this product is very promising, and we are this year this product is taking our most attention. And we are pushing this product very hard these days, and hopefully we will get some new results. Another very interesting clinical trial that is still ongoing is following hematopoietic cell transplantation. We take the R19 and inject them to people who suffer from bone marrow failure after transplantation in order to restore the blood cell population in all the mentioned lineage, white, red, and platelets. And as you can see in this slide, we see some very significant and nice results that show a significantly improvement from baseline count that were observed in all the cohort of the hemoglobin and platelet count. In the 6-month following up, 10 patients show a response in all 3 blood cells type, and 46% of the patients become transfusion independent, which is very, very powerful result and very promise. The dash line in this graph show the baseline threshold of the baseline at the beginning of the clinical trial. What makes, just to show you that most of the people, or some of them at least, will show very low platelet account, and as compared to the 6-month period after the injection of the product. So this trend of result, very promises one, is very encourage us. And our next generation product is one of will contain the CRISPR technology. We want to take advantage of this product and this technology to expand our capabilities and to make our cell more suitable for other clinical indication and make our cells more effective to be secrete more regenerative soluble factors that will help us to get a better results. So this is basically what Pluralstem done from the research bench up to the clinical trial that is still ongoing. And I want to thank you all for the listening and for your attention, and I open to questions. Thank you all.
Thank you very much, Dr. Shalom. It seems like an exciting time to be at Pluralstem. We have enough time for one question from this segment, which is what other applications can be utilized for the Pluralstem allogenic stem cell production platform?
What kind of application? Well, it depends what kind of application you mean. I mean, if you mean or referring to clinical application, as far as beyond the cells, it seems that this or our cells, the PLX cells, are suitable for a variety of cells, and any generative clinical indication could be suitable for the cells. If you're talking about other application, like how we can utilize our platform, so this is another question, like our expansion platform could be suitable for a variety of cell type, up to from stem cell variety, up from the stem cells cell type, and up to the alternative meat product. Actually, we signed with one of the biggest food company in Israel in order to take advantage of our 3D platform to expand a given or a specific cell type of cells in order to take the adventure of this platform and to take it to the food industry. So there is all kind of applications that's suitable for our platform. Yeah.
That's brilliant. Thank you very much. We can now move into our fifth and final segment today, which is the regulatory framework. Our first speaker is Marta Reo, Scientific and Regulatory Affair Director at Asphelion, who has over 7 years in the pharmaceutical drug development industry and also over 7 years in the regulatory affairs segment. Thank you.
Hello, good afternoon, everyone. This is Marta Rayo speaking from Asphalion, a scientific and regulatory affairs consultancy firm. Today, I'm talking in representation of the nTRACK consortium, and I will present a case study for the nTRACK project. I will try to be brief and try to recuperate a little bit of time because we are a little bit behind of schedule, because Mark this morning and Mario this afternoon already explained the introduction about the project, etc. And I can go directly focus to the regulatory parts and lessons learned. We will start explaining why regulatory guidance in the nTRACK project is required, and talk about the classification of the product, and finally focus on lessons learned. When I will be talking about the nTRACK and all the lessons learned from our regulatory process and the 4 years of the project execution, I would like to remind you that these lessons learned can be extrapolated to other tracking agents. Here, the brief summary of the project. So basically, we are utilizing nanoparticles for stem cell labeling. So this is the general concept. Why is regulatory guidance required and interesting even at an early stage? The main reason is especially because we are at an early stage of development. In our case, there are multiple potentially relevant regulations that might apply for tracking agents. And especially if we consider that we are dealing with nanoparticles and nanoimaging agents, this perhaps may complicate a little bit more the classification. We could eventually fit under the definition of medical devices. We could eventually fit under the definition of medicinal products. And when combined with advanced therapies, also under the definition of advanced medicinal therapy products and other type of products. And the fact is that these regulations may have different information requirements and may involve different regulatory authorities. Considering regulatory issues early in development will help to focus the research by defining the primary purpose and to avoid repeating studies for regulatory information requirements. Therefore, which seems obvious for the benefit of the project, it was really a challenging task. And in this particular case, we will be briefly discuss a little bit more about its classification. In the nTRACK project, we conducted several regulatory activities which taught us important lessons. At the very beginning, we prepared a regulatory roadmap as a major challenge, having the need to define the classification. Afterwards, we gained some knowledge and we gained information and advice from several authorities. And we will explain more in detail later the information obtained from them. And lastly, we celebrated a regulatory workshop with experts and different stakeholders to gain knowledge on the needs and difficulties we faced. Without going into detail in this part about how to classify the nanoimaging agent, the fact is that this was a fundamental challenge. Main possibilities that we were discussing within the consortium at the beginning. We were discussing the classification as a medicinal product, medical device, and maybe others. And the conclusion reached by the consortium was relevant because depending on the classification, different regulatory pathways may apply. And consequently, different requirements may apply as well. And the classification depends on the precedents. As Mark was mentioning this morning, there were limited precedents and there are not specific guidelines available. The classification also depends on the primary function of the product, understood as the intention of use. It is not the same to diagnose something or to monitor a disease. We were discussing which would be the clinical use, etc. And lastly, we were discussing if any of the nTRACK components achieve its principal intended action, by pharmacological, metabolic, or immunological action. As there were no similar products marketed and no clear guidelines for this type of products exist, we started the following our regulatory activities within the project, trying to gain knowledge on the pathway and to define the requirements with a special interest in preclinical needs to support a future clinical study. So independently of the classification, one of our main goals was to define the preclinical and quality requirements that we would need to fulfill in order to move forward in future with a clinical trial. After long discussions, we agreed that our product, and this perhaps can be also extrapolated to other nanotracking agents, neither fit well within the definitions of a medical device or a medicinal product. And as a standalone product, I mean, without the combination of cells, the definition of medical device seems more suitable than a pharmaceutical medicinal product, but alternative definitions or uses or classifications may also be considered, like, for example, the definition of excipients. And I will talk a little bit more later about this. Feedback gained from different authorities really confirm what we thought, at least that the nanoimaging agent, for the purpose of monitoring biodistribution of cells during development, does not fit well within the use definition of either medical devices or medicinal products. So the classification depends more on the company's claim of intended function and indication. And we were advised to choose ourselves. So any manufacturer should choose the preferred regulatory classification and justify it. And then according to this, generate the necessary data. But independently of the classification, something that was constantly mentioned by all the authorities we reached is that the product should be safe and reliable. What about the lessons learned? Let's start with the method that we follow to draw lessons. As mentioned, we gained experience and we collected experience from the consortium and from the development process. We get the feedback from different authorities. In particular, we conducted an ITF meeting with EMA at the very beginning, and then a scientific advice with a national competent authority, and some informal conversations with FDA. And finally, we celebrated a regulatory workshop with experts' participation. On the premise that regulatory guidance or standards protocols were mostly not available, information from consortium partners and from the development process was very relevant to us, and especially to define the intended purpose of the nanoparticles and the needs in different areas of development. For example, for quality control of the nanoimaging agent, in this case, our nTRACK protocol, our nTRACK product, protocols would need to be developed for production and characterization, especially for the upscale of the nanoparticles, labeling protocols with nanoparticles for each type of cells, and quality criteria for the therapeutic action of the cells, again, per cell type. More specifically, at the beginning of the project, we started our regulatory conversations with the regulators, with an ITF meeting with EMA. The Innovative Task Force meeting is a free and preliminary consultation with the EMA that provides a forum for early dialogue between developers and regulators. And the feedback given by the regulators is the opinion of the participants, and it should not be understood as a official opinion. So the feedback gained through this meeting would not be interpreted as a regulatory guidance. The general feedback we received from them was a clear encouragement for development, and as there is a need for this kind of tools of therapy for cell therapy development. And additionally, the ITF participants from EMA recommended us to consult a national competent authority, especially focusing on the data required to move forward with clinical. So in fact, this is what we had already from our regulatory roadmap. To perform this ITF meeting, we compile a briefing dossier. You have here a short summary of the type of information we put in this briefing document, and we prepare a presentation to be shared with the EMA participants. And as already anticipated, the feedback from the ITF reinforced our plans to seek a formal scientific advice with a national competent authority. So we proceed with the selection of an authority at a national level with relevant experience in clinical trials using therapeutic cells. In this case, answers from the national competent authorities were based on the questions we posed to them and based on the information we provided in a briefing document again. The main objectives for us in this scientific advice was to get feedback in quality requirements, especially in specifications, stability plan, manufacturing, etc., and standards to be followed. The second objective was to get feedback in the preclinical development conducted so far and in the plan of the upcoming studies. And with the final idea to get validated a clinical trial design proposed by us, of course, supported by the previous data from quality and non-clinical point of view. The lessons learned from these conversations with a national competent authority. In terms of quality, they, of course, we would need for a future clinical trial application to propose specifications, a manufacturing method, etc. But they were putting some emphasis in the characterization of nanoparticles, especially in the size distribution and aggregation. Moreover, it should be demonstrated that the labeling with our nanoparticles doesn't alter the function and safety of the therapeutic cells, and then they advise to conduct strict comparability studies of the non-labeled cells with the labeled cells. On the preclinical studies side, the following recommendations were given to us. They recommended to characterize the optimal ratio with particle cells. This should be determined to correlate the number of cells to the effectiveness in the non-clinical models. Data is also necessary on how to differentiate and distinguish in vivo between viable and non-viable cells in the animal model again. Biodistribution and toxicity for both our imaging agent, I mean our nanoparticles, and the labeled cells. But this would depend on the specific data showing if the nanoparticles are affecting or not the cell's behavior. So the type of data that would need to be generated here could be generally understood as a complete package of data, but in case the nanoparticles would not be affecting or impacting the cell's behavior, this data perhaps could be more limited and more scarce. It was also asked for how long and where the nanoparticles remain in the body, and this should be safe. Nanoparticles accumulation in specific organs should be also addressed, and concerns about the interference of nanoparticles with future imaging techniques or procedures that patients might require was also raised. It was recommended to additionally conduct an in vivo study comparing our nTRACK nanoparticles with label cells against non-label cells. And as we were using mathematical and computational models that were also explained this morning by our colleagues, they recommended and they insisted in the need of validate these models. In terms of clinical data, apart from discussing issues about the potential design and so on, the impact of the imagination on the benefit-risk ratio for the patient was something that really was under the interest of the agency. After that, the last piece of the lessons learned was obtained from the regulatory workshop that our colleagues from the IRBM prepared and conducted. This workshop was an online workshop because it was conducted in the last years. And it consisted in 2 sessions on 2 consecutive days with 2 main objectives. The first one was to disseminate the nTRACK project, and the second one to obtain expert input on cell labeling. Participants invited were experts from regulatory bodies, academics, experts from, well, people from companies developing cell therapeutic products, and people from companies developing nanomaterial labels, and clinicians with interest or applying labeled cell therapeutic products. The main conclusions from this workshop were that the main reason for labeling therapeutic cells is to gain insight into biodistribution and clearance. So this is aligned with the discussion that we were having today, because this gain information will definitely optimize development and improve treatment options. And as a kind of consensus, the different participants agreed that an ideal label system should be inner, compatible with any type of cell therapy, and should facilitate a number of imaging techniques, and of course, being clear fast from the body. So this was the wish list for an ideal label system. And regarding the regulatory classification of a nanomaterial label, the participants agreed that is needed early in the development process for the reasons that we already commented, because it would be more relevant to define from the beginning the standards that may apply, the guidelines that may apply, etc., in order to not repeat studies and so on. However, the different participants agreed that up to now there is no single anonymous unambiguous opinion on the classification of the nTRACK labels. And an alternative option from the experience of one of the participants in the US suggested to consider the label as a novel excipient. So this label, these nanoparticles, could be part of an investigational medicinal product consisting in a cell therapy product. With regard to the preclinical safety data, retention or expression of the label by cells was considered important. So this is aligned with the feedback from the regulatory authorities. In terms of quality, the combination with label cells was necessary to characterize concentration of the label, cell viability, and cell function. So this is again harmonized with the feedbacks that we received from the regulators. So the different participants agree on the need to characterize the quality criteria after cells have been labeled. And finally, the timeline for critical steps in regulatory process was interactively created by the participants in a mural. So very brief, in one of the parallel sessions, the participants designed a timeline for the different activities to develop a new label system, not a cell therapy product, to develop a new label system. It was basically following the scheme of a new drug development in terms of phases, but with a special focus on the specific needs for the development on the label. For example, in the material selection and the prototype selection, what it is important is to ensure the feasibility of the label manufacturer, also with the feasibility of ensuring a proper cell uptake. And you have also some more details on the different steps and the different stages that were defined by participants. But it's pretty parallel to a classical development of a medicinal product. And as the workshop driver was regulatory, the group finalized the session identifying the main regulatory actions that would be or would result in the vesting. And the main actions were starting with the determination of classification, and based on that, identify the standards and guidelines applicable, and then approaching regulatory bodies. In this case, we were talking about a scientific advice because we were focused in the European scenario. And finally, the main goal of this advice should be to define a proper preclinical development. So this is more or less what we did in our nTRACK project during these 4 years of Horizon project. Even these actions were discussed at an European level because the main focus of the workshop was development in Europe. The steps could be extrapolated to other territories, but of course, checking always the standards and the regulatory bodies to contact. And so, as general concluding remarks, these 2 last slides is what we learned during this 4 years project, and these conclusions are totally extrapolable to other label systems and nanoparticle label systems. There is a high uncertainty about the legal status of nanomaterial labels for cell tracking. The navigation between the tangled requirements of different regulations makes the development more difficult and challenging. A straightforward roadmap for gathering regulatory required information on nanomaterial labels for cell tracking could save resources. Different objectives of cell tracking in vivo may represent different needs, and then preclinical development should be based on a case-by-case basis and adapted to the specific objectives. But independently of the classification of the nanomaterial labels, the product has to show that it's safe and reliable. And finally, comparison of labeled cells against non-labeled cells should be done both in vitro and in vivo in relevant animal models of the targeted disease. So what Mario was mentioning about the large animal models and how this model would represent the human disease and so on is relevant to move forward with the comparison of non-labeled and labeled cells. The importance of in vivo differentiation between viable and non-viable labeled cells in the animal model is key. Also, it is important to be able to correlate the number of cells to the effectiveness of the product in animal models. And biodistribution and toxicity for both labels, the label itself, the nanoparticles, and the labeled cells, depending on the specific characteristics of each of them, it's also relevant. And in summary, as there is no sufficient guidance for nanomaterial labels to be tested clinically, the developer should justify how to test the safety of the label and how the benefit-risk ratio for humans is fully supported. Thank you.
Thank you so much, Marta. I really hope that other projects will benefit from some of the works over the past 4 years regarding our lessons learnt. Maybe they can avoid some of the icebergs, so to speak. We're going to briefly return to an earlier segment, back to Horizon 2020. Our next speaker, Dr. Andres Diñez, is the founder and director of Biotalentum. He has expertise in stem cell technology, cell differentiation, and genetic engineering, to name but a few. And today he'll be giving us an overview of the Horizon 2020 project, iNanoBit.
Hello, everybody. I hope you can see me and hear me well. And thank you for your attention for the last talk of the day. And I will talk about the iNanoBit project, which is actually the contemporary of nTRACK, because in the same call, we received also the EU funding. Well, we focus on diabetes because diabetes is an extremely important disease. It's more than 400 million people around the world and lots of side effects, unfortunately, with diabetes. So although we can treat the insulin levels with insulin injections, but it's not really preventing the kidney damage, the macular degeneration, the capillary problems, neuropathies. So many people die from the side effects. And on type 1 diabetes, where it's early on, probably the immune system in most cases destroy the beta cells, there is a possibility to replace the beta cells. So currently, how to treat diabetes, we can use insulin injection, but as I mentioned, it's not the full picture. And many people would need pancreas transplantation. But pancreas or pancreatic islet transplantation is very limited because there is a limited number of donors. So that's why another option came up, which would be the xenotransplantation, when for humanized pigs, pancreatic islets could be transplanted into humans, or making the beta cell in an artificial manner. So in order to achieve all these, we need probably a bioartificial pancreas, BAP, which would kind of protect the transplanted cells, because if they are immunologically foreign for the body, which is the case, of course, in most human-to-human transplantation as well, there is a need for immune suppression. So immune suppression in long term is not ideal because it creates not only a lifelong dependency for treatments, but also susceptibility to diseases or even cancer or viral diseases. COVID and others can be very dangerous for people with organ transplantation. The other approach, if we protect them from the immune system, in this case with a semi-permeable membrane, which allows small molecules to move across, for example, insulin or nutrients, oxygen, glucose, they can move through the semi-permeable membrane pores. But the larger molecules like immune molecules or the cells, immune cells, are prevented to enter. So in that case, actually, we can even use kind of normal pig cells, not only from the fully immunized human immune system tolerant pigs. So this is a very interesting way for regenerative therapy, and the iNanoBit project focused on integration of nano and biotechnology for islet transplantation. We have quite a few partners from industry, both for making these, Defimed is making, for example, from France, these bioartificial pancreas medical devices, and Biotalentum making beta cells, but also we have LMU in Munich making xeno islets. And we have other universities making nanomolecules and also BBS nanotech in Hungary. And we have imaging companies, Mediso and iTerra, making the imaging possible. UCL is also working on the transplantation into pigs. So this is how it looks like. So we have two sources. We have the human-induced pluripotent stem cell-derived beta cells, and we have the pig xenoislets in the device, in the French device, and then it goes into a humanized pig, type 1 diabetes pig with transgenic modifications and cloning. There is a new breed of pigs for testing the devices. It's also at LMU in Germany. And then there are two kinds of monitoring devices. One is a SPECT-CT, but it's not a normal SPECT-CT. It's a very high resolution for identifying very small group of cells or even single cells in pig. So that was possible before only in rodents, in small animals, but now it's in large animals. And the other system is optoacoustic imaging, which is a very interesting non-invasive device. I will show you more pictures later on. So it was a very complex issue how to achieve all the components. One was to create these porcine neonatal islet-like cell clusters in a transgenic pig, which has beneficial immune and anti-immune characteristics. So it's better protected from the immune system. And indeed, the German team worked on that very successfully, but also on top of that, there was an interest in creating insulin promoter fluorescent reporter line for in vitro imaging. And we developed that further to create pigs with infrared fluorescent protein, because the iTerra device I will talk about needs the infrared spectra for the non-invasive detection of cells and objects. And basically, that's a totally new pig line, transgenic pig line generated in Munich for this project, which carries the infrared fluorescent protein. And we did something similar in my company, Biotalentum Limited in Hungary. We also created induced pluripotent stem cell line with the infrared fluorescent protein. And that took some time, but also we've shown that from these iPS cells, we can successfully make these are 3D spheroids pancreatic progenitor cells. And from these, this is the characterization of the spheroids. We can see not only the characteristics step by step coming up for the pancreatic progenitor markers, but also we can see that the insulin content, insulin secretion is pretty good. So encouraged by these in vitro results, we did the transplantation into small animal studies, into mice, under the kidney capsule. In that case, there was kind of immune protected mouse strain. Basically, in these cases, we made the kidney capsule. It means that we don't need this device combination because we create the immune suppression. The good news was that after 13 months following in the mice, we terminated the experiments in different points with different mice. We've seen very nicely how the human C-peptide was secreted by the cells and also how they lowered the glucose levels. They were not diabetic mice, so that was not a question of life or death, but we've seen this additional human C-peptide and probably insulin was also affecting the mice glucose levels, and also it was a very prolonged procedure. When the mice got very old, then we had to sacrifice the last one as well. But before sacrificing, we used this infrared fluorescent protein detection, and with an appropriate device in these kid mice, we were able, the so-called IVIS infrared system we've seen in the live mice. This is anesthetized live mice. In the kidney capsule, it was so obvious that the cells are still there and alive. And when we explanted the kidneys, we've seen basically insulin production by histochemistry, and also we've seen glucagon producing cells. So these structures, they matured in the mice of the transplantation because they were pancreatic progenitor cells. They matured into full pancreas, active pancreatic beta cells. So that's very, very encouraging for further studies in large animals. So as I mentioned, Defimed, a French company, created this mail-pump device, and this device was tested into different sites in pig, in large animal models, because using these devices, it's very important how well they get vascularized, and also to avoid a kind of fibrotic capsule around the device. So the device has to interact very positively with the host body, having the vascularization and not creating a kind of isolation fibrotic reaction. And the trick actually is that the device is transplanted, in that case along the abdominal muscles, because that's a very well vascularized area in the pig, and also probably in the human in the future. So it's between the muscle layers, and there is a pre-vascularization period, 6 to 8 weeks, because the problem with the beta cells, they are extremely sensitive on oxygen levels. So if we don't care enough on the oxygen levels, then they might die prematurely. So once the device is vascularized, then there are these ports where we can put the beta cells in. So these ports are basically under the skin. So they are sterile, kept sterile, but then they can be used to load the device with the beta cells. And indeed, this way, it's possible to create a functioning artificial pancreas. And it was tested with pig pancreatic islets from the donor pigs, and it looks very promising. And of course, the whole project was also about imaging and following the transplanted cells or pancreatic islets from pig. And that was also with lots of progress, very positive progress. So at Milano Bicocca University and at BBS Nanotechnology in Hungary, these multifunctionalized nanoparticles for imaging are created. So they have fluorescent molecules, but also they have radio labeling. So this is a system which allows multifunctional imaging after transplantation. And I mentioned this optoacoustic imaging machine. The German company iTera is producing it in Munich, and it's a testing run under the skin of the transgenic pigs. And basically, the device works on a way that it's an infrared laser, which is penetrating pretty well the tissues to a few centimeters depth. And with the infrared laser, if it hits cells which can be excited by the infrared spectrum, for example, it can be in some cases in some cancer cells, but in our case, the infrared labeled stem cells, or in the pig, we did also with some fibroblast cells obtained from the founder pigs. So in that case, the excitation and the laser energy is absorbed. It creates a little change in the volume, and that change in the volume can be detected by a ultrasound device. So it's an extremely sensitive but totally non-invasive method. So this method worked pretty well in the pig skin studies, and also after transplanting these fibroblast cells into the medical device from Defimed, it also worked, but also it triggered some design changes in the device to avoid any artifacts from the design of the original design. So this is a learning curve, but it's progressed extremely well in the project. And nevertheless, Mediso invested lots of energy and time and also resources to create a new multi-pinhole collimator, which allows really focusing on the pig liver, because in pig, similar to human, beta cell transplantation might be done also through the portal vein into the liver. So this is the schematic image that how this specifically designed system can image the pig liver, and this equipment is being installed right now in Munich. So this is a very successful step. It was a little bit delayed, unfortunately, due to the COVID restrictions and changes, but this works really nice. So I think overall the project progressed extremely well. So in conclusions, we managed to have both rodent and large animal transplantation trials, and we have all the elements getting together. We still have about a year to progress with the project. We had some extension due to COVID. Hopefully, during that time, we can really get to the bottom of all the still pending questions, and especially the large animal studies. And in animal transplantation, we've shown that we can restore the glucose insulin control in mice. And also we are moving now to the large animal studies. And still there are challenges, because for the large animal studies, we have to do major scaling up and scaling out for the human beta cell production. And this is, of course, always a big jump. And this vascularization of the medical device is a key issue. There are some competing products, and that's where they had real trouble and difficulties. So this is what we hope that with the Defimed device we can overcome. And on the level of the infrared fluorescent reporter expression, we have very good results. And this is also something important message for the other projects and the other teams working on detecting transplanted cells, maybe not only beta cells, but mesenchymal stromal cells or other differentiated cell types, because this is a very interesting opportunity. And if somebody is interested, we are very happy to share our experience. We have an IPS line, so we can make many cell types from that. So thank you very much for your attention. These are the teams and some other contributors helping us in imaging and other experiments in progressing with this complex task. And this is the team at the kickoff time in Brussels, but we didn't age much over the few years, and I hope that we can really deliver something as expected with this project. Thank you very much for your attention.
Thank you very much, Dr. Dinez. It's always good to see success in our sister projects. Our final presentation has two different speakers. The first is Dr. Martha Lundberg, the Program Director at National Heart, Lung and Blood Institute. And the second is Dr. Mia Thu, co-founder and CEO at Cell Medical, where they'll be giving us insight into nTRACK's committee perspectives.
Good afternoon. Can you hear me? Hello? Oh, right, great. So I'm Martha Lundberg, and my colleague, Dr. Mia Thu in California, we're giving this joint presentation on the nTRACK's committee perspectives on imaging for cell-based therapies and the benefits, gaps, and challenges. Next slide. Next. So I'm a full-time employee at the National Institutes of Health, notably the National Heart, Lung, and Blood Institute. And this is talk is prepared as part of my official federal duties. And this is the disclosure for Dr. Thu. She's co-founder and stockholder of Cell Medical, and as full-time CEO of this company. And the talk is prepared as an industry member of nTRACK. Next slide. So today I'm going to talk about the use of nanotherapeutic and nanobiologics to treat cardiovascular disease. This type of therapeutic will benefit from advancements in high throughput technologies, both in vitro and in vivo, as well as machine learning based data analysis. The insight obtained in this manner can inspire screening other formulations or provide promising candidates for large animal studies. Techniques to scale up nanotherapeutics, formulation, and non-invasively image their in vivo behavior will further facilitate clinical translation. Next slide. So employing trained immunity targeting nanobiologics to treat cardiovascular disease, trained immunity and the innate immune system in general plays an important role in many aspects of cardiovascular disease progression. Specifically, I have a little bit of a background noise here. Specifically, CVD and the associated associated molecular patterns, such as oxidized LDL, induce trained immunity and thereby result in sustained inflammation. Nanotherapeutics facilitate targeting the underlying cell pathways in myeloid cells and their progenitors. NIH investigators have discussed formulation and labeling approaches to facilitate the monitoring of these nanotherapeutics in vivo, and they've used multimodal imaging and flow cytometry. Importantly, they detail how drug-loaded nanobiologics can be used to target specific metabolic programs. Next slide. Next. So there's really no better way to demonstrate the promise and success of CAR and T cell therapies than looking at the patients, such as Emily here on the left side. Emily was diagnosed with leukemia when she was 5, and her chances of survival was less than 20%. Currently, Emily is well, and we show that some of the challenges in the field are working. But it's also important to take into account for marketing the cell therapy, the cell biodistribution, the engraftment, and the toxicity and safety. Next, I would like to transition into a description about HESI, which is the Health and Environmental Science Institute. Next slide. So HESI is an independent scientific nonprofit organization based in Washington, D.C., United States. It addresses risk assessment and safety challenges in the field. It's a neutral platform, which is transparent, independent. It operates internationally, and they have more than 15 different scientific committees and programs. HESI acts as a connector by bringing together multidisciplinary teams to solve scientific challenges around risk assessment and safety. We also facilitate collaboration across academia, government, industry, and NGO scientists. Our committees produce impactful and evidence-based science for better, more informed decisions. Finally, the third point here is that it speaks to the translational nature of HESI activities. Our end goal is to move knowledge into application by creating and testing technology platforms and scientific frameworks that can be used more effectively to predict effects on humans or the environment. Next slide. So I'd like to go into a little bit more detail of the subcommittee that both Dr. Thu and myself are members of. HESI achieves their mission through these various subcommittees, and the one that we are on is the Cell Therapy Tracking, Circulation, and Safety Committee. This is a collaborative committee where scientists from all over the world encourage the development of studies that ask the right questions, structure the right framework, and develop solutions that inform decision-making. In terms of outputs of HESI programs, they can be easily transferred from the laboratory or journal page to real life and are used to enhance human and environmental health at local, national, and international levels. Next slide, please. So the nTRACK's leadership is chaired by Tineke van den Hoorn from the Netherlands, as well as Bill Shingleton at the UK. And furthermore, we have subgroups, called Points of Administration and Biodistribution, which is chaired by Brooke Helford, and another subgroup chaired by Dr. Turo in industry, Dr. Gerardo Bando, as well as Charlotte de Wolf in the Netherlands. The HESI staff that is involved in coordinating this subcommittee work is Cecilia Flores, NBC, and Connie Chen, also NBC. Next slide, please. As of September 2021, we have at least 39 organizations that contribute to the nTRACK's work. We have 10 academic or research centers, 8 government and regulatory agencies, 3 various consortia and non-government organizations, as well as 18 companies that include both developers and biotech. The public-private ratio here is about 21 to 18, or 54% public and 46% private. Next slide. So this is the organizational structure of the nTRACK's committee. And the idea here is to facilitate translation of cell-based therapies to the clinic by driving the development of tools, methods, and knowledge required to evaluate safety and fate of therapeutic cells. We have over 100 participants in this committee with more than 35 organizations. As I mentioned before, Bill Shingleton and Tineke van den Hoorn are the co-chairs of this committee. Within the committee, we have 2 subgroups, what we call point of administration and biodistribution and tumorigenicity. Together, we work across the world and in collaboration with Japan to carry out the work, both in cell tracking, education and outreach, research, etc. Next. I should have taken you through this whole thing. But this is in collaboration with the Japanese consortium. So this just talks a little bit about the education and outreach in terms of publications, scientific sessions, and webinars, As you can see here, we've done a lot over the past year. A paper has been published in cytotherapy. We've participated in the ISCT virtual group, as well as put on a webinar in terms of regulatory perspectives. Next. In terms of specific research, Bill Scott is heading up a joint studentship research project, putting together a multi-organ perfusion model. This multi-organ perfusion model, or MVP, contains 2 or more organs to enable the study of cell migration and fate in an isolated human perfusion model. This project infuses a primary organ of interest. In this case, it is the heart, I believe, or the liver, excuse me, and it's connected to interrogate primary and bystander organs with multiple different modalities. Finally, they characterize the perfusate in terms of using technologies such as MSD, RNA-seq, Hyperion, etc., to identify and understand shifts in phenotype in response to some of the MVP environments. Now I would like to just turn this over to Dr. Fu, who will talk to us about some of the clinical values of this field. Dr. Fu?
Thank you, Dr. Mother Lundberg. Hello, everyone. My name is Mia. Can you see the screen?
Yes, next slide.
Thanks, Dr. Lundberg. Hi, everyone. My name is Mia Thu. I will be continuing from here with the educational and outreach program that nTRACK is further doing and developing for the cell therapy developers and cell therapy industry. As part of the outreach effort and nTRACK's exercise, we spoke to more than 50 multi-stakeholders in the industry from the public sector and private sector, including physicians, regulators, cell therapy developers, and investigators, in order to understand better about how non-invasive imaging insight can benefit the work that they are doing in accelerating product development or helping the patient sooner than later in oncology application as well as regenerative medicine application. And this is what we learned from them. Imaging insights such as biodistribution can allow them to understand where the cells are and where they're going and how long they stay there, for example, to assess the safety. On target on disease specificity of these cells can help them determine the toxicity management. And cell type specific quantitative infiltration, they told us that will allow them to devise a dosing strategy. And last but not the least, persistence engraftment or targeting associated disease response can help them figure out, understand treatment efficacy. From here, I'll be giving you a quick example of use cases, how these imaging insights are contributing to the understanding of preclinical and clinical benefits during the research and product development. I will be showing the representative imaging from the clinical imaging modalities, and it won't be inclusive, but it will be representative. As the first insight, the point of administration and biodistribution information is extremely important in understanding the accuracy of cell delivery in the body, which is currently a black box, and which is what nTRACK is also contributing to the field and assisting in results interpretation. The image that you're looking at here, the left one, is the 19F MRI image of fluorocarbon labeled natural killer cells. And with the MRI, the investigator can figure out immediately or right away whether there is a successful delivery or misadministration and misinjection. And this is important because we want to make sure that the cells are getting to or delivered to where they are. On the right image, what you're looking at is the accuracy of cell delivery if the label cells are given intravenously. In this image, you're looking at homing or biodistribution of iron-labeled intravenously deliver MSCs in Alzheimer's disease model in red. The second insight is quantitative quantification of cell type specific on target, on disease specificity for safety and toxicity management and product development. In this image, you're looking at PET imaging and MRI imaging that is used to visualize the accuracy of cell homing at the disease site. The left image, you're looking at PET of 18F FDG labeled BMC cells. It is CD34 plus enriched cells at the heart disease. What you could see here is that the cells that are delivered through the coronary artery did home at the heart, but they are at the peripheral of the infarct rather than homing directly in the infarct, which is shown in the star, as you can see here. This allowed us to understand the cell homing and the timing of the product itself during the research or in the clinical trial. The right image that you're looking at is the MRI of iron-labeled neural stem cells in a patient with brain trauma. You could see that the injection site show up as the hypointense signal, dark signal, but over time it distributed within the brain. This is the information that we learned that the users can help in terms of understanding their safety and toxicity management or homing information. The insight 3 is the quantification of cell type specific infiltration that is given, for example, by intravenous injection of mesenchymal stem cells that are labeled and homing at the diseased joint that you're looking at in here. The left panel is the MRI image that is shown over 4 weeks time period where the cells are homing and infiltrating at the disease site over time with the red arrow, and the right panel is the optical imaging. This allowed us to determine or help design whether there would be the higher doses necessary or the lower doses, or how long you will want to wait for the infiltration information. Insight 4 is which is the current focus of the industry as cell and gene therapy is fast moving and exploding, which is, can we use early assessment of the cell homing or targeting at the disease site as the potential treatment response? And we don't have enough data at the moment yet, but learning it or borrowing it from the oncology space where tumor targeting of a particular cell therapy product has been shown, as you can look at in the image, within 2 days of their tumor targeting at the disease site correlate with the tumor volume reduction. The question is, can we borrow this and then apply to the regenerative medicine field as well? The image that you're looking at here is in regenerative medicine, engraftment or persistence of these cell therapy product and understanding this potential efficacy and treatment response. The top panel, you're looking at 2 patients that receive iron-labeled islet transplant in liver, and the bottom panel is that similarly. What you're looking at, or what the paper has shown, is with the engraftment, the signal loss is retained over time period, which means that when there is an engraftment or persistence, you could see the signal over a long period of time. when there is the graft loss, for example, the cells are not persistent any longer, you don't see such signal in the liver or in the transplant site. There is not enough data done yet in terms of correlating this information with the treatment efficacy, but the early assessment of insulin and other measurement has been done. And as with that, I'm highlighting a few use cases, and there have been CT, SPAD, PAD, MRI that has been used to look at many different cell types in many different diseases, and with many routes of administration, and they are a good set of data to do that. But I would like to highlight that one size does not fit all. Different imaging modalities address different questions by the cell therapy developers or investigators, and with different strength. And with that in mind, nTRACK and our group have been putting together the resources called Data Portal for Imaging Cell Therapies, where we curate all the publications, clinical stage, as well as the preclinical stage available in the literature for the users to be able to quickly and easily search. For example, the number of cells, the disease site, the injection site, as you could see it on the right side. We will be launching that in Q2 of this year. And if you visit this webpage, you'll be able to see the initial landing page there. With that, I would like to transfer back to Dr. Martha Lundberg, who will be closing this talk. Thank you.
Hi, can you hear me? So cell therapy enables very positive effect in clinical trials and translational science in order to benefit our patients. HESI is strongly involved in facilitating the build-out data-driven decisions, and then improving unanticipated safety issues, toxicity management, the dosing strategy, and product lifecycle management, as well as product fine-tuning. The clinical team works together through these data-driven decisions in order to treat the patient in a more effective manner. And this is using cell tracking methods that can inform the biodistribution, on-target and on-disease specificity, infiltration and persistence, as given by the examples from Dr. Wu, as well as targeting and associated treatment responses. And then these insights feed into the cell therapy product development, and again, back to the clinical team. Next slide. So NIH has supported a number of both cancer-based research as well as cardiovascular disease. And despite a lot of work going on in the preclinical area in terms of immunotherapeutics, the success rate, especially in the cardiovascular field, is somewhat clinically limited. So this is a key challenge for the field, is our success rate in clinical translation. So how do our scientific findings address these challenges? Well, the imaging approaches here allowed for temporal and non-invasive insight for in vivo behavior and plaque targeting, as well as its therapeutic effect on atherosclerotic plaque inflammation. Many of the imaging tools used in these approaches are already applied in humans. So this strategy can help with clinical translation. For us, in the NHLBI, some of the key gaps and takeaways with the atherosclerotic plaque progression and instability, we are hoping that MRI will lead the way in imaging modalities to improve targeted regenerative medicine therapeutics. Next slide. And I would like to just wrap up and thank the HESI nTRACK committee chairs as well as the subgroup working group chairs on clinical administration and biodistribution. Also, want to remind everyone that we have more than 100 committee members on nTRACK, and they span across the EU, Japan, and USA. Next slide. If you have any questions, both Mia and myself, as well as Cecilia Maurice, the point of contact at HESI, would be happy to respond to any questions or concerns you may have. Thank you.
Thank you both. It's excellent to see this truly international collaboration, particularly in this field. Now we're moving on to the final Q&A section of the day. My first question is for Marta Rayo. So the question is, what was the most unexpected regulatory hurdle that you faced under the nTRACK project?
Can you hear me fine?
Yes, we can.
Okay. The most unexpected hurdle, with no doubt, was the difficulty in classifying the product. This is something that even not unexpected in the European scenario, because we have several authorities, several notified bodies, we have national authorities, EMA, the European Commission, and so many players. They cannot be completely harmonized all the time. So it's something that could be somehow expected. But the difficulty we face in the reality that we are almost ending the project without a clear picture of what would be well accepted by all authorities is for sure the most unexpected hurdle.
That's great. Thank you very much. Our next question is for Dr. Dinez. And that question is, recently the first pig xeno heart transplantation was performed in the USA. How is iNanobit xeno-islet work progressing towards clinical stages?
It's an excellent question. Indeed, the great news from the States, I think it's for 2 months survival. I know the patient died very recently, but I think the 2 months survival of a full heart transplant with a big heart, it will really catalyze the whole field. I'm very close to my heart because literally I was working on a cloning team in Roslin Institute in 2001. We were the first to clone a piglet in Europe, 3 months after the American number, first of the world. So this is continued to be a great scientific challenge and competition. And I think that kind of competition can really stimulate also the regulators and all others. Don't forget, this is a multi-billion business in the creation right now in regenerative medicine. So I think we are living exciting times, and great stories like the American heart transplantation will stimulate not only xeno, but also other type of human regenerative medicine. So I'm looking forward also with the xeno islets to transplant them.
Thank you very much. And a question to Dr. Lundberg and Dr. Thu. So how many stem cells or immune cells are you able to detect in clinical imaging modalities, MRI and PET?
Thank you, Adam. Thanks for a great question. So in the stem cell field, there are many different stem cell types. Investigators as well as the clinicians are using. So it would depend on the type of the stem cells as well as the particular system and a particular organ and location. So what has been shown is that, for example, for macrophages at the 3 Tesla MRI, you could see as few as 60 cells per phage cell. However, once you go into a smaller cell type, like say, for example, hematopoietic stem cells or T cells, then you need a larger amount of number of cells, for example, 10,000 or 100,000 cells, depending on which imaging modality specifically and the strength of the MRI. I hope this answers your question.
That's great. Thank you very much. Thank you. So, unfortunately, that's all the time that we have for today. I'd like to thank all of our brilliant speakers. And of course, I'd like to thank all of the attendees that have taken the time out of their day to join us. If you want to have the latest developments, I recommend that you come and visit our website, n-track.eu, or alternatively, come to projects.latat.org. Hope you have a nice evening.
Recording stopped.